spring, and this is the one. So those of you who have participated before, we're glad you're back. And for those that are new to this, I hope you will have an enjoyment today with the summit. So we are trying to, what we've done before, the program will be highlighting academic innovations in the drug discovery ecosystem across a number of different people, again, across the country. We began our series in 2018, and we continue to, we'll continue to have these. Hopefully in the fall, maybe it will be live, and so I'm sure everybody would like that. I would also like to welcome our newest member to the Arizona Center for Drug Discovery, co-director Dr. Selena Hsp90. And so she is going to welcome you and then provide some information as we start the summit. So again, I'm glad everybody is participating. I'm glad you're here. I hope all is well, and I hope all of you are vaccinated. So anyway, so Dr. Hsp90.
Thanks. Thanks, Dr. Norman, for the introduction and your continuous support to ACDD. I'm going to share my screen. Hope you can all see it. Thank you all for attending today's program. As the Dean mentioned, I joined the College of Pharmacy and ACDD 6 months ago, so I'm relatively new. From a background of 15 years in pharmaceutical sector. I was recruited in 2005. I was recruited out of my postdoc at Washington University in St. Louis by Munk Research Labs to join a newly formed Alzheimer's department. At Merck, I was given my own lab space. I hired and trained a very fine group of biologists. A lot of them are still there, and led multidisciplinary drug discovery teams for almost 10 years. Primarily in the neuroscience area. I worked in all stages of preclinical drug development, from target validation, lead ID, lead optimization to preclinical candidate selection. But after going through multiple reorganizations, and I have to say Merck was a great place to be and learn all drug discovery, but too many reorganizations made me decide to explore new opportunities. And I took an offer from Evotech to lead a large collaboration with the CHDI Foundation on Huntington's disease. I moved to Hamburg in Germany, and where I also led multidisciplinary teams working in drug discovery projects, again, target validation, IPS screens, lead ID and lead op, but also some more technical projects such as library management, which I didn't know anything about, but it actually comes very handy now. And also protein production and development of new technology that was coming out of academia. It was a fantastic opportunity to learn from CHDI how to integrate academic creativity with CRO industrialization. But it turned out that I also had some creative ideas of my own, which attracted some VCs in biotech. So I left Evotech in 2017 to explore this arena. I went from Hamburg to London to Boston to San Francisco, and finally made my way back here in Tucson in 2019. Actually, I was in Tucson many, many years ago. And always wanted to come back. It seems to happen a lot to people that come through Tucson. So I initially joined Regulonix, which for those of you who didn't know, is a spin-out of biotech spin-out of UA as a consultant before taking the position here with ACDD in October last year. So I think that's enough about me, and with no further ado, I will present a short overview of what we're trying to do in the Arizona Center for Drug Discovery. And I hope that you get excited to join us in the near future. So as stated here, our vision is to become an innovative and sustainable drug discovery hub at the forefront of academic translational research, which is an ambitious vision, but of course it's always good to have an ambitious vision. We also, our mission then is also ambition, which is to foster a collaborative and academic environment to rapidly catalyze development of differentiated and first-in-class medicines for unmet medical need, guide translation of biomedical findings at University of Arizona into future operational drug discovery programs that can be partnered with pharma or attract investors to form new codes. We also seek to cultivate external relationships with pharma and venture companies to build an innovative and diverse portfolio that's aligned with market demand. So how do we get to deliver on our vision and accomplish our mission? Well, first by integrating already available U of A resources to build the infrastructure and operational framework required to do drug discovery. We have multiple centers and colleges that contribute to both physical and human resources and financial resources as well to ACDD. As shown here, all of them are shown here, and several of the lead representatives of these colleges and centers are part of our SAB, of the ACDD SAB, and they are there in a position to provide input and also disseminate our goals and initiatives across the multiple centers and colleges. In addition, the College of Pharmacy has committed to expand its drug discovery efforts with the construction of a brand new SCAG building. It's a fantastic building with state-of-the-art facilities and a commitment to hire 6 new endowed chair positions in drug discovery, 3 of which have been already accepted, and some of them are already joined in the past 7 months. So Dr. Thatcher, Dr. Lee, and Dr. Zhu are highly involved in translational research and will be and are part of our ACDD team. My key immediate goal when I joined ACDD was to build an assay development core to provide faculty with access to industry standard expertise and resources to run successful screening campaigns. For access to physical resources, we work closely with the University of Arizona Functional Genomics Core, which is managed by Matt Kaplan, which many of you know, and where multiple high throughput plate readers and robots are available for usage at a recovery cost. And we were very fortunate to recently hired within ACDD, Charlie Chen, a highly skilled scientist from local pharma. So he started at Selective, and then which became Sanofi, and then Icogen. And he is going to then play a key role, hands-on role, in fully establishing the ACDD core service in the coming months. Another immediate goal to support these screening efforts was to procure a new compound collection. While the ACDD had acquired the Spectrum library, which includes FDA-approved drugs and also drugs approved in Europe and Asia, we wanted to build a curated collection that would allow researchers to identify new and patentable chemical matter. So we procured a library from ChemDiff with 35,000 compounds, but this library is actually made of subsets that allow for more flexibility in terms of costs and project focus. So we can see here we have a PPI bias set, a CNS bias set, a diverse set, a non-annotated set, and a fragment set, and a covalent set. And of course, these libraries differ in terms of properties here. Just an example, in polar surface area, you see the CNS has compounds with a lower degree of polar surface area, which is important for CNS penetration. ACDD also keeps software licenses to support screening campaigns. So we provide license of seats with the CDD Vault. So it's a database and assay data analysis software. And we also maintain license with Schrödinger for virtual screening and docking. We're also strengthening our medicinal chemistry core, which is led by Dr. Wei Wang, my co-director, in terms of both physical and human resources. So there's new equipment that has been recently acquired, including the microwave reactor and a brand new nuclear NMR spectrometer, which then will provide increased capacity of our new chemists to support ACDD projects with probe synthesis, heat optimization, heat structure activity relationships efforts. Oops, sorry, I missed my presentation. In terms of organization, we have created a functional core team comprised of U of A faculty with significant drug discovery expertise in key areas. So in addition to the co-directors, Dr. Wang, who brings a medicinal chemistry expertise, and myself covering assay development and screening, We have the additional 7 core teams that are shown here with specific skill sets, in addition to having extensive drug discovery experience. These specific skill sets provided by the faculty are of great benefit to the faculty that join ACDD in support of their programs. With our core team, we have established a process by which we provide support and resources to drug discovery projects. Our goal is to reach out to investigators, identify novel ideas that can be translated into new treatments. The initial consultation period will depend on the stage of the project. And once enough evidence is gathered, the PI presents a proposal to the core team, which then assigns and prioritizes resources to resources over the other projects. We gauge probability of success primarily based on commercial impact, project feasibility, and funding prospects. We have started reviewing projects in this new format as of February 2021, so just a couple months ago. We have 2 forums established at this point, the ACDD core team review meeting, which reviews all drug discovery projects on a monthly basis with the goal of providing feedback and guidance on assay development and screening strategy. And our second project review forum is part of an initiative with the University of Arizona Cancer Center, and that's headed by Dr. Joanne Sweezy. The goal here is to focus on cancer projects that align with the center strategy and prioritization. And the prioritization of these projects would then be tied to pilot fund that's provided by the Cancer Center to use towards assay development and screening efforts with ACDD. And I would like to thank Dr. Sweezy for her support to the center with this opportunity. So finally, I come to the last slide, which is really basically showing what we want to do. It's building a pipeline of drug discovery where we have a continuous flow of projects moving further to more commitment from advancing to screening and for selected projects, and then moving further to more committed medicinal chemistry efforts in order to generate in vivo proof of concept. Our metrics of success will be many. For example, publications and grants, drug candidates, intellectual property, pharma partnerships, licenses, and spin-offs. And to work for guidance with some of these opportunities, we work closely with our partner in the Tech Launch, Arizona, more specifically Rocky Gibbons, our Director of License. She has been very instrumental in providing ACDD and its members with a better understanding of how to protect and commercialize the University of Arizona assets. So I finally come to the slide where you can find more about our website by visiting the ACDD website under the University of Arizona College of Pharmacy. There we will find also a new white paper that we put together, which summarizes our services and resources. And finally, I would like to thank our contacts here, myself, and Wee-Won, the co-directors of ACDD. And I'd like to thank immensely the help of Ali, Director of Communications, Tony Schlack, Director of Information, and most of all, Liz Miller, our executive there associated to the Dean, who really supported that facilitate this event. I thank you. I would also like to take the opportunity to thank all speakers for agreeing to take the time to participate here today and making this event a huge success. We have 150, we had 150 registrations, which I think might be the record for this event. So thank you so much. And I will look forward to listening to your presentations. So thank you for your attention, and let's get to this exciting program started. Before we go on, attendees, you're invited to submit questions using the Q&A at the bottom of your screen via the Q&A feature during each speaker's presentation. We will attempt to get to all your questions, and all unanswered questions will be collected, and responses will be recorded and shared with attendees after the summit.
Thanks, Selena, for your that nice overview of the ACDD. We have very exciting the things happening, and good morning, everyone. It's my great pleasure to co-host this morning section with my colleague, Professor Greg Zetcher. So we have five distinguished speakers. The first one is Professor Khanna, my colleague here at the the Department of Pharmacology and Medical School. Roger, he is a professor of at the in the Department of Pharmacology at the U of A, and he is also the chief scientific officer at the Regulonix. Is a Tucson-based biotechnology startup company focusing on the developing non-opioid, therapeutics to treatment of chronic pain. Roger, received a PhD in physiology from University of Toronto and the postdoc at the UCLA. He has been, really, impressive, doing really impressive research work, basic and translation research work. And his research focuses on the disrupting pathologic protein interactions with using biologics and small molecules and to modulate ion channel activity in neurodegenerative disease and pathology. So that with that, please welcome Professor Khanna. Roger, could you? Okay, go ahead. Yeah, great. We can see your slides.
Good morning, good afternoon. Thank you very much, Dr. Hsp90 and Dr. Wong, and thank you, Dr. Schnellman, for setting this up. It's a great pleasure for me to talk again at this symposium. I talked at one of the early symposium in 2018, where we talked about some of the work for my startup company, Regulonix. So today, I'm going to take a little page of from serendipity and innovation, combine them together, and tell you about a story that we've uncovered in the past 6 to 9 months, and how quickly this has really become something that we're very excited about. And before I do that, I'd like to just give you a flavor of what the lab does. We are indeed an academic lab doing academic style drug discovery, which is very small compared to the big pharma and some of you folks this afternoon. But our main focus is to bring mechanism-based research to the clinical doorstep. And we've been doing that in a variety of collaborations. We have, and this is probably news to Dr. Wong, we have 2 newly funded R41 SCTRs to target a protein-protein interaction. So congratulations, Dr. Wong, targeting CAV alpha beta. And we have a burgeoning collaboration and a grant with Dr. Jun Wong to take some of his compounds, viral inhibitors, to see if they would be important for chronic pain. As you know, chronic pain is a big problem. The field is really dominated by, unfortunately, opioids, which have many, many side effects. So the key for our lab's effort is to be designing drugs for chronic pain that are non-opioid based. So we rely on, for example, the Sonoran Desert for non-opioid based natural products. We've collaborated with other members of the campus, College of Medicine in anesthesiology, with Dr. Mohab Ibrahim, who's been designing a very innovative approach to use light as a way for chronic therapy. And as Dr. Wong was saying earlier, my lab's efforts really have been to target protein-protein interactions, and we have a couple of R1s to dissect some key contributions made by an ion channel that I'll talk to you about today. However, today's talk is really going to focus on this really mysterious and surprising discovery that we made of analgesia caused by SARS-CoV-2. Yes, I said analgesia. So this virus causing pain relief, and I'll walk you through some of that. So why are we working with chronic pain? First off, why haven't we been able to solve chronic pain? Well, problem with chronic pain, as with other diseases, it's a constellation of different conditions that lead to chronic pain. Some of these are shown on this slide here, and you can appreciate that there's quite heterogeneous distribution, which means that there are probably multiple targets that contribute to this. And the other harrowing feature for chronic pain patients is that you have, despite knowing chronic pain for so long, patients often wait about 10 years or so to get their condition diagnosed. And only for a handful of these major diseases or pain conditions is there some level of regulatory guidance for pain management. So it's a lot of unfortunate cases that this has still not been solved. The reason why it's really not been solved, chronic pain in general, is because of the homogeneous or the heterogeneous nature of the targets. There are so many different targets that contribute to pain processing. In a very simplified example, here's the idea that pain may originate from both the peripheral nervous system and the central nervous system. There is something to be said in the brain processing the effective components of pain. But it's been shown very emphatically that even in peripheral sensory neurons, these so-called nociceptors, or the harbingers of the neurons that initiate pain processing, you have excitability generated through these neurons. And this is sufficient to confer the chain of events which lead to chronic pain. If you take that and deconvolve that and look at some of the major players, and that's shown in this slide, what you see is that noxious stimuli of various kinds, thermal, heat, mechanical, and so forth, are sensed by a constellation of ion channels and receptors. And really, a critical channel here is a voltage-gated sodium channel, which that basically sets the threshold of the firing of these ion channels and the ensuing relay of signaling that happens. This then triggers recruitment of other channels along the axon, particularly those in the sensory neuron, which is where the pain sensing cells and the ion channels and receptors are, the dorsal ganglia. And then this translates into transmitter release through opening of another channel that's super important in pain, which is a calcium channel family. And it's really the interplay and the balance of interactions that these channels have that determine chronic pain. For example, the importance of 1.7, this voltage-gated sodium channel that sets the action potential and the contribution of the upstroke of the action potential. This is really a very key channel because mutations in this channel, both gain and loss of function, have equivocated this channel to be clearly linked to pain. So how does one start to study these channels and try to target them? Most pharma and big academic labs have focused on individually trying to block some of these channels, particularly the voltage-gated sodium channel or the calcium channels. And this has been met with very limited success given that these channels are very homologous to each other, as well as there are other side effects because of direct targeting of this channel. Because you want to preserve physiological pain, which is important to live, but you want to curb pathological pain. So given these challenges, what our lab has done is sort of stepped outside our comfort zone and tried to look at the conversation that these proteins are having in the intercellular milieu to find additional interacting proteins that can then regulate these proteins. So wouldn't it be awesome, wouldn't it be great to have a protein that can regulate both this very important channel which starts the signaling of pain and also, so electrogenesis, but then also have a channel or a protein that can regulate, modify somehow, and control the activity of these channels, the calcium channels, which regulate transmitter release. So with one fell swoop, you could control both events. And you can do this through many different proteins. My lab about 15 years ago discovered a protein called collapse and response mediator protein, CRIM2. This protein is pretty interesting because of its pleiotropic functions, and it was initially described for its role in axon growth and collapse. And we'll come back to this in a second. It's got some roles outside the nervous system, in particular driving lymphocyte migration in MS patients, for example. It may contribute to pain signaling there. And my lab really demonstrated novel roles in terms of neuroprotection in the face of excitotoxicity or stroke. But today's conversation and talk is going to be really focusing on trafficking by CRIM2, the regulation of trafficking of these key ion channels I've been introducing to you a few minutes ago. And so over the last 10, 12 years, my lab's published a series of papers that have described a novel platform for the trafficking of the voltage-gated calcium channel. This channel is the known target of a couple of pain drugs, for example, gabapentin being one of them, or Prialt being the other one. And those two are direct blockers and have some problems and side effects. So our approach has been to understand the dialogue between CRIM2 and this channel. And this interaction leads to excess calcium currents, which can be blocked by designing a biomimetic peptide that can interfere with this interaction, or through small molecules that modulate some of the post-transition modifications of this particular protein. Importantly, in terms of the sodium channel, which is the harbinger of the action potential, we've delineated a program where controlling phosphorylation and simulation with small molecules or through genetic approaches, we can really nucleate an event that results in internalization and therefore has an effect on sodium channel trafficking. So this is really what small molecules designed against this pathway is what I had presented a couple of years ago. And we've been working on this and moving this company, Regulonix, which is funded by several STTRs and a grant from UA Venture, Fletcher McCuskers group. And so we've been marching along that. And then, as you all know, this happened. Nobody wants to hear more about it, but we're going to talk about it. And I'm going to switch gears to telling you how, from this COVID fog, we have come up with a new target, a balance of innovation and with a touch of serendipity. And so very early on, we were being inundated, everybody was being inundated by all kinds of reports coming about of what's happening with SARS-CoV-2. It's causing this constellation of symptoms, and it's devastating. A big part of that was, and surprising early on, was the fact that there was a large fraction that had asymptomatic acquired SARS-CoV-2, this COVID-19 virus. And at this asymptomatic stage, we didn't know why people were still able to transmit the disease. So that was a sort of a head scratcher. And more and more has now been figured out and been reported as to what's happening. But we wondered at that time, very early on, whether during this asymptomatic stage, the reason people are able to transmit this disease, because they think they don't have it, is because perhaps in the early stages, before the immune system has a full-blown reaction, perhaps in the early stages, there's some bit of dampening of pain. So there's a resilience endowed by the virus. So I'll be glib about it and say, perhaps the virus is giving you a gift in terms of, well, a double-edged gift in fooling you with pain relief. So as I said, as we now know so much more about COVID, and still it's the tip of the iceberg, we now know that there are neurological complications of COVID-19, and various thoughts are as to why this is happening, rearranging activation, plasticity mechanisms, microglial activation. There's a huge component, obviously, as one would predict from this kind of a virus, immunological component. And you can see a lot of different things have been reported. Whether or not pain, either more pain or less pain, it's really not, it's been controversial. There are reports now of pain being reported in COVID patients. But I'll have you focus your attention on the left panel of this left side of the slide and tell you that there have been sort of anecdotal reports, and this is a published report, where people with chronic pain, for example, this is a patient, a 70-year-old patient that was hospitalized due to the fact that he contracted COVID-19. And this patient had also chronic pain. But during the time that they contracted COVID, their chronic pain was gone. And at about 1 month follow-up, the patient had recovered from COVID-19, but their pain was still, was starting to appear back. I've been contacted by various people, and I'll show you why, based on the story we just published, from across the globe, people from the US, people from Canada, people from South Africa, where if you just scan through this table briefly, almost all of them have some sort of chronic pain, you know, through fractured vertebrae, through some sort of genetic diseases, musculoskeletal pain, arthritis, and diabetic neuropathy. In all of these people, they so happened, so they had pre-existing chronic pain, they contracted COVID, and if you scan through here, their pain is gone while they had COVID. Pain bearable, no pain whatsoever, no pain. And in some cases, it's remarkable that pain is still gone in these patients. So this is something very surprising. And so we kind of, you know, being a lab very interested in chronic pain, we thought maybe we could learn something from SARS-CoV-2 virus itself. And so we started to kind of think more about it. And so as with the cliche moment of eureka, I think we had one of these moments of eureka for this project. So so far it had been known that SARS-CoV-2 enters the nervous system and enters our bodies through this ACE2 receptor. And this has been sort of mapped out both physiologically, both structurally, and there's a lot really known about this now. And but at odds with this entry through of the virus through this were a couple of findings. First, that particularly from the Italian population, where we noted a lot of death early on, in the Italian population, both the elderly and later the young people who contracted COVID-19, it was surprising that they were contracting it because these groups of people have lower amounts of ACE2 normally. So how is this happening? Because if there's not enough ACE2, so how's the virus entering? Entering these cells. And the other thing was ACE2 is not expressed on all sensory neurons. So we didn't really know whether any kind of influence would be on pain or not. So this then was ripe for discoveries of other doorways, if you will. And there were two beautiful papers in Science in the early to mid last year, which showed that neuropilin could be a new factor or new doorway for SARS-CoV-2 to enter. And this is where we had our moment of eureka. Because as I've been telling you, our lab had been looking at protein-protein interactions, focusing as one example on this cytosolic protein called CRIM2, collapse-end response mediator protein, which I showed you had associations with both sodium and calcium channels, and those interactions could be blocked to curb pain. So sitting upstream of this massive pathway is this receptor on the membrane, neuropilin, which was just discovered in those two papers to be another doorway for the virus to enter. So we wondered if NRP1 had some contribution, perhaps through this signaling to pain. A little bit about NRP1. It's a complicated protein, lots known about it in the context of cancer research predominantly. It's organized into this kind of structure, and it gives you an idea of what the binding partners are. You can see there's a lot of heterogeneity in what these binds. And so this was initially, we thought it'd be a challenge, but we then used a nice strategy to whittle down and see which ligands were important. I can also tell you that NRP1, much like the sodium and calcium channels, is in the right location in the nociceptive system. So for example, if we look at transcriptomic data, if you look at mouse and human dorsal root ganglia, the cells that house the pain system, you can see that this protein is expressed, the mRNA for this protein is expressed in human sensory neurons. It's also expressed in the immune system, and that's really what it's been targeted for in the context of pain. But we wondered what was the relationship between neuropilin and this particular pathway. We had already shown you, and we've been working about this with this CRIM regulating ion channels to curb pain for the last 15 years. And now we noted that neuropilin had a partner or entryway for SARS is upstream of this protein. So we wondered whether it's through, whether through coupling to some of the co-receptors, whether neuropilin could be a new player and contribute to pain. To really get to the heart of this, Dr. Motao, who's an assistant professor in pharmacology, had a great idea where he looked at firing frequency of nociceptive neurons. And we had a different palette of ligands because there are many different combinations of neuropilin and other co-receptors. So we wanted to figure out through which receptor the signaling was happening. And in order to do that, we incubated these neurons with, for example, somafrins to initiate pathway through this complex, or through VEGF-B to initiate this pathway. And really, we found that only when we stimulated with VEGF-A, vascular endothelial growth factor, do we see sensitization or enhancement in excitability, which could possibly lead to pain. Just to give you a little bit of background on VEGF-A, this has been known for probably a decade to be important in pain. Both mRNA and protein of this particular for VEGF-A and its receptor are upregulated in forms of, in models of forms of injury. And if you target some of these pathways pharmacologically or genetically, you can ablate the pain responses. And this has been shown in different models of injury. And an antibody against VEGF can also bring about a reduction in some of the experimental pain. So what we did then is to use structural information. So this is the structure of the B1 domain of neuropilin, which engages in this pocket VEGF-A. Remarkably, this is the same area where piece of the spike protein from the SARS-CoV virus also binds. And because I had told you that neuropilin had been investigated in the context of cancer, there are several drugs that are already available, among them a compound called EG229 that also happens to bind this in this region. So using this and putting all this together, we know that VEGF-A is exciting the cells, it's making them hyperexcitable. So we now wondered if that excitability could be blocked by a piece of the spike, since it would compete with binding to VEGF or with this particular compound. And exactly what we saw. So this is pretty exciting to us because this showed that the spike protein here was having some effect on pain relief. And to put it all together, we are able to find a ligand that prioritizes signaling once initiated with VEGF-A, and it may work through CRIM2 to then regulate the activity of these two channels, which CRIM2 is already known to do, and then perhaps that would be how we could achieve a reduction in pain by suppressing excitability and transmitter release. So it's a pretty exciting new pathway that we uncovered because of the upstream regulation by VEGF through neuropilin. So in how do we prove this in an animal? So we took naive animals, and I told you VEGF can induce pain. So in naive animals, we can assess pain by measuring their tactile withdrawal. And so sensitivity to touch. So with VEGF, naive animals have this pronounced pain. So lower the number, the more pain. They have pronounced pain, which lasts over the course of about 20 minutes. And this can be blocked with spike. So that's this right here, or with the inhibition of NRP1. And you can clearly see the cumulative effect of the sensitization and the block by spike. So this was seen in this relief of pain, the relief of this mechanical allodynia was seen in males, in females, and also for a thermal measure of pain. How does it work? We had told you that it works through regulation of ion channels. So VEGF is able to facilitate an increase in sodium currents as well as in calcium currents. And these two can be blocked by co-incubation with spike protein or with the NRP1 inhibitor for both sodium and calcium. So this really then navigates back to a pathway initiated by NRP1 and VEGF-A that can be blocked through blocking these channels, which is what we've been interested in all along to curb pain. At the next level of the relay, beyond the spinal cord, is the slice. So a graduate student in the lab looked at recording excitatory neurotransmission, looking at these potentials, the excitatory postsynaptic currents. And you can see again, VEGF can, VEGF can potentiate this excitability and firing frequency, which can, with inhibition directly of the NRP1 inhibitor or the pathway, or spike can inhibit and normalize this excitability, so to dampen pain. And finally, in injured models of pain, so we can initiate pain in animals with injury. And when we do that, we can see that if you take animals that are in pain, so that have very quick sensitivity to touch, they can be dose dependently reduced by spike or by inhibition with this NRP inhibitor. So one last proof to show that this protein is important, we edited it using CRISPR-Cas9. So you can see a loss of the receptor, and you can see now if we try to amplify the current with VEGF-A, we don't. In the control case, we can, with a guide RNA, scramble control CRISPR, we can see amplification or potentiation of sodium. But in the case where we have neuropilin knockdown, we have no sodium potentiation. The same thing happens with calcium. So this is a bona fide target. And having identified it, now we turn to crystallography with Dr. Perez-Miller to take advantage of this binding pocket I referred to. And what she did was to dock about close to 500,000 compounds, particularly some from the Natural Compound Library. And we found that there are two modes of binding that the compounds adopted, shown here. Some of the series of chemical structures, the top 20 hits are shown here, and these were selected and were helped by Dr. Marcel Patek for these studies. So we took some of these compounds and tried to validate them. So the first assay was to simply look at disruption of the interaction using an ELISA. And remarkably, even from the first round, remember, no SAR has been done, just the first round, we were able to come up with sub-nanomolar. So we're already at the 0.6 nanomolar in terms of inhibition of this interaction. So pretty promising. Does it work in cells? This is an experiment done with Dr. Motal, where he looked at an autophosphorylation of the co-receptor for neuropilin, the VEGFR2. And so if you put VEGF-A, the receptor is autophosphorylated, and you can detect that with an antibody, as seen in this in-cell Western. And if you do the same experiment in the presence of spike or the inhibitors, you expect this to be diminished. And that's exactly what we saw with all of our compounds, validating then that this interaction can be uncoupled in vitro, in cells. And finally, and most excitingly, as we saw, we can block the potentiation by VEGF with this particular compound. And if you take this compound, remember from our first virtual screen, no SAR has been done. This compound, given intrathecally into an animal that has pain, can relieve pain with one intrathecal injection for at least about 4 hours, whereas the animals injected with a vehicle control stay in pain. So quite amazing to put all this together. Serendipity innovation came together to really help use SARS-CoV-2 virus to identify a new target, neuropilin-1, for pain, and it's inhibiting it with these new compounds is showing promise for pain relief. But not only that, remember, this is a virus. We're in the midst of having antibodies, and everybody's hopefully vaccinated. But small molecules could be still useful for other viral infections. And so Sam Campos's group over in campus tested the ability of this surrogate virus to infect cells and to see whether our compounds would inhibit that. And we see at least 2 of our compounds are indeed able to also do that. So in addition to their potential use as potential anti-pain drugs, they could be used for anti-cancer. And finally, we're now build a model, pharmacophore model, to take all of these binding modes and start to go after the next round of SAR. We were just awarded an STTR. Dr. Wei Wong is an ACDD will be working with to get the compounds made. And so I'm going to stop there and give you a wrap up. As I said, we've been working on chronic pain and designing non-opioid drugs for chronic pain. We took advantage of serendipitous discovery of neuropilin causing pain relief, SARS-CoV through neuropilin causing pain relief. We've now come up with first-gen inhibitors, which are already in the nanomolar range, that are acting as small molecules for inhibiting pain. They may also show some utility in cancer. We plan to pursue that with people across campus and the US to take these into the cancer model. And I'll leave you with the most important slide. I want to acknowledge the disclosure for Regulonix and to tell you about all these wonderful people. It really takes a team to do this fantastic work. I'm just a spokesperson. Dr. Mutao is a recently minted assistant professor in the department, has been spearheading a lot of this beautiful work. And I'm going to stop here and see if you have any questions. Thank you for listening and thank you for your time. You're muted, Dr. Wong.
Oh, sorry. Yeah, thank you, Rajesh, for your wonderful, the research and achievements. So there is one question for you. Let me read for you. Spike proteins are used by the virus to enter the cells, and you are showing that spike proteins can trigger the signaling pathway that dampers pain. Is there a hierarchy of the events that happens, either signaling or entry?
Thank you. That's a great question. So that's exactly the point we were faced with initially. We had neuropilin receptor, which coupled to many receptors on presumably the cell of interest. And really, what we had to do is to figure out ligand prioritization. So once we figured out that VEGF-A was the only one initiating the pathway, then we focused our efforts on VEGF-A. We do not really know currently as to what the sequence of events are. We know that neuropilin or VEGF activation through neuropilin talks to CRIM2, which can then have activity changes on the channel. We know that is the sequence, but we don't know in terms of hierarchy. We imagine that CRIM2, because of its post-translation modifications, will be able to direct traffic towards the channels. And perhaps this modification is what is driven by activation by the neuropilin VEGF2 combination versus neuropilin and SAMA or any other combination. So that's really the current thinking.
Yeah, second question is, sub-nanomolar inhibition is quite impressive. Based on your molecular model, what do you think is driving such high affinity interaction?
So first off, that is super high in the first SAR to have that much of a sub-nanomolar. I'm going to attribute that to the brilliance of Samantha Perez-Miller, who's a fantastic computational person as a crystallographer that we've been happy to work with, and also Dr. Marcel Patek, who is exactly what this whole venture is all about. ACDD, talking and having these conversations with people who are in industry. So Marcel is president of Bright Rock Path, and he's been instrumental in bringing his knowledge of 25 years plus in pharma. So I think that's a big reason for why we're already there so fast, because they really did the docking and modeling, I guess, at a very high level. But we suspect that the interaction is also quite high affinity. So we have now resorted to means like microscale thermophoresis, and my wife's group, Dr. Mike Khanna's lab, is going to look at through SDD-NMR and through more sophisticated methods to really get and pinpoint the kinetics and the biophysics and really figure out whether these are real and if we can go lower with our SAR.
Okay, thank you. Yeah, third question, you have a lot of questions. Yeah, so third one, are there any suspected liabilities that could come with the inhibition of this NRP interaction with VEGF-A for chronic pain?
Well, thank you, Ken, for that question. He has an inside scoop on it because we've been having conversations with UA Tech Launch here. And so, yeah, there are definitely liabilities. It's because VEGF-A antibodies have already been used and they're available in the clinical space, but they tend to cause some neuropathy. And the reason for that is the VEGF-A story I've been telling you is not as simple as how I laid it out to be. Indeed, VEGF can provoke pain, but there's extensive splice variation that happens for VEGF-A. And one of the splice variants is pro-nociceptive, while the other is anti-nociceptive. So it's really the potential complexity and the toxicity could come, as it has for the VEGF antibodies, could be if you get that balance wrong between the pro and nociceptive forms of VEGF-A. So we are working to go around that problem.
Okay, the first question is, could the spike protein cause vascular defect as VEGF-A/NRP1 signaling? It's important to maintain, vascular homeostasis. Also, could you comment on the vaccine using spike protein?
Yeah, so those are excellent questions. I'm out of my depth here. We have not started to explore the effects of this pathway in the vascular context, but I really want to do that because you can appreciate that could lead to some serious effects. As far as the vaccine, I would rather not comment. It's too early for us to tell, but I can reach out to the person at a different time. Thank you for the questions.
Okay, yeah, another question. That is the pain dampening by the virus in chronic pain patients reversible?
Yeah, so this is really now what we're hearing a lot more. I've been getting emails from a lot of people. So to answer your question, the pain dampening in the people that got virus is indeed reversible. In some cases, in actually more than half of the cases, unfortunately, the pain has returned. And in some of the remaining lucky ones, if you will, the virus has gone away, but the pain is also subsided. And these are anecdotal reports. We're trying to collaborate with groups here to figure out this and really study this. So stay tuned. This is a pretty exciting sequence of events, if you will.
Okay, thank you, Roger, for your wonderful talk, answer great questions and very beautiful work. I really like it. Thank you.
Thank you very much. Thank you.
Okay. So let me introduce the second speaker, Professor Brian Blagg, from University of Notre Dame. Brian, we have known for a while, and actually we first met in China in one meeting. I know his work for a long time, working on the the Hsp90, 70 is really the exciting and important targets. After he makes this target become more popular. Brian is he received a PhD from Utah. He's a hardcore synthetic chemist, and did a postdoc with Delborg in Scripps. And then he started his independent career in 2002, and starting the University of Kansas and medicinal chemistry extremely well, and remaining until 2017. And then he was recruited by University of Notre Dame, and he will become the director of the Warren family, the research center and the drug discovery development. He is also the the Chinese Huisking professor in the chemistry and the biochemistry. And I say that Brian research focus on the design, synthesis, and evaluation Hsp90 inhibitors and 970. I know that I just saw yesterday he published nice work in Oncology chemistry, developed selective Hsp90 inhibitor. Very impressive work. So please join me. Welcome to Professor Blagg.
Thank you very much, Wei Wang. I really appreciate the introduction and the opportunity to give a talk here today. So for all those involved in this, I greatly appreciate the opportunity to kind of give you some updates on some of the research we've been doing, including some work regarding collaboration down at the University of Arizona. So as Wei mentioned, the title of my talk today is Chaperone Modulators for the Treatment of Cancer or Neurodegeneration. And so the majority of this talk is going to be really focused around Hsp90, and Hsp90's kind of got a bad rap, and for a good reason. I think as a society or medicinal chemistry group, we went after Hsp90 the wrong way. And let me explain to you how we did it the first time and how I propose that we change it to pursue Hsp90 as target, not just for cancer, but for many other diseases as well. So let me just clarify by starting off that there are 4 Hsp90 isoforms. Alpha and beta are both found in the cytoplasm. Beta is constitutively expressed, whereas alpha is inducible under heat shock or high temperature. GRP94 is localized to the ER, and TRAP1 is a mitochondrial resident protein. They have an internal ATP binding site that's very unique. It belongs to the Burgert fold. So there are only a few proteins that actually bind ATP in this bent conformation. It belongs to GHKL family of proteins, and so it makes Hsp90 a druggable target. The problem is these 4 isoforms share greater than 85% identity, okay, which is significant, meaning that it's very difficult to selectively inhibit one of the isoforms. And so this leads to the fact that there were 17 molecules in clinical trials that target Hsp90 for the treatment of cancer. And what they exhibited was a pan-inhibitory activity. That is, they inhibited all 4 isoforms with similar affinity due to the fact that they're 85% identical. One of the consequences of pan-inhibition of Hsp90 is shown down here on the bottom right in this Western blot. And what you see as around 1 nanomolar concentration, you see HER2 and AKT, which are Hsp90-dependent client proteins. And when you inhibit the chaperone, it can't fold them, the client proteins get degraded. But what was alarming is at the same concentration that you see inhibition of Hsp90 take place, leading to client protein degradation, you also see increases of Hsp90. And this is really problematic because if you're trying to inhibit a target that you're also inducing the expression of, the next day you're going to need more drug, and this is going to push the patient towards a maximum tolerated dose as one sees a dose escalating toxicity. So this is a significant problem in the clinic. Other issues that arose in the clinic were hepatotoxicity, which seemed to be scaffold related, but ocular and cardiotoxicity were observed and appears to be on target related. Okay, and so this induction of the Hsp90 that I mentioned on the prior slide is referred to as induction of the heat shock response. That's a pro-survival response. It's meant to overexpress chaperones to refold proteins that were misfolded during the cellular stressful event. Now, regarding ocular and cardiotoxicity, this was quite alarming to us. We want to know, was there a way to get around this? Can we circumvent the ocular and cardiotoxicity? And it appeared to be on target. So in 2009, the Endurian co-workers reported that the hERG channel is an Hsp90 dependent protein. And of course, the hERG channel is a potassium channel heart that regulates the rhythm of your heart, and inhibition can lead to long QT syndrome and things alike. And so in a patch clamp assay, you're not going to pick up inhibition of the hERG channel because this is a relatively short assay, but it turns out Hsp90 assembles the hERG channel. So this is something that takes about 24 hours really to measure. So you're not going to see it at patch clamp assay, which is the reason why many of these compounds got through the pre-IND studies and ended up in humans. And so Laura Peterson, who's now a professor down at the University of Florida, worked for me at the time, and she got involved in this study, and she wanted to ask, is there a particular isoform that the hERG channel is dependent upon? So she did a number of studies, which is really summarized in this little Western or co-IP in the middle right, which shows that yes, there's only one isoform that's responsible for assembly of the hERG channel, and that is Hsp90 alpha, suggesting that if you inhibit the other isoforms and you don't inhibit Hsp90 alpha, you can be devoid of cardiotoxicity. So this was the first piece of data suggesting that you want to go after isoform selective inhibitors. More recently, a study was reported where they showed that Hsp90 alpha inhibition is responsible for the degradation of retinal proteins. And so when you inhibit Hsp90 alpha, this is actually leading to the ocular toxicity as well. So the take-home message is find a way to inhibit Hsp90, but avoid Hsp90 alpha. Okay, so this was the evidence suggesting that we wanted to make an isoform selective inhibitor. So Hsp90 is a homodimer. It's kind of suggested by this cartoon here. And we're going to focus the beginning of the talk on this internal ATP binding site. And at the end, I'm going to talk to you about a compound currently in phase 2 that targets the C-terminal binding site as well. So regarding the Berger fold I mentioned a few minutes ago, it binds ATP in this bent conformation, which is rather unique. As you know, most proteins bind ATP in an extended form, which is the reason why most kinase inhibitors are relatively promiscuous. They bind to a lot of proteins, about 3,000 proteins that bind ATP in the human cell. So Hsp90, when it binds ATP, binds in its bent conformation, much like it binds these two natural products, gildanomycin and ridoscol. And if you look on the top left, these are overlaid co-crystal structures of gildanomycin shown in yellow and ridoscol shown in magenta, overlaid. And what you see is at the bottom of the pocket, there's a resorcinol ring for ridoscol, and that's overlaps with the carbamate of gildanomycin, whereas gildanomycin's quinone ring is towards the protein solution interface, interacting with aspartate 54. And so when I first started my lab in 2002, there was very little SAR known about gildanomycin and ridoscol, and they were both relatively complex natural products. And only at that point in time, Sam Danishevsky had just recently finished a total sense of ridoscol. So what we propose is that you can make a chimera of ridoscol and gildanomycin, which we term ridanomycin, shown here in the bottom left. And we propose that if we made this chimera, we could elucidate structure-activity relationships for both ridoscol and gildanomycin simultaneously. And so we made this molecule. We showed that it's a good inhibitor of Hsp90, elucidated SAR, and then we also made these open chain analogs we called ridanomycin, Radimid and Radester, shown here on the bottom right. All three were good inhibitors of Hsp90. And then we started to ask the question, can we make selective inhibitors of individual isoforms? We began a collaboration with Dan Gawer's lab, and he's a structural biologist. And what we found is that we went after GRP94 to begin with, because GRP94 was the most unique. It's only 85% identical, and it's 85% identical because it has a 5 amino acid insertion into the primary sequence. And what happens when you insert this 5 amino acids, you get this 5' extension pocket shown up here on the top right. Okay, it's a little tiny pocket that exists solely in GRP94. So when the co-crystal structure of Radimid bound to Hsp90 beta on the left was solved, there was only one conformation observed. It had the resorcinol ring at the bottom of the pocket, just like you saw with the natural product. And the quinone, or in this case, the hydroquinone, was more towards the protein solution interface. However, when the co-crystal structure of Radimid bound to GRP94 was solved, you see there were two co-crystal structures observed, one of which you had the resorcinol ring at the bottom of the pocket, but the quinone or hydroquinone was now pointing towards a protein solution interface. The second co-crystal structure showed this hydroquinone to point back into the 5' extension pocket. And we knew that's really what we want to do. We want to make a compound that fits back in a 5' extension pocket because it doesn't exist in the other isoforms. So if you backtrack down the hydroquinone ring system, what you see is that there's a cis-amide bond being formed. So when the amide goes from trans to cis, the cis-amide forces the hydroquinone into this 5' extension pocket. So this is pretty insightful and led to a study by Adam Dorefeld, who's a professor now at the University of Minnesota, who made a molecule called BNIM. It ended up being the first GRP94 selective inhibitor ever identified and the first isoform selective inhibitor. And it's shown here on the top left. We've since optimized this to make a number of analogs, for example, KUNG29. And then we sent it to the NCI for a 60 cell panel assay screen because we had difficulty finding cells that this compound was inhibiting the proliferation of, because we assumed this was going to be a good anti-cancer target at the time. And what we found is that when we got the NCI 60 cell panel assay back, we found that it was really only active against multiple myeloma cells, and other cells it didn't even touch. And so we've gone up to 100 micromolar concentration against most of these cells and never had any sort of negative activity whatsoever. So this was kind of intriguing. And then we asked ourselves, well, are there other client proteins or substrates that are dependent upon GRP94 and not dependent upon others, Hsp90 isoforms? And through proteomic studies with silac-treated cells and knockdown and knockout of GRP94, We identified both the integrins and toll-like receptors to be GRP94 dependent substrates. And as you can see in the bottom right, when you incubate cells with the GRP94 inhibitor, you induce the degradation of integrin alpha-2 without affecting AKT. AKT is it's a substrate for the cytosolic isoforms of Hsp90. So we know the integrins, they're degraded when you inhibit GRP94 and you have a non-toxic compound. So we started developing these compounds as promising antimetastatic agents. And you see the wound healing scratch assay in the top right of this triple negative breast cancer cell line. And you see the compounds are non-toxic, but very effective at 500 nanomolar concentration at inhibiting the migration in this wound healing scratch assay as a model for metastasis. So this is really exciting for us. Okay, so we're moving forward with these compounds as potential anti-metastatic agents. But then we started a collaboration with Chad Dickey's lab down at the University of South Florida, and he started looking at other disease states in which the ER, which is where GRP94 resides, has a dysfunctional triage system, much like multiple myeloma. And what he observed really quickly is that glaucoma is a disease in which you accumulate misfolded proteins in the eye. And this leads to increased ocular pressure, decreased aqueous outflow, and eventually blindness. And so there's various forms of glaucoma, and one of which is genetic, and it's called primary open-angle glaucoma. And it turns out there's a protein called myocilin in your eye that has no known physiological role. You can knock it out, and there's no observable phenotype. However, if you mutate it, it immediately aggregates in the eye, leading to the accumulation of aggregates. And what you see here on the far left is you see wild-type myocilin is readily secreted by the trabecular meshwork cells in the eye, whereas the mutated form of myocilin is not. Okay, so it's accumulating. And this is confirmed on the right with the confocal microscopy images where you see the overlap between myocilin and ER, calnex and stain, and the yellow punctate in the bottom right. And so we started asking, is there a chaperone responsible for this? And so Chad did these studies where he knocked down various chaperones, as shown on top left. And you see Hsp70, Hsp40, CDC37, Hsp90 beta. It really had no effect on the accumulation of mutant myocilin. But the moment you knock down GRP94, you saw a significant reduction in mutant myocilin accumulation in the eye. And you can see in the bottom, there's another confocal image where we looked at the siRNA for GRP94, and this led to a significant reduction in mutant myocilin in ER. So through a number of studies, we were able to show with the PAN inhibitor on the top left that these compounds in a dose-dependent manner were able to prevent the accumulation of mutant myocilin. And on the right, we use our GRP94 selective inhibitor. It exhibited a similar activity. The benefit of using the GRP94 selective inhibitors is that they're non-toxic, whereas the 17-AG, the PAN inhibitors are. And through a number of studies, the mechanism was finally revealed. And what happens is normal myocilin is not a substrate dependent upon GRP94. However, when it's mutated, it cannot adopt the normal conformation and thus becomes addicted or dependent upon GRP94. So GRP94 tries to bind mutant myocilin and tries to fold it into the proper conformation of wild-type protein, but is unable to. And as a result, the two proteins co-aggregate, leading to increased ocular pressure. So GRP94 actually takes on a toxic gain of function in these cells, okay, and leading to the accumulation of mutant myocilin. However, when you add GRP94 inhibitors to this, it disrupts the interaction between mutant myocilin and GRP94 and allows mutant myocilin to be degraded through an autophagic mechanism. So we've been really excited about this. We moved these compounds forward. We've also tested them in vivo. And more recently, we've begun to work with a venture capitalist out of California, and we hope to take these molecules into clinical trials. And you can see on the left, we have an animal model for ocular pressure. And on the left, we're administering our GRP94 inhibitors via eye drops to these animals. And you see significant reduction in intraocular pressure with our compounds. And on the right, you see this also works not only in the primary open angle form of glaucoma, but also in a steroid-induced model for glaucoma, okay, which isn't dependent upon a mutant myocilin. but yet the steroid-induced glaucoma model has a failed triage system as well. So these GRP94 inhibitors appear to be useful for many different types of glaucoma. So we're very excited and hoping to move these forward. We then started asking, can we make a Hsp90 beta selective inhibitor? Remember, we don't want to target alpha because of the cardio and ocular toxicity, but beta appears to be a great target for cancer. The problem is Hsp90 alpha and Hsp90 beta are 95% identical in the internal ATP binding site. In fact, there's only 2 amino acids that differ between these binding sites, and they're outlined in these boxes in the top left. Now, if we look at this on the top right, we see Hsp90 alpha on the left and Hsp90 beta on the right. And what you see is that in alpha, you have a serine 52, and the hydroxyl moiety is involved in hydrogen bond network with these 2 conserved water molecules that are found in every crystal structure of Hsp90 that's been solved. Whereas on the right, you have an alanine 52, and you see that this methyl group cannot engage in a hydrogen bond network with the 2 water molecules. So this suggested to us that maybe these 2 water molecules weren't held in quite as tight in Hsp90 beta as they are in Hsp90 alpha. This was our hypothesis. We then looked at the other alteration in the amino acid sequence, and that is a isoleucine to leucine transition. So you see on the left in the pink, you have Hsp90 alpha, and you have an isoleucine there, isoleucine 91. Whereas on the right, where I've circled in red, that's a leucine 91. So really what you do is you have a 1,2 transposition of the methyl group. It moves from the beta carbon to the gamma carbon, opening up this red hole I've circled. And so our hypothesis was that we can make a beta selective inhibitor if we were to modify the resorcinol ring, displace 1 water molecule in Hsp90 beta that's not being held very tightly at this location, because in Hsp90 alpha, there's a methyl group that would prevent that. So we made several molecules, and in the end, made the molecules shown on the bottom. Okay, and this compound we called KUNB31. It represents the first beta selective inhibitor of Hsp90 ever identified. It's about a 50-fold selective inhibitor, but it has 180 nanomolar affinity. So really a promising lead compound. The co-crystal structures were solved in collaboration with Bob Matt's lab, shown here on the bottom right. But the biology with these beta selective inhibitors was really cool. So if you look in the top left here, with the beta selective inhibitor, we're not inducing Hsp90 levels. Okay, remember that was one of the key detriments of the pan inhibitors, that you induced Hsp90 levels, and this led to dose escalating toxicity. So this is really cool. We overcame that. And we also know that the hERG channel is dependent upon Hsp90 alpha maturation as well as retinopathy. So do we get selective engagement of Hsp90 beta in cells? That was the real question. So through a number of proteomics studies that we've conducted, we were able to show that CDK4, CDK6, CXCR4 are all Hsp90 beta dependent client proteins. And what you see is that these clients are readily degraded between 1 and 3 micromolar concentration of the drug. Okay, showing that indeed we are inhibiting Hsp90 beta in the cellular environment. Now, if you look at the bottom left, what you see is that ERK5, RAF1, and survivin are not beta-dependent proteins. Instead, they're Hsp90 alpha-dependent. And what you see is that between 1 and 3 micromolar, those proteins aren't affected at all. In fact, they're not really affected till you get around 30 micromolar concentration. So even in the cellular environment, we have probably 15 to 30-fold selectivity in these cells. So this is really exciting, really promising. And we think we have a good new approach towards cancer now. And so a postdoc in my lab, Sanket Mishra, started looking at other scaffolds, and he designed this compound on the bottom left that we just recently published in JMed Chem. And this compound is a 19 nanomolar inhibitor of Hsp90 beta. And we modified this compound significantly. We now have a compound that's about 20 nanomolar binder to Hsp90 beta, and doesn't bind Hsp90 alpha until about 50 micromolar. So huge therapeutic window. We've increased solubility quite a bit, and we've actually spun off a company and received STTR funding. And what you can see with this, this compound now is that it too is very good at inducing the degradation of CDK4/6 as well as CXCR4, inhibitor of apoptosis protein, all of which are Hsp90 beta dependent proteins, whereas RAF1 and survivin were not affected because they're alpha dependent proteins. We see on the top right, there is no induction of Hsp90 beta, Hsp90 levels, so we got away from the pro-survival heat shock response, dose escalating toxicities. And on bottom right, you see that there was no effect on the hERG channel with the beta selective inhibitors, even at 2 micromolar concentration, whereas the siRNA for Hsp90 alpha or the pan inhibitor gildanomycin led to its degradation. So we were able to overcome these hurdles, and our original lead compound we put into an animal model for bladder cancer. And this is due to the fact that FGFR, or fibroblast growth factor receptor, is also a beta dependent client. And we showed that it was readily degraded. And we put this compound into a number of other animal studies, and we're now seeking additional resources to move these compounds into phase 1 clinical trials for the treatment of cancer, which was really exciting for us. At the same time, we started using these isoform selective inhibitors in a collaboration with John Stryker's lab down at University of Arizona. And as you all know, John's a phenomenal researcher and really into deconvoluting the mechanism for pain. And what you see is that he started looking at the pan inhibitors of Hsp90, and what he observed early on is that the pan inhibitors of Hsp90 actually blocked the effect of morphine in animal models of pain, which was really intriguing. So John started asking the question, is there a reason why? Is there one isoform that's involved in this process? And so in the brain, what he found is that when you use the Hsp90 alpha selective inhibitors, which we just published last week, as Wei Wang mentioned, I didn't talk about today, but he used these compounds in an animal model of pain with paw incision model. And what you see as you increase concentrations of the alpha inhibitor, you actually decrease the effectiveness of morphine. And this occurs in a dose-dependent process. And you see when you use the Hsp90 beta selective inhibitor, it had no effect, nor did the GRP94 selective inhibitor shown on the bottom right. This is really intriguing to us because when he did the same experiment, but he injected the compounds into his spinal cord, what he saw is that the pan inhibitor, okay, directly into the spinal cord actually attenuated the effects of morphine, okay? It made them more pronounced. So somehow the brain was making the effect of morphine less useful, whereas inhibition of spinal cord, Hsp90, made it more useful. And without really getting into the details of these studies that John will be looking to publish in the near future, I don't want to steal his thunder, he figured out something that's really exceptional and really cool. And it appears as though there's a selective way to engage the opioid receptor, immune receptor in a spinal column without engaging the mu-opioid receptor in the brain. And this could have significant effects because you can inhibit pain without inhibiting or affecting the addiction. And turns out you have to do that because if you look at when you administer a drug both to the brain and the spinal cord, the brain supersedes over the effects of the spinal cord. So you really need to make a selective inhibitor of spinal cord, Hsp90. And so hopefully in the near future, John and his laboratory will be reporting some of those studies. This is the mechanism he's come up with to suggest how this is working. In this particular case, it looks like Hsp90 alpha is contributing to the effects of the mu-opioid or the morphine on the mu-opioid receptor. And when you inhibit Hsp90 alpha, it prevents that. Whereas in the spinal cord, it's entirely different and much more convoluted, turns out. Okay, and so the last part of my talk here is going to be focused on the Hsp90c terminus. And this is the dimerization domain because Hsp90 is a dimer. And what we're going to take a peek at is this molecule novobiocin that's shown below it. And this was the first inhibitor of the Hsp90c terminus ever identified. And what we did when we first started is we made a library of novobiocin analogs trying to figure out the SAR. And what we observed really quickly was that if we just modified the side chain, okay, this is the benzamide side chain. So if we go back one slide, you see the side chain, you have an amide. And in this case, you have a benzamide side chain of novobiocin, which is reincorporated into DHN2. this molecule on the right. Whereas Ku32, the molecule shown on the left, just has an acetamide. And it turns out these molecules have completely different activities, which is really exciting because this is an assay for beta-galactosidase that's reminiscent of the heat shock induction. So if you take these cells and you have beta-galactosidase that's been engineered behind a heat shock binding element promoter, you take the cells, you incubate them at 42 degrees for 5 minutes, you see an induction of the heat shock response, shown here as HS+. Okay, this is a thermal induction of the heat shock response. Now, if you take the same cells and you incubate them with the compound Ku32 up top, you see a much more pronounced induction of the heat shock response. So it's about 3 times greater than thermal induction. And then if you combine both thermal and chemical induction, there's really no additive activity. So chemical induction of the heat shock response supersedes thermal induction of the heat shock response, shown on the left. But what's intriguing is if you take this acetamide and you reincorporate the benzamide, as shown in DHN2, you actually block induction of the heat shock response. And this is really kind of exciting. So if you look here at HS plus for Ku32, that's a thermal induction of the heat shock response, which is now blocked when DHN2 is present, as shown on the bottom right. And this is consistent with novobiocin, which was used as a control on the far right. So this benzamide side chain makes all the difference. And when you have a acetamide and induce a heat shock response, when you have benzamide, it prevents it. So we started asking the question, what do you do with a molecule that induces a very strong and robust heat shock response? And remember, the heat shock response is inducing all sorts of chaperones, Hsp27, Hsp40, Hsp70, Hsp90s. All these proteins are chaperones that are overexpressed to refold denatured proteins or protein aggregates. And so we got an IND enabling grant from the NIH at one point in time to look at this molecule as potential therapeutic for Alzheimer's disease. And this is an animal model that was conducted in collaboration with Mary Lou Michaelis at the University of Kansas. And what you see is when we administered Ku32 to these mice, and this is a JMTL3 mouse, so it's a hyperphosphorylated tau model, you can see a 65% reduction in neurofibrillary tangles when you administer the drug chronically over 15 weeks. It turns out we also saw complete reductions in A beta plaques as well. And we were really excited by the results. And we were ambitious in considering taking the molecule into clinical trials for treatment of Alzheimer's. And we began to be a little sheepish because we felt like the clinical trials for Alzheimer's were a bit more subjective than we felt comfortable with. So this is when Rick Dabrowski, a colleague in pharmacology at KU, came to me and said, you know, people always worry about the central neuropathies. What about the peripheral neuropathies? And Rick is a researcher that works on diabetic peripheral neuropathy. And it turns out that the #1 reason for amputations in the country is due to peripheral neuropathy as a result of diabetes. And this is much more severe than I ever knew. And so it turns out there's a mechanism by which you can render mice diabetic. You add the molecule streptozotocin to them, and the molecule selectively wipes out the beta cells of the pancreas, which are responsible for making insulin. And by about week 14 or 16, you have to euthanize the animals because they become so neurotic as a result of the glucotoxicity in their body. So originally, we began administering our drug at week 6. And at week 14 and 16, we really didn't see any difference between the animals that received our drug and the animals that never received streptozotocin. So we thought we had done this experiment wrong, or the control wrong, or the drug was phenomenal. And so Rick Dabrowski came to me and said, you know what, nobody goes to a doctor when they think they're going to be sick. People only go to doctors when they know there's something wrong. So we began administering the drug at week 12, okay? And what you see here, administering the drug, this is 20 mg per kg. It turns out it's equipotent at 7 mg per kg. Within 4 doses or 4 weeks, we see complete recovery of thermal sensitivity. Okay, this is really exciting. Moreover, we see the reinnervation of nerve fibers back into the epidermis. We see the remyelination of axons. Okay, we see the complete recovery of cellular respiration. We see complete recovery of mechanical sensitivity as well within 4 weeks or 4 doses. So not surprising, we licensed this compound out. It went to Therapeutics. where it is currently in phase 2 clinical trials for treatment of peripheral neuropathy. And we hope to get a secondary use of it at some point later on, hopefully for Alzheimer's. Or we also know it works very well in animal models of paralysis. Okay, we've looked at it both in animal models of genetically induced paralysis as well as trauma-induced paralysis. And what we can see is that 100% of the mice recover about 90–95% of their function, meaning that if you put a camera over these guys, you can't tell from the top that they're impaired whatsoever. You put a camera from the backside, their gait's just a touch off, but nonetheless, they're running around their cage quite readily. So I started off the talk telling you about the PAN inhibitors that went in clinical trials for the treatment of cancer. There were 17 of them, and they were really problematic, mainly as a result of the PAN inhibitory activity. And over time, we've been able to develop compounds that can selectively engage. In fact, at this point, we have inhibitors of all 4 isoforms. We've published 3 of them. And what we've shown is that if you inhibit GRP94, for example, selectively, it's a really promising anti-metastatic target, as well as a promising target for glaucoma. Whereas Hsp90 beta selective inhibitors appear exceptional for the treatment of cancer. And more recently, John Stryker's been able to show that if we can selectively engage spinal cord Hsp90, this could be a promising new approach towards a dose reduction strategy for opioid addiction. And then finally, in the very end, I told you about our C terminal inhibitors that are currently in clinical trials for the treatment of peripheral neuropathy. So hopefully I've kind of outlined what the problems were and what I propose, or my group proposes, as mechanisms to get around it. We're really exciting. I think we've stumbled across a few things that we never perceived possible. So we're really exciting. And of course, all this was done in collaboration with my group, as well as we probably have 60 or 70 collaborators that we're extremely grateful for. This is Adelma at Notre Dame, and of course, the funding agencies for supporting our research. Once again, Wei Wang, Selena, and everybody else, thank you for the opportunity to give a talk today. I look forward to more collaborations in the future and continued involvement in your center. Thank you.
Thanks, Brian, for a great talk and a very interesting research. Yeah, so that we have 2, we try to keep the time, but we have 2 questions. First one, I'm interested in the involvement of GIP with TLRS. Which TLRS? In dorsal or on the cellular surface TLRS?
Yeah, it's so it turns out
TLRS or the,
yeah, so the toll-like receptors, it turns out the toll-like receptors are dependent upon GRP90 for trafficking to the cell membrane from the ER. So when you inhibit GRP94, you inhibit their trafficking, and it actually they get degraded instead. And there's several TLRs, not all of them. I think 2, 4, 5, I believe, are the ones that come to mind. But there are probably others too. I just don't recall what all of them are.
Okay, yeah. Next question is, the ENI, based upon the Chaperone model, it has been argued isoform selective chaperone inhibitors have limited value. What are your thoughts on this model?
I completely disagree with it. I think we really kind of screwed up going after the PAN inhibitors. Okay, so 17 compounds went into clinical trials that inhibit Hsp90 the exact same way. Okay, so that's kind of a shock. And even in the study section, you still see people proposing to do it the same way again. So that's alarming. But what I can tell you is that with the isoform selective inhibitors, we've unveiled things we never even thought was possible. Like, for example, the GRP94 inhibitors for glaucoma. Who would have thought about this? I mean, that was something that was way off our radar. The studies John's done and is doing, I don't want to steal his thunder, it's phenomenal. The data he has, and we've already had pharmaceutical companies contact John about some of this data. So it's not inconsequential whatsoever. In fact, I think it's game-changing.
Okay, thank you, Brian, for your wonderful talk.
Thank you.
Thank you. So then let me introduce the third speaker, the Professor Stephen Munk from ASU the Biodesign Institute. Dr. Munk received his PhD degree from UC Berkeley. Actually, he's also hardcore synthetic chemist And they did a postdoc with, the Deerberg, the in Purdue, where, and Purdue, Dr. Munk, he now is the deputy director of the Biodesign Institute since, the October 2017. He is, experienced in the drug discovery, development, and the manufacturing, both as a scientist and also the leader. Okay, please join me. Welcome, Dr. Munk.
Thanks very much for the kind introduction, Wei. I'd like to thank Selena and the organizers for the kind introduction here, and certainly thank the other speakers for some very interesting discussions. And I should note that the photograph that I use as my sort of screenshot is me in one of Ash Stevens' operating bays. My last job was working at Ash Stevens, a chemical development company, and that operating bay was where we provided clinical supplies of 17-allylamino galvanomycin to both the National Cancer Institute and various commercial organizations. That PAN inhibitor, of course, failed in the clinic, but I thought that was a very interesting aside there. So my current role here is as the Deputy Director of ASU's Biodesign Institute. The work that we conduct here at Biodesign is inspired by nature, and not necessarily all biology, as you'll hear, but certainly takes its inspiration from nature. And our goal really is to assemble large teams to address grand challenges. All right, so today we have a total of 17 centers, and I should make a couple of comments here. I'd certainly like to thank Eric Ryman for his service in many, many roles within the state of Arizona and nationally, in part as director of the Neurodegenerative Disease Research Center. We were fortunate in recruiting Jeff Kortauer, who just started as the newly appointed full-time director. I think Jeff will have one job rather than many, which Eric had. And so Jeff joined us in March as the new director. He's an Alzheimer's and Parkinson's expert. He has some really interesting animal models and some very unique ways of delivering drugs. So Biodesign is an organization of 17 centers, ranging from things like structural biology, some bioelectronics and biosensors work, developing wearables, sustainable macromolecular materials manufacturing, looking at green chemistry to prepare polymers, and sustainable manufacturing methods. So each of these centers is focused on a grand challenge, and we assemble a multidisciplinary team of faculty members, graduate students, postdocs, research scientists to address those problems. Our faculty all have academic homes. So for example, Petra, Josh, and Sid Heck are in the School of Molecular Sciences. Rolf is in the Fulton School of Engineering. Others have different academic homes depending on their discipline. Faculty come from all disciplines within ASU and work together, collaborating to address the grand challenge. So at one end of the spectrum, Josh Robert, our executive director, who's also the director of the Center for Personalized Diagnostics, and I might add, alum of Berkeley. He took undergraduate organic chemistry from Henry Rappaport, my PhD advisor. So although we didn't know each other in those days, we overlapped at Berkeley and have Rappaport in common. So Josh is an MD, PhD, medical oncologist who has a strong interest in proteomics, and he's used his knowledge of proteomics to develop a blood test that, when coupled with mammography, is 100% accurate at diagnosing breast cancer without the need of unnecessary surgical biopsies. And those really are rather unpleasant procedures, so it's really nice to avoid those if possible. That test is now CLIA certified and has been licensed to a company for further clinical development. So that's kind of one end of the spectrum at Biodesign. As a consequence, really, of Josh's work and are having robots in place, we were able to set up a clinical testing lab very quickly. Exactly a year ago, the ASU Biotechnology Clinical Testing Lab currently can do 10,000 tests per day. To date, we've done over 750,000 tests, and our average turnaround time is under 30 hours. For those of you who have had both the nasopharyngeal swab and the saliva test, I can tell you that the saliva test is much more pleasant. The nasopharyngeal swab really is more of a probe banging away deep inside your nasal cavity to collect the sample rather than simply drooling into a tube. So we can actually do tests very rapidly with very pleasant sample collection and are able to identify the COVID variant strains because we're looking at a total of 4 proteins, and you can see certain proteins drop out with the variants. So it's been very useful, I think, to the state of Arizona to have that capacity here. And that was very nice for us to be able to pivot from basic research to clinical testing very, very quickly. At the other end of the spectrum, we have Stephanie Forrest. Stephanie Forrest is a computer scientist by training. And you might ask why a computer scientist has a home in nature-inspired research institute. Well, her view is that computer software these days with millions of lines of code really is very much like the code of life. So it should be treated as if it were like the code of life. So Stephanie actually is developing software that becomes self-healing so that when you have an error in a very lengthy program, say a 10 million line computer program, you don't have to go through line by line to find the error in line 5,486,283. The software actually is able to repair itself. It has an immune system. It can repel viruses, and it can evolve as needs change. So it's a very interesting approach to computer science inspired by nature, but not necessarily biology. As I said, biodesign takes the convergence of a variety of disciplines to address grand challenges put forth and really not only do interesting research, but translate that research into public good, as you saw with both the DISA breast, Josh's breast cancer test, and our CLIA certified COVID testing laboratory. So among the disciplines that we have in biodesign are engineering, physics, chemistry, of course, my favorite subject, informatics people, mathematicians, whole variety of people working together in large teams to address big problems. So our goal really is to eliminate the threats that challenge the world, for example, energy, pollution, mobilize large teams to address those problems, and then shepherd the solutions not only to great scientific journals, but to the public domain. So in terms of threats, one way that we're looking at threats is to look at wastewater as a diagnosis of public health. Initially, Rolf Halden was studying things like opiate addiction, and it turns out that you can measure the percentage of a population using opiates by looking at wastewater. So if Phoenix has one opiate intervention program and Pittsburgh has a different opiate intervention program, rather than targeting individuals, you can actually look to see if the overall program is being effective in terms of managing opiate addiction. You can also see how many people are smoking cigarettes, taking their diabetes medication, or as we've found in the last year, how many people in a given population are infected with COVID. So like many, Rolf is looking at wastewater as a way to diagnose COVID infection rates. Another newly formed center, so Rosie Kramnik-Brown actually has been a long-term faculty member here in Biodesign and spun out of Bruce Rittman's Center for Environmental Biotechnology to begin her own center looking at health through microbiomes. What Rosie has determined is that the microbiome of autistic children is really quite different than that of non-autistic children. And she's actually now engaged in clinical trials using microbiota transfer therapy to actually manage autism, and has had tremendous success there with these transplant experiments. So it's ongoing research, but perhaps it would be an interesting way to manage autism, a therapeutic that really isn't necessarily a drug. And it turns out that the microbiome in your gut really has a lot of effect on neurochemistry. A lot of these metabolites generated by the microbiome actually wind up having a tremendous effect on a variety of diseases, not only autism, but may have implications in Alzheimer's as well. So an area that's of substantial interest to me, given my background in drug discovery and development, is structure-based design. And Petra Frama, the Center Director for Applied Structural Discovery, really a structural biologist by background, also a pretty sharp physicist, she figured out that she could make molecular movies at free electron laser facilities. These facilities typically live at places like the Stanford Linear Accelerator, a 2.2-mile long accelerator costing billions of dollars over many years. There are 5 such free electron lasers currently in the world. Swiss Fell, DESI in Hamburg, Japan, Korea, and the Stanford Linear Accelerator. So what you're actually able to do with these free electron lasers is watch in real time as drugs bind to receptors on the surface of cells. You can watch to see how a virus attaches to and infects a cell. But unfortunately, beam time is very hard to get since there's 5 in the world and many competing groups looking for beam time. So Petra recognized that one could accelerate electrons not only with electromagnets as SLAC does, but with lasers. So she realized that you could shrink these from 2.2 miles to 30 feet and actually develop a beam line that was at the scale that a university could absorb. And we're actually building a compact free electron laser in the basement of Biodesign C. So these instruments will produce X-rays 1 billion times brighter than standard X-rays, permit images to be recorded prior to destruction of the sample. These very powerful X-rays do detonate the sample, but you're able to record a diffraction pattern prior to detonation, ultimately gathering data before damage occurs. So as of summer of 2019, we had a very nice empty vault in the basement of Biodesign C. What you see as of the winter of 2020, you actually see an accelerator seriously under construction. Here you have some of the people looking at the laser generation equipment, and ultimately in the winter we were successful in generating our first electrons. So it's actually happening. It's very exciting, and the project is moving along very nicely. You see a good shot of that as of March 1st of this year, having produced the first electrons. Now I'd like to switch gears and tell you about Sid Hecht's work. Sid is a medicinal chemist by training and very interested today in mitochondria function and dysfunction in human disease. Mitochondria are responsible for the production of about 90% of the ATP or energy that our bodies use. Also responsible for the production of reactive oxygen species. When reactive oxygen species are out of balance, out of control, increased reactive oxygen species cause tremendous damage to your body. They're implicated in a variety of genetic disorders, for example, muscle weakness, organ failure, neurological disease, Alzheimer's, Parkinson's, Friedreich's ataxia, cardiomyopathies, cardiac infarction. So really a whole host of diseases are mediated by this very interesting organelle. So Sid has actually developed compounds that can protect the brain. There's CNS, they can penetrate the CNS and mitigate damage by reactive oxygen species. So coenzyme Q is the natural endogenous agent here, and you see that it has a nice tail here, 50 carbon tail that's necessary for anchoring into the membrane. And it is able to mediate electron transport in the respiratory chain, so you can actually mitigate reactive oxygen species damage in a cell with coenzyme Q and some analogs. So Sid has developed some analogs, which you see here in the upper right. He's done a very extensive SAR study, determined that a 14 carbon tail is actually the best. You do need 14 carbon tail here. A single carbon or a few carbons does not afford a potent agent, and if you get something longer, its activity is diminished. The other thing that he's shown is you do need both an amine function and an ether here. They can be in either regiochemistry, but either the diamine or the bis ether are not as active in these studies. What they've been able to do is demonstrate that they can increase ATP production and produce ataxia lymphocytes, a good model for these disease states. They can block lipid peroxidation. These are stable in liver microsomes, so very good for metabolic stability. And they're detectable in the brain, so you get good passage of the blood-brain barrier. So it really, they function catalytically as a scavenger for superoxide radicals. In another series of compounds, Sid has developed these analogs based on methylene blue. Again, you need an alkyl tail here to anchor to the membrane. These are very potent inhibitors of ferroptosis and iron-induced necroptosis, cell death. And these two are quite potent in Friedreich ataxia models. So ATP is increased and there is substantial protection of cells here. So very nice work in medicinal chemistry here at Biodesign. Some of the teams that we've assembled here. So this is actually Josh's team looking at personalized diagnostics. So he has a team consisting of molecular biologists, clinical oncologists care, and actually sees patients once a week at Mayo, as well as doing basic immunology work here at Biodesign. We have engineers, cell biologists, informatics people, chemists, of course. You always need chemists in these kinds of programs, and software engineers. So we're actually developing databases, and this too is very useful in the COVID crisis. Mike and his team developed good databases for us to help manage the data that we're generating in the COVID testing laboratory. In terms of global engagement, Bruce Rittman here, Center Director for the Environmental Biotechnology Center, he's actually a collaborator with microbes. And what he has done with his partners, the microbes, is immobilize them in bioreactors and is able to purify water. So he's able to take mining sludge, remove heavy metals like palladium, so he can start a business, which he has, to sell recovered heavy metals and give you pure drinking water along the way. So you get a twofer here. You get pure drinking water and you have nice business opportunity. He has these sites installed all over the world, and a couple of years ago was a recipient of the Stockholm Water Prize. This is a prize established by the Swedish royal family. They felt that water was a problem as worthy as other Nobel class of problems. So they established the Stockholm Water Prize, and Bruce and his colleagues won for their work collaborating with microbes to purify water. You've certainly heard about some of the solutions that we've been able to shepherd from the laboratory to the public domain. Our first executive director, Charlie Arnson, now retired, was able to use plants to generate therapeutic proteins, antibodies, and vaccine candidates. Among the proteins that he was able to generate led to the ZMAP antibody, which was used to treat Kent Brantley here. Kent was a US-based physician who went to Liberia to treat Ebola victims and contracted the disease himself. So thanks to Charlie's work, Kent was treated with ZMAP and his life was saved. So he and his team were Time Person of the Year in 2014, and is very grateful for Charlie's work here at Biodesign, leading to a therapeutic treatment for Ebola. Another very interesting technology that was developed here is that developed by Grant McFadden. So Grant leads the Center for Immunotherapy, Vaccines, and Virology, and he's studied the myxoma virus for many years. The myxoma virus is a very interesting virus. It only affects European rabbits. So it's deadly to European rabbits, not to North American rabbits, not to hares, not to any other species of rabbit, strictly European rabbits, but also cancer cells. So Grant has actually spun out a company. He's raised $25 million from Boehringer Ingelheim to commercialize Hsp90 virus as a treatment for cancer. So we're very proud to have that spin out here in Arizona, and they're looking very good at this point and looking to do a Series B as well. So very exciting to see a spin out to treat cancer here. Like the U of A, ASU has tremendous core facilities to support our researchers here at Biodesign, also researchers at ASU, and also those in the community. So anybody in the state who has a need of genomics capabilities, very high resolution LC mass spec, imaging, cryo-EM, glass blowing can come to ASU, use the core facilities that we have at a very nominal fee and have access to very high-end instrumentation, millions of dollars worth of mass spec equipment, for example, at a very affordable price. So we're very committed to helping our researchers here at ASU, as well as supporting the local science community. So we have had tremendous impact on spinning technology out. I mentioned wearable sensors. Erica Portani and the late NJ Tao have commercialized a wearable metabolism tracker, now approved by the FDA. So breathing is actually something you can use to look at things like diabetes with measurement of breath. We spun out a company looking at potentiostats for electrochemistry, various diagnostic techniques. I mentioned the Disobreast and Oncomix. To date, the economic impact is close to $3 billion here in Arizona, over 3,000 jobs. Today, 17 research centers. We're certainly looking to continue to grow the number of research centers here. 35 startups, cumulative funding in excess of $500 million, and today over 100 diseases studied. So with that, I would like to thank you all for your attention. Again, thanks, Selena, for inviting me, and I'm certainly happy to address any questions that you might have here.
Thanks, Dr. Munk, for your great, the overview and the detail about, the Biodesign Institute at ASU. Yeah, very comprehensive. So we have one question. Discovering novel compounds to treat disease are good endeavor. What about repurposing FDA-approved drugs to treat different diseases?
Well, certainly.
After all, these drugs are on our mindset. Yeah.
Well, certainly, people are looking at existing FDA-approved drugs here at Biodesign as tools. Philosophically, I think it's a spectacular use of time, and I think you actually see that with things like remdesivir. That was originally looked at in other diseases. AZT is another great example, an antiviral that was originally developed in the 60s as an anti-cancer agent. It turns out it failed in cancer, but when HIV became a scourge in the 80s, it was the first FDA-approved compound. So I think the library of compounds that the world has, especially FDA-approved compounds, is a rich treasure trove of leads or actual drugs that can move into the human treatment regime very, very quickly. So I think that's a wonderful way to go about discovering either therapeutics immediately or good leads for therapeutics. And certainly things like the nucleoside antiviral agents have been the topic of study for many, many years, and still bear fruit, as we see with remdesivir.
Thanks, Steve. Yeah, so if no further questions.
Oh, I have a question. Sorry,
sorry,
yeah.
Go ahead. Yeah.
I realize that I don't have access to the Q&A, so I'm just gonna pop in and have a question. Thanks, Stephen, for letting us know about what's available over there. I mean, we are so close, but sometimes we are not aware of these facilities and resources that U of A can take advantage as well. And I know you spoke about big questions and big problems that Biodesign is trying to do, but are you guys open to then collaborate with smaller questions like drug discovery programs that we initiate here? can we reach out
Absolutely.
to
I
you?
think you're actually going to hear about one from Travis. So we are open to collaboration at a grand scale, at a small scale, at any scale. So I think, yes, the answer is yes. We'd like to collaborate with
Thank
all
you.
the Arizona operations if we can. TGen, NAU, U of A.
Thank you. Okay,
That's
great.
Yeah, I have one more question for Steve. Does the Biodesign Institute have HT as a high school screening, drug discovery, compound screening facility?
Sadly, no. So that's something that some of us are looking to resurrect.
But the good things that we have here.
Well, it sounds like a good way to collaborate then.
That's right. Okay, if no further questions, to join me to thank that Dr. Munk for his great the talk. Okay, so then let me give my hand to my colleague, Professor Greg Zacher, to introduce Dr. Stewart Campbell. So Greg, please.
So I get to introduce Dr. Stewart Campbell because Stewart was my first ever PhD student. And behind me, you can see his thesis from a long time ago. So obviously there are lots of embarrassing stories I can tell, and I won't take up Stewart's time by doing that. The first question he asked me when he came to work for me doing a PhD was, can I carry on playing varsity football? And I told him no, which is probably good considering his accomplishments over the intervening decades. So Stewart left my lab to go and do a postdoc with the late great Bert Fraser-Reed at Duke, where he learned some real carbohydrate chemistry, and then tried to apply the inositol work and the carbohydrate work with Insmed, which was a company that was trying to do great things based upon inositol, which is obviously the basis of multiple signaling pathways and hasn't still really been exploited. Stewart has 3 drugs over his career that have moved to the clinic. And Surface Logics in Boston, I think, was a big move for him, moving to the Boston community that allowed him to get to where he is now. I noticed that one of the drugs, belumosudil, very, very recently did not quite make it in terms of approval. But very, very recently, also note that Stewart was made CEO of Axial. And I'm super excited to learn about what he's doing because this is a really hot area. Looking at the microbiome and how one can use small molecules to intervene in the microbiome and its function in human disease. And I think Stewart's actually collaborating, or certainly knows the people at ASU that are working in this area. So I'm very excited to hear about Stewart's research that he's leading again into the clinic in a very hot and important area. Stewart.
Great, thank you, Greg, and it's a pleasure to reconnect on this basis, and I'm very pleased for the opportunity to speak with everybody today. I hope people can see my screen okay. Are you seeing the slides as intended?
Yes.
Okay, great. So what I want to tell you today, thank you for the kind introduction. I see the date on there and you've aged me quite directly, which is great. But I think from our standpoint here at Axial, I think we take a little bit of a detour from structure-based drug design to an area that's just really beginning to take root, and that's the area of leveraging the microbiome and its interaction with the host to as a means for drug discovery. And that's really the essence of what we do here at Axial. And I wish there was clear, rational drug design opportunities. There will be eventually, and we have some ideas there. But what I'm going to tell you about today is really an effort to get to some very basic proof of concept work done, and we'll use our autism program as a case study here to show how we can actually learn from the microbiome and actually come up with clever interventional approaches to help alleviate some of the conditions associated with some neurological diseases. And so what I would like to do, since I'm at a company, I have to put up this forward-looking statement, which nobody ever reads. It's like the license box you check when you download an app to your phone that nobody ever reads. But essentially what we're about here is we're really focusing on neurological conditions, so neurodevelopmental disorders and neurodegenerative diseases, and more recently, some exploits in cancer as well, as a logical extension of how we approach leveraging what the gut microbiome can do to and for the host. And so just to give a snapshot of the company, we are at clinical stage. I'll tell you the autism story here, which is about ready to start phase 2B. And we're quite excited about that. But really the company was formed by Professor Sarkis Mazmanian from Caltech, based on his 15 years or so of work in the microbiome and in particular its effects on the immune system, and more recently on how it can interact with brain function. And so we saw from Professor Munk a few moments ago, he highlighted Rosie Krajmalnik-Brown's work, where they're actually looking at the difference in the gut microbiome between kids with autism and matched control populations. And while the differences are quite pronounced, it doesn't necessarily lead to an obvious drug intervention strategy, hence their approach to actually try to replace the microbiome, which is actually a brilliant strategy that seems to be bearing fruit. We're taking a slightly different approach. I wouldn't say it's competitive or even complementary. There are some overlaps, but we are taking a very directed approach where our goal here is to identify specific targets in the microbiome or in the gut, I should say more generally, that are responsible for contributing to these types of disorders and diseases. And we then develop small molecule gut targeted therapies. So we really do not need, and in fact try to engineer out, systemic absorption. So this is in complete contrast to everything I learned as a medicinal chemist, to try to get things into peripheral circulation and reach their target tissues. Our target tissues are in the gut lumen, so we don't need systemic absorption. And we've actually translated this in a couple of clinical programs, one in autism that we'll talk in more detail about, and also in Parkinson's disease, which I won't talk about, but essentially there we've identified bacterial amyloids as both an instigator and perpetuator of neurodegeneration in preclinical models, and we have some clinical mechanistic data to support that with a pharmacological tool molecule that we took into the clinic. And then we have a strategic investment from Taiho to take our approach and apply it to oncology. And I'm not allowed to talk about those because of our relationship with Taiho. So when we talk about the gut-brain axis, it's good to sort of level set everybody so we know what it is we're talking about. And what we really see here are three major thoroughfares or highways by which the gut can communicate with the brain. And so there's kind of the hardwired route where the enteric nervous system communicates through the vagus nerve to the central nervous system. And this provides a direct tissue avenue from the gut to the brain. And this is where we leverage our Parkinson's, this is where our Parkinson's program derives from. Secondly, we have through metabolites and hormones, things produced in the gut can actually reach the brain and vice versa through the circulatory system. And the one that most people are probably aware of is the interaction between the gut and the immune system, which then can drive neuroinflammation and neurodegeneration. But until recently, people have not really considered things beyond the gut tissue and the brain, and really the elephant in the room is the gut microbiome. And so when we talk about the gut microbiome, we're talking about we ingest food, things from the outside world, chemicals, etc. And it literally feeds the 10 trillion bacteria that live in our GI. And that's, in a typical person, we have roughly 500 or so bacterial species. This doesn't include fungi or protozoa and viruses and so on. And what's really, I think, compelling is that the number of gut bacteria in our GI system equals roughly, it's actually a little bit more, but it's roughly equal to the total number of human cells we have in our body. And then the number of bacterial genes exceeds the number of human genes by a couple of orders of magnitude. So if you think of an alien coming down to Earth and trying to characterize a human being, it might look at the human as simply a very sophisticated vehicle for supporting microbial growth. And so maybe the best term to use, and one that we see more often now, is the microbiota gut-brain axis as opposed to just the gut-brain axis. And the reason we say that is because upwards of 50% of circulating metabolites at any given moment in our blood come from microbes, either directly or they're derived from microbial metabolites. They're a major source of neurotransmitter synthesis. Dopamine, serotonin, GABA, all produced by bacteria in the gut. It's not the exclusive place they come from, but they're a significant contributor to the pools. Short chain fatty acids come from non-digestible fibers, such as carbohydrates and so on, that make it to the gut microbiota. And these are known to be highly effective immune modulatory molecules. And then several diseases actually have compromised intestinal permeability. Parkinson's, autism, MS, obviously things like Crohn's disease and colitis and so on, all have some version of what we call leaky gut. And this has several consequences regarding our relationship with our gut microbiome. It allows microbial products that are generally kept out to now have better access to the peripheral tissue through translocation, which can drive all kinds of things, infection, stress levels, etc. But we think of mostly this as being a barrier to keep things in the lumen and controlling their flux into periphery. But what we also have to think about is what goes the other way. And what we're starting to realize now is that the oxygen flow into various areas of the gut in a compromised barrier situation can actually alter the microbial population. And so you'll have bacteria that are strictly anaerobic will be compromised, and they will be outcompeted by bugs that can survive in both an anaerobic and slightly more aerobic environment. And by doing so, that can cause a shift, and then that can have consequences on somebody's health. And so how do we know all this? And there's a set of technologies that we see quite often being used, and our ability to understand the microbiome is increasing almost on a daily basis. And so if we go back, we rewind a few years when all we had was a stool sample from which we could extract microbial DNA, and we could do look at things like the 16S ribosomal RNA sequences and get a relatively crude view of who is there. So this would be part of the descriptive aspects of the microbiome. And so understanding who's there is only a very small part of the problem. And so 16S gives you maybe, you could get to the species level in certain cases, but generally the resolution of this technique sort of stops at the genus level. And so what we see most frequently now with sequencing techniques becoming easier to access and less expensive is a metagenomic sort of deep shotgun sequencing, which tells us not only who's there, but what their capacity is, what they're capable of. It doesn't tell us what they're doing, but we have a much more granular view of what is the functional capacity of the microbiome. But again, this is only part of the problem. And so what we've seen more recently is progression into understanding the functional role of the microbiome. And for that, we need to do transcriptomics, so see what genes are turned on, this actually proves to be really, really technically challenging. And I won't get graphic here, but if you've ever tried to extract messenger RNA from a stool sample from a Parkinson's patient who is supremely constipated, and so the amount of water in there is actually really, really low, there are some significant technical challenges here to get good quality mRNA out of a stool sample. So people tend to rely more on proteomics and more recently on metabolomics. And this is where we have applied, at least in autism, the bulk of our effort is doing, excuse me, understanding what metabolites are being produced by microbes in the gut. And we can look both in the stool and in the periphery, blood and urine, to find out what the microbiome is actually doing. And so with this variety of techniques to understand the microbiome, we also now need to understand how to interpret or evaluate these profiles in the context of disease. And so we use this to identify specific targets and then go through pharmacological validation of these targets in the context of disease. And so we take human samples, and we'll often take germ-free mouse models of disease where there's no microbiome, and we'll bring in human microbiota from diseased and healthy patients, and we'll see if we can transfer any of the symptomology and pathologies of a particular disease or disorder by just simply introducing human microbiota into a mouse model. So it's humanization, but in a different context. And so what we've been able to do here is use these omics technologies and these pharmacological tools to both identify and validate new targets. We'll talk about the autism as a case study in a moment. Once we have a target identified, we then apply our medicinal chemistry expertise to drug that target. And typically, we deal with small molecules that do not need to get systemically absorbed, so there's no need to cross the blood-brain barrier. And remember, our target here is neurological indications. And we think that by keeping things in the gut, we have a higher probability of having an improved safety profile over something that may be systemically absorbed. It's not always true, but it's a reasonable premise to start from. And then, of course, it's not simply having the molecules and making them big and multiply charged so that they don't, they're not orally bioavailable. We really have to think about the activity against the target and then deliver the molecules to the lower part of the GI where the microbes generally live and where they're most prevalent. So most drug delivery technologies rely on different release profiles and so on that are designed to release the drug either at a certain site in the small intestine or over time as it transits through the upper part of the GI. And so there's relatively little, not zero, but relatively little out there in terms of lower GI or colonic delivery. There are a few platforms available, and we've evaluated most of them and have our own as well. So this is really how we approach things. And now I'll jump into the autism story here. And so just to give you a bit of background on autism, the prevalence, it's actually a really prevalent disorder. 1 in 54 kids in the US is the latest estimate, and that's up considerably from even 5 years ago and 10 years ago. So there's over 1 million kids in the US with autism and several million worldwide. It's probably underdiagnosed worldwide. The prevalence is, or the incidence rather, is increasing at an astounding rate. And this is not accountable simply by better diagnosis. There's really something else going on. And both the sad part and the opportunity is that there's very little available from a therapeutics perspective for treatment of autism. There's nothing approved in the US to treat core behaviors. The core behaviors being repetitive stereotyped motion, lack of social communication, and social withdrawal. There are only 2 drugs approved. They're both atypical antipsychotics, and they are approved to treat irritability associated with autism. This is actually a big deal, and it'll become very relevant to our story and our plan as we go through the case study here. So keep irritability in mind with known drugs that are approved but have very significant side effects that really limit clinicians' willingness to prescribe these drugs. So just to remind you again about where we're headed with autism, is we're really focused here on the interaction between the gut and the brain through peripheral circulation. And again, Professor Munk referred to metabolites coming from gut microbiota and them having a profound impact on certain disease states. And that is in fact the case here in autism, and that's where our intervention is targeted. So our motivation and rationale for taking this approach, we mentioned no approved drugs. We know that GI disturbances are prevalent in children with autism. Depending on the studies that you look, it's anywhere from 40 to 70% of kids with autism also have GI issues. And these GI issues range from constipation to diarrhea and sometimes oscillations between the two in the same individual. So it's a very debilitating comorbidity and has a pretty big impact on their quality of life quite often. And what has been alluded to and implicated in several studies preceding what we've done is that the gut microbiome may well be an active contributor through the gut-brain axis to autism. And so it does provide potential new avenues for intervention. So in short here, and I'll get to some details on this, is our mechanistic hypothesis is that there is a class of neuroactive microbial metabolites. We've coined the term the NMMs, and these contribute to autism behavior phenotypes by altering brain myelin plasticity. This is not published yet. It's under review at a top journal at the moment. I think it's close to acceptance, but not quite yet. And essentially, these metabolites can reach the brain and alter the brain's myelination capacity. So our treatment hypothesis here is simply to reduce these neuroactive microbial metabolites in the gut, to reduce their systemic and brain levels, which should improve behavior. And in preclinical studies, this therapeutic that we have was shown to reduce systemic levels of these metabolites and improve autism-like behaviors in mice, and I will show you that. But to back up a little bit, how did we get here? So this goes back to a publication in 2013 from Sarkis Mazmanian's lab at Caltech, when they were looking at an immune activation model for autism. And there are about 36 autism mouse models out there. And the one that they were focused on was this known link between infection in the pregnant mom and the elevated risk of autism. And so they developed this model whereby if you induce an infection in what equates to the second trimester of pregnancy, you can actually induce autism-like behaviors in offspring. And so to try to understand what was going on, the original hypothesis was that this was being driven by the immune system, and the infection would create an unfavorable immune response, which could then compromise normal development. And so that there was obviously an immune component to the development of autism in this model. But what they found was that when they did an untargeted metabolomics profile of the blood of these mice that were autistic, or had autism-like behaviors, I should say, and compared them to wild-type mice, they found significantly elevated levels of some metabolites. And one, 4-EPS or 4-ethylphenyl sulfate, and we'll get to the structure of that in a moment, was significantly, about 40-fold elevated in the mice in which had the autism-like features and were induced by injection of this polynucleotide in the pregnant moms. And so this, at the beginning, was thought maybe this could be a biomarker for risk of autism or degree, severity of autism or something like that. But they went a step further and actually injected synthetic 4-EPS into wild type mice. And what they showed, and we show an example here, is that if you take wild type C57 black mice, inject 4-EPS in saline IP, you can induce a very significant anxiety phenotype. And this is the open field test, where you put the mouse in essentially an open bin with borders on it, and you measure all kinds of aspects of how that mouse behaves, and including one of the telltale key measurements is how long it spends in the middle of the open field, which is reflecting a sort of an open, vulnerable situation to predators. And so the more anxious the mice are, the more they will stay out of the, excuse me, the middle of the bin, and they'll stick close to the walls. And there are several other aspects of anxiety that were shown simply by injecting 4-EPS into wild type mice. And what's interesting is it had no ambulatory function on the mice. The mice, it wasn't like the mice were sedated or didn't move very much. They moved the same amount, but they just didn't go into the center area. And so this was our first clue that things like 4-EPS that come exclusively from microbes can actually contribute to a behavior. And what's interesting is they did IP injection, but the terminal half-life of 4-EPS in mice is less than an hour. So they did daily injections for 3 weeks, and the 4-EPS sticks around in the mouse circulatory system for less than an hour, and they still saw this profound effect. And so this was great. We can actually get some sense of what might be going on in these behaviors in mice, but what the heck does that have to do with a human being? And so what we did when we first started Axial 4 years ago is we went and did a cohort study, non-interventional. We looked at, and we collected blood and urine from the subjects that were in the UC Davis charge study. It was a big genetic epidemiology study for autism. And we accessed these samples they had, and we did untargeted metabolomics. And out of over 1,000 metabolites we looked at, 4-ethylphenyl sulfate was one of the most dysregulated metabolites between the autism group and the control group, such that, and bear in mind, there's no recognized normal level or normal range for 4-EPS because nobody's really studied it before. So we somewhat arbitrarily chose the 95% CI of the mean for controls as the upper limit of normal. That's just a mathematical choice we made. And we see that about 1/3 of the kids in the cohort with autism had significantly elevated levels of 4-EPS. And you see this is a log plot here. So some of them have sort of 100-fold higher levels of 4-EPS. And these levels up here correlated to autism severity and social behavior as well. So now we have evidence in mice that a metabolite coming from microbes is pharmacologically active in an autism-relevant manner. And we see the same metabolite dysregulated in a significant cohort of autistic kids. And so this told us that there may be some translational value in what we were seeing in the mice. And so we've since broadened our scope of our hypothesis here. And what we did, and this is the part of the work that's just under peer review right now, is we went back to the mouse situation, and instead of doing injections or oral gavage, we said, let's try to come up with a system in which these metabolites are introduced to the mouse in a more representative fashion. So we took germ-free wild-type mice, and we introduced into them 2 microbes. And the 2 microbes combined, they colonize, and they combine to produce, constitutively produce the precursor to 4-ethylphenyl sulfate, 4-ethylphenol. And so this comes from the digestion of tyrosine. So you need protein in the diet. The microbes digest the tyrosine and produce 4-ethylphenol, which is rapidly absorbed, goes to the liver, where the liver tries in its traditional way to detoxify, and it ends up sulfating 4-ethylphenol to produce 4-ethylphenyl sulfate. And it turns out that this is the bad actor in this system. And then we've since also taken the data that's been produced by others where they've shown correlations of paracresol levels to severity of autism in kids. Indoxil gets transformed to indoxyl sulfate, which is a known neurotoxic molecule. And so we've got now this collection of metabolites produced by microbes in the gut. When the liver tries to detoxify them, they end up circulating as these sulfated forms, which can lead to aberrant behaviors. And so when we looked at the microbes, or the mice that have the microbes that produce significant amounts of 4-ethylphenol, we had a corresponding control group in which we had the exact same microbes, but we engineered out the last steps in the 4-ethylphenol synthesis, so that we ended up with the two groups of mice where the only difference between the two was the presence or absence of 4-ethylphenol coming from microbes. And what we see is that the groups of mice that are constitutively exposed to 4-ethylphenol through the GI, and so 4-ethylphenyl sulfate in circulation, they actually, we were able to now see impacts on ultrasonic communication, which is typically how mice communicate, such that the mice who were exposed to 4-ethylphenol communicated significantly less than those that were not exposed to 4-ethylphenol. And we replicated the anxiety data here as well. So now we're seeing that these metabolites, when they're introduced chronically through the microbiome, can actually induce a plethora of behavioral changes. And I want to be very careful here to make sure that we don't leave you with the impression that 4-ethylphenyl sulfate causes autism. That's not at all what we're saying. What we're saying is this collection of microbial metabolites, and there are others in this panel, so we've expanded this panel to about 8 or 10 molecules of this structural class, that these have effects on a variety of brain functions. And what we've since done is tried to shed some light on mechanistically what's happening. So it's one thing to say, okay, you've got this materials being produced in the gut, they get into the blood and the animals behave differently. Well, how is this happening? And what we've shown is that these mice that are exposed to 4-ethylphenol and hence 4-ethylphenyl sulfate, the oligodendrocytes in their brain their development is arrested at the oligodendrocyte precursor stage. And you need oligodendrocytes fully functional in order to have proper myelin plasticity, because they're the cells that actually make and lay down myelin onto neurons. So we've seen in imaging studies differences in myelination patterns in critical areas of the brain, like the amygdala and the prefrontal cortex, where we're seeing changes in behavior correspond to changes in myelination. And this is through oligodendrocyte maturation. So we're able to overcome oligodendrocyte maturation deficits with other pharmacological agents to show that this is in fact the mechanism by which these metabolites are operating. So this creates a very interesting interventional approach. Number one, we could go in and restrict the diet so that these aren't produced, or we could try to modify the microbial population so that we alter these levels. All feasible if you have the right amount of time and money to do it. It's still a tall order. We decided to intervene right at this level. So rather than worry about who's producing these molecules, we want to sequester these molecules in the gut with a non-absorbed molecule that would then usher their excretion in the stool and thereby reduce the levels. So it's a pretty simple but elegant strategy that divorces us from needing to manipulate the microbial populations in a very targeted way. And so it allowed us to actually conceive of an intervention that could prove this concept pretty quickly. And so what we did was we identified a molecule, AB2004, and to one of the earlier questions from one of the attendees about why not use repurposed molecules, this is exactly what we're doing here. This isn't the end game, but this is a way for us to very rapidly prove the concept preclinically and get into the clinic, and that's what we've done. So we've taken AB2004, which is designed to bind these and sequester these molecules, very high affinity, very good binding kinetics, so that as it transits through the GI system, it picks up these microbial metabolites and pulls them out so they don't get into circulation to the same level. This is an approved drug outside the US. It's never been looked at in a CNS indication and never used in a pediatric population. So we're doing this for the first time here. The safety profile is excellent. And I'll come back to referencing those approved drugs in autism with black box warnings, serious side effects. We don't have the side effects with this drug. And so it's very convenient for dosing kids with autism because it's simply a powder that is sprinkled into food and they can consume the food that they would normally eat. So we're not breaking their normal daily routine. So we took 2004 and we put it back in our mouse model. And I won't go through each of these bar graphs, but what you can see is that the introduction 2004 was able to significantly reduce these metabolite levels, as 4-EPS is shown here. And we're able to show that the control mice that are exposed to 4-ethylphenol, when they're actually treated with AB2004, we compare the second bar and the fourth bar, we see that we're restoring normal function with AB2004, both in terms of anxiety and in terms of repetitive behavior, in this case, by marble burying and grooming. And so we have now preclinical proof of concept. And with that, we went into a clinical study we conducted in Australia and New Zealand, completed just before the pandemic broke out. So we were able to get the full cohort through the protocol. And it was really a safety and tolerability study where we also were looking at target engagement, which for us is, can we move the microbial metabolite levels in blood and urine? And then we did a battery of behavioral assessments. And so it was a single cohort, multiple dose escalation study. So 8 weeks of exposure to AB2004. And we obviously starting with kids who are 12 to 17 years old. And we wanted to make sure that the drug was safe and well tolerated as we escalated. And then we took the drug away and we followed them again for another 4 weeks. And I want to mention just one point about behavior testing. This was an open label study. So anytime somebody hears open label study where you're measuring behavior, the alarm bells should go off about a placebo effect. And we need to really be cognizant of that. And so what we did was we measured behavior in a variety of ways for this reason, and also because the translation of behavioral changes in mouse models is not always clear how that translates into a human. So we wanted to make sure we covered a variety of behavioral measurements to see where our signal was. And happy to say that the drug was in fact safe and well tolerated, so no surprises. We had no drug-related adverse events. And this is in sharp contrast to the atypical antipsychotics that are on the market. And so when we looked at anxiety measurement as measured by the pediatric anxiety rating scale, and remember, we did not include or exclude based on any behavioral phenotype. So we had a good mix of kids in the study. And when we look at the whole cohort, we did not see a significant change in anxiety levels. But when we did a post hoc analysis and pulled out the kids who had high anxiety, and that, according to this pediatric anxiety instrument, is a score of 10 or above, we see a significant reduction in anxiety. And when we took the drug away, the anxiety changes persisted for at least 4 weeks. And what we see here is significant reductions in this panel of microbial metabolites. And when we look at the metabolite changes in the high anxiety cohort, we see significant reductions, but a different pattern. And the reason I highlight this is this now presents a really nice opportunity to start doing machine learning to understand what the combinations of metabolites are that are contributing most to the changes in behavior that we're seeing. And when we went to look at other, we saw several other clinical signals. One of the most important was in irritability. And I remind you again that the other two approved drugs are designed or approved to treat irritability associated with autism. And what we see in this case is even at the full cohort level, we're seeing significant reductions in our key metabolites, and we see a significant reduction in irritability using the aberrant behavior checklist. And this is one of the most established ways of measuring aberrant behavior, including irritability in kids with autism, and was exactly the same instrument used that led to, by the other companies, that led to the approval of those other drugs. And so when we do the same kind of post hoc analysis, and we look at the population that has high irritability at baseline, and that's defined as a score of 15 or higher, we see a very significant reduction in irritability, and it's on par with the approved drugs. And so if we think about the placebo effect here, with this instrument, this irritability assessment, the placebo effect is very well understood. It's been pushed through 5 phase 3 level clinical trials and has been assessed and reviewed by FDA, and they have for these other drugs. And so it's very well understood that this type of signal we're seeing is most likely real. Obviously, we have to replicate it. And so I mentioned that we did a 4-week follow-up. And so when we look at what happens, I've added a third column here to the bar graph, and that is what happens in the recovery period is the irritability score starts to creep back up. It's still lower than at baseline, but it's starting to creep back up from the end of treatment. And what we see over the course of this 4 weeks is metabolite levels have rebounded and gone back to baseline. So there's this nice toggling of changing metabolite levels and changing behavior. So there seems to be a good correlation there and gives us confidence that our mechanism is operative here. And so where we are is we've had a successful pre-IND meeting with FDA. As I mentioned, the first study was done open label in Australia and New Zealand. So we are expecting to hear back around our IND momentarily and proceeding to a phase 2 randomized controlled study that looks like this. It's a pretty substantial one of about 200 kids, which we'll do in Australia, New Zealand, and the US. And so that will start in the first half of this year. So we're quite excited about where this is headed. And with that, I would like to thank everybody, the different institutions that helped us get this clinical trial and all the preclinical work, the Axial team, and thank you for your attention.
So thanks, Stewart. I mean, that's really thinking outside the box. There's one question up here about antibiotics and any role for antibiotics modulating the microbiome in autism and behavioral disorders.
Yeah, it's a really good question. And in fact, what really started the thinking around this at Caltech back in around the 2010 timeframe was a very small study out of the UK where they took 11 kids with autism, gave them long-term treatment with vancomycin to wipe out their microbiome, and most of their behavioral symptoms improved. Then they took the antibiotics away, and the microbiome obviously rebounded, and their behavior rebounded as well. And this is actually being used by the folks at Arizona State, where they do a wholesale microbial transfer therapy, but they first clean out the microbiome with as much with antibiotics as much as possible, so that the new microbiota that are being introduced have a chance to colonize.
And I have a quick question. I mean, aryl sulfates, you don't expect these to really get in the brain. You don't expect them to be necessarily biologically active. And the other thing is the liver is very important in terms of sulfation and desulfation. So these aryl sulfates are microbial products. There's no host metabolism that's associated with the neurotoxic chemicals.
So maybe I wasn't clear, but the thing that's produced by the microbes is the actual phenolic precursor, and then they're actually sulfated by enterocytes in the gut and also by the liver. And one of the things I didn't mention was that in mice, they actually do get into the brain. So we've done sort of brain PK, and you can actually see them get in there. And so, there is good evidence. Now, whether that occurs in people, we don't know. So.
I have a question as well. Thanks for the really nice presentation. I thought that you mentioned that you would like to develop a drug that binds metabolites and take it out in the stool, but you're using a drug that's FDA approved. So AB2004 has been modified to not be absorbed. I mean, you expect that an FDA approved drug, it does get in the body. So, can you talk about that? I don't know if you can talk about that. But.
Yeah. So this is, it's not absorbed.
Okay.
And there are FDA approved drugs that are not absorbed. You think of the bile acid sequestrants and
okay, all right.
So it's the same kind of concept there, except this is not a simple ion exchange resin. So it's made by a molecular imprinting approach. So you have these small molecules you use to actually create the binding pockets, and then it's a material, and then you remove those sacrificially during the production process. So we end up with a relief structure in the material that is ideally suited to bind these metabolites.
Okay. And in terms of the constipation in Parkinson's patients and also I guess diarrhea in autism, right? Is that also a...
Yes, big deal. Yeah.
It's a big deal. So you have this. So is that what starts first? Is that the abnormal bacteria that colonizes them and it causes these disturbances in the intestinal tract, or is that the disturbances in the intestinal tract that actually cause the bacteria to, the bad bacteria to colonize?
Yeah, I think the short answer is we simply don't know.
Okay.
We do know in Parkinson's is that in a majority of patients, if you do a medical history on them, they will have had GI problems, including constipation, sometimes for decades before they get their Parkinson's diagnosis. And this led to this whole idea that Parkinson's may actually start in the periphery and somehow migrate to the brain. And it was this whole prion hypothesis with alpha-synuclein and working its way through the vagus nerve and so on. There's actually a lot of support for that, but it's still a controversial hypothesis. And I think what we'll find is that there's really a couple of different classes of Parkinson's patients where you have almost like a convergent set of symptoms based on different etiologies. And one group, the disease would start in the periphery.
Thank you.
Thanks, Stuart.
Okay, thanks, Greg. So the last speaker of this morning section is Professor Travis Dunckley from ASU-Banner Neurodegenerative Disease Research Center. The Professor Dunckley has received his bachelor degree and PhD degree all from the U of A in molecular cell biology. And his research and expertise is lying in the neuron genomics to genomics technology development for the biomedical and translational research. And recently, he now collaborated with my colleague, Professor Chris Hume, on the development of the DUCK1 kinase inhibitors for the treatment of the AD. So Travis, please.
Thank you. Is that screen shared?
No. Okay, coming.
Okay, great. Yeah, thank you, Dr. Wang, for that introduction, and thank you for the opportunity to present what has been a long-standing collaboration between myself and Dr. Chris Hume from down there at University of Arizona College of Pharmacy, who's been a phenomenal medicinal chemist on this project and really invaluable, as you'll see as I go through this talk. So I think we're all aware that Alzheimer's disease is already a huge public health problem. It's currently the 6th leading cause of death in the United States. And because it's an age-related disease, we have an aging population, and there is no effective disease modifying therapeutic for Alzheimer's. These numbers are projected just to increase pretty much exponentially, so that there are going to literally be tens of millions of people affected by Alzheimer's disease by the year 2050, unless we can derive some therapeutic intervention to either delay or stop progression of this disease. So there are 3 key pathologies of Alzheimer's disease. So probably one of the earliest pathologies is a loss of synaptic connectivity between neurons, and eventually this progresses to a point where neurons are not communicating properly, and there are learning and memory deficits associated with the disease. The 2 other pathologies, which I think most people associate with Alzheimer's disease, are these extracellular amyloid plaques composed primarily of beta amyloid peptides, and then the intracellular neurofibrillary tangles, which are composed mainly of a hyperphosphorylated and modified form of tau protein. So when I was considering, how to tackle Alzheimer's disease, I chose to focus on these intracellular tau pathologies, really for 2 reasons. One is just pragmatically, everybody was focused on amyloid. And then sort of secondarily, if you look at which of these pathologies actually correlates more closely with cognitive decline in Alzheimer's disease, it's actually the intracellular tau pathology. So just a little bit about tau. So tau is a microtubule binding protein. It has 4 microtubule binding repeats that attach it to microtubules, and its function is essentially to stabilize the microtubules, which is critical along axons, because microtubules are the highway system, if you will, for neurons, whereby the neurons transport things from the cell body out to the functional part of the cell, which is the synapse. So tau undergoes this cycle of phosphorylation and dephosphorylation, which serves to regulate its microtubule binding affinity. So tau that is hyperphosphorylated has a lower microtubule binding affinity than unphosphorylated tau. So in Alzheimer's disease, this pathway is essentially skewed to the right, leading to hyperphosphorylated tau, aggregation of this hyperphosphorylated tau into paired helical filaments, and then subsequent full maturation of tau pathology into neurofibrillary tangles. So in looking at this pathway, I chose to focus here on this step of hyperphosphorylation, which is one of the earliest points at which you could reasonably therapeutically intervene. So what I did was develop this cell-based assay to measure tau phosphorylation. And this is an antibody-based assays wherein you have an antibody that's specific for phosphorylated tau that's conjugated to a fluorescent secondary. And then you have a different antibody that detects all tau protein conjugated to another fluorescent secondary antibody. So then you get signals of red and green fluorescence from your cells that correlate to phosphorylated tau and total tau expression levels in your cell. So this assay is amenable to high throughput screening. So we did that using an RNAi based approach, screening for various small interfering RNAs that will affect tau phosphorylation. So here, basically in each of these boxes, there's an siRNA for a given gene. Each box has a different gene targeted, and you can screen through thousands of genes and identified those that affect tau phosphorylation the most. From that screen arose one particular kinase of interest that the rest of the talk is going to be about. This is called DYRK1A. This is a serine threonine kinase. You can see here that siRNA targeting DYRK1A lead to significant reductions in this phosphorylated form of tau protein. So DYRK1A, as it turns out, does more than phosphorylate tau. As it turns out, it phosphorylates a number of proteins that are key for Alzheimer's disease. So sort of here on the left in this reddish color are pathways that converge on tau aggregation and neurofibrillary degeneration. On the right in the blue are pathways involving amyloid beta that serve to increase A beta expression and amyloid pathology. Both of these pathways work together to contribute to Alzheimer's disease. So one of the things that made this target particularly attractive is the opportunity to affect both amyloid and tau pathways by hitting a single target. So here we expanded the previous screening results, silencing DYRK1A and showing effects on tau phosphorylation at multiple different sites. So these are sites here in the case of serine 262 that are affected sort of early in the tau hyperphosphorylation pathway and Alzheimer's progression. Threonine 231 would be what's considered an intermediate site or intermediate stage of pathology, and then serine 396 is associated with more mature neurofibrillary pathology. DYRK1A, as it turns out, affects tau phosphorylation at all of those stages. So the next thing that we did was treat cells with this small molecule called harmine. At the time, this was probably 10 years ago now, actually, harmine was the most potent and well-known DYRK1A inhibitor, the most selective DYRK1A inhibitor. So we put that on cells. We showed a very nice dose response for reduction of tau phosphorylation. However, harmine has a number of off-target liabilities, so it's never going to be a drug. So it was really at that point that Chris Hume jumped on board. And what was known at the time were that there were these three crystal structures of the active site of DYRK1A with three different ligands in the active site. On the left is harmine. In the middle is this another compound called Indy, which basically has the same binding mechanism. And then on the right is this third compound called D15. So Chris took a look at these, made some hybrid molecules to engage some relevant interactions here, and then move ahead from there. So the series started at DYR001, which had nominal activity. Chris then progressed this to these two molecules, DYR219 and DYR266. And you'll see that they have some nice activity for reducing tau phosphorylation. Both of them are better than harmine. They don't have the structural liabilities of harmine as well. DYR219 in particular, in some surrogate blood-brain barrier permeability assays, showed some really nice permeability, so predicted to get into the brain really well. So what we did was put that compound into a mouse model of AD. So in considering selection of which mouse model, we wanted to keep in mind that we have a target that we think is going to be involved in both amyloid and tau pathology. So we selected a mouse model that presented both of those pathologies. And this is what they call the triple transgenic Alzheimer's mouse. So this is a genetically engineered mouse that expresses various human mutations that cause early onset Alzheimer's disease. So they express a particular amyloid precursor protein mutation. They express a presenilin-1 mutation, which is involved in APP processing. They also express a tau mutation. So the combined effects of those mutations are an age-dependent onset of Alzheimer's pathology. So in these mice, amyloid pathology develops at 6 months of age. Tau pathology develops usually around 9 months of age and then progresses in severity as those mice get older. So we took these mice at 10 months of age. They have both amyloid and tau pathology present. We dosed them daily via intraperitoneal injection once per day for 8 weeks at 12.5 milligrams per kilogram. We then did a number of behavioral assessments on the mice, open field testing, contextual fear conditioning, radial arm water maze, followed up with a neuropathology assessment. So here in the radial arm water maze, you can see the last two columns here are the transgenic mice. The white bar are the vehicle-treated mice. The checkerboard here are the drug-treated mice. So you can see very significant reduction in the number of reference memory errors that those mice are making, and a reduction in the number of working memory errors. So it looks like here, the compound improves the memory of these mice. We then looked at amyloid plaque pathology. Saw a very nice reduction in amyloid plaque, and that's quantitated here in the bar graph, about a 50% reduction in amyloid plaques in the brains of those treated mice. And that reduction was associated with reductions in insoluble A beta peptide levels as well, and that's quantitated here on the right with vehicle, again, in the clear bars, drug treated as a checkerboard. So we also showed DYRK1A inhibition reduces the phosphorylation of the amyloid precursor protein. So this was previously published that DYRK1A phosphorylates APP on threonine 668, and that phosphorylation contributes to increased proteolytic cleavage of APP and production of A beta peptides. So the DYR219 treatment reduced that phosphorylation, and that's quantitated there, a pretty significant, about 40% reduction in the levels of phosphorylated APP. We also showed significant, about 40% reductions in phosphorylated tau levels. However, perhaps not surprisingly in retrospect, what we did not see were significant reductions in that mature neurofibrillary pathology in those mice. So just to summarize here what I showed you, DYR219, which was our proof of concept molecule now going forward, improved working and reference memory, reduced amyloid plaques, reduced full-length APP, soluble A beta peptides, and reduced insoluble hyperphosphorylated tau, but didn't change overt pathology. And we published that a few years ago in Aging Cell. So returning to this schematic of tau phosphorylation and its role. So the APP or the tau pathology is really here on the far right. And we're doing an intervention that's upstream here. So the idea here is that we didn't see reductions in this pathology because we treated at a time point in which it was already forming. So intervening up here likely would not have an effect. So the next experiment that we did was to treat the mice earlier before they develop any pathology. So this follow-up experiment, we dosed mice at 6 months old. And this is, they're starting to get a little bit of A beta pathology, but they have no tau pathology, and they're not going to develop it for another 3 months in an untreated case. So we dosed mice at 6 months old in 2 groups. One group received drug for 3 months. The other group received drug for a full 6 months. We assessed pathology at the end of both of those time points. So what we saw was a really nice delay in the onset of tau pathology. So on the top are the vehicle-treated controls, either at 3 months here on the left or 6 months on the right. You can see some tau pathology forming here and progressing rather significantly by 6 months. In the drug-treated mice, none of the mice had any tau pathology after 3 months, and this was an N of 12 mice per group. At 6 months, a few of the mice showed a sort of a spurious tangle here or there. About half the mice still had no tangle pathology, however, as well. So what we see here is a really nice delay in the onset of tau pathology with this approach. And that's just sort of backed up here by the Westerns, looking at total tau and phosphorylated tau, and then sort of quantitating here all of the mice from each of the cohorts, where you see very significant reductions here with the drug, which is shown here in the pink at both of the time points. In terms of the amyloid plaques, we saw essentially the same thing. Very significant delay in onset of plaque pathology. None of the mice had any amyloid plaques following 3 months of treatment. They were beginning to form some plaques here at 6 months, but still significantly reduced. And that's quantitated here. Essentially what we found was that the drug-treated cohort after 6 months basically looked like the untreated group at the start of the study. So this is essentially a 6-month delay in the onset of amyloid pathology in those mice. This is quantitation using ELISA of the insoluble A-beta and A-beta 42 peptides, and they essentially show consistency with the plaque observations, reductions here, drug-treated versus vehicle, significant reductions at both time points in insoluble A-beta 42 peptides. Now, one of the surprising things that we found in the course of this study with 219 is that in the cell-based assay where we look at reductions of tau phosphorylation, what we also saw were a significant dose response effect on the expression of DYRK1A itself. And this shown on the right, this reduction is due to proteasomal degradation of DYRK1A because we don't see it when we treat with drug plus a proteasome inhibitor. DYRK1A is no longer degraded. So as it turns out, DYRK1A is it's a member of a smaller family of kinases that are that undergo an autophosphorylation event when they're produced. So DYRK1A autophosphorylates on a tyrosine, a couple of tyrosines actually. And if the protein fails to autophosphorylate, it doesn't fold properly and it gets degraded. So type 1 inhibitors, which DYR219 is, at sufficiently high concentrations will interfere with that autophosphorylation and promote degradation of the kinase. So it looks like this drug does that. So when we saw this result, we went back and looked at the brain tissue from this study. What you see at both time points here with DYR219 treatment is a significant reduction in the levels of DYRK1A protein. And we think that this reduction is probably contributing to some of the efficacy that we see with the compound as well. So these results were published in 2019 for anyone interested in sort of following up on some more detail about that study. So 219 has been a great proof of concept molecule. It let us sort of validate the therapeutic approach using mouse models. However, it has a number of issues where it too is probably not going to be an effective therapeutic down the road for Alzheimer's disease. And those are sort of highlighted here on the left. So this circular plot here is a kinome scan wherein the compound is tested against a panel of 468 distinct kinases. Each red dot represents a hit of activity of the compound against that particular kinase. And from that, you generate a selectivity score. For DYR219, the score is 0.19. So it's inhibiting about 19% of the kinases down to about 35% of untreated or of normal activity. So we felt like we could improve upon that selectivity. In addition, the compound has no oral bioavailability and poor solubility, both two issues that Chris felt were readily addressable based on the structure. And its half-life is very fast in the brain, cleared essentially within 15 minutes. And again, returning to the prior slide, we think we're able to get long-term effects with such a rapidly cleared molecule because it's promoting degradation of the compound when it gets in the brain, or degradation of the target protein when it gets into the brain. However, so Chris undertook a lead optimization effort, and then some 300 compounds later arrived at this lead compound called DYR533 that addresses all of the the prior weaknesses that I just highlighted. Its selectivity score now is 0.03, so much improved. It's much more stable in all of the in vitro surrogate assays, the microstomal stability, hepatocyte stability, very stable compound. We did plasma protein binding on this in 5 species. Very reasonable plasma protein binding. It's 100% orally bioavailable in mouse, 80% in rats. It has no genotoxicity based on the micronucleus test. And we did a number of CYP inhibition studies. It's perfectly clean there. And we did a hERG ion channel and has no activity against hERG as well. So we're pretty excited about this compound and pushing it forward. So we will be doing that as said, some subsequent follow-up tests with UR533 showing sort of in vivo efficacy, hopefully with that molecule as well. And then with a long-term view toward pushing this through some IND enabling studies. So with that, I'd like to acknowledge the folks that contributed to the study. Chris and his team down at U of A just did yeoman's work on the medicinal chemistry here to arrive at these compounds. Folks here at ASU on my side and others. Ramon is sort of a great in vivo Alzheimer's a guy here who has a number of models and can work through just in terms of future collaborations. If anyone's interested in Alzheimer's, a number of animal models, please look him up as well. And the works was funded sort of historically through the Alzheimer's Drug Discovery Foundation, Harrington Discovery Institute, and currently through the NIH through an R01. So I'm happy to take any questions.
Okay, thanks, Travis. So I have 2 questions for you. The first one is, what causes the PPP phosphorylation? Have you looked into the activating the phosphatases?
Okay. If I could try to understand the first question, I'm not clear on what PPP phosphorylation is. If they're asking what causes the tau phosphorylation, I think the ultimate upstream signals of that are yet to be deciphered. There's some potential synergy with amyloid itself, promoting tau phosphorylation. We looked into phosphatases, certainly. We screened every phosphatase encoded by the human genome in that initial RNA-based screen. So there were a number of phosphatases that came up from that. However, as you can imagine, the most likely therapeutic approach there is to activate the phosphatase. And just in terms of sort of simplicity moving forward, we felt like it would be easier for us to inhibit a kinase and activate a phosphatase. So that's the sort of the rationale for why we've chosen to focus on the kinases thus far. But certainly there were phosphatases that affected tau from that screen. There are also published phosphatases as well. Likely others are pursuing that route.
Okay, the second question is, how did you deliver the compound? How it is taken up by the cells? Maybe also my further follow-up question is how to get into the brain, BBB cross.
Okay, so the compound was just injected IP into the belly of the animal. It is sort of, it's a very small molecule. It readily crosses the blood-brain barrier. It's not really subject to efflux, so it's not actively pumped out once it gets in there. But the answer is it's passive diffusion across the blood-brain barrier and into the cells.
Hi, Travis, I have a question. Nice talk. Dirk one, do you know if the potency of your compounds are similar towards phosphorylation of mutant tau compared to wild type tau? Because Alzheimer's patients don't actually have mutations in tau, but yet tau phosphorylates and causes neurofibrillary tangles. So I was just wondering whether the potency would be, the compounds would be more active in the wild type tau than in a mutant tau.
You know, I think that's a good question. I think the data are pretty solid that DYRK1A phosphorylates wild type tau. So to the extent that DYRK1A is going to be involved ultimately in Alzheimer's disease, yes, I think we're going to have equal potency. Just sort of in vitro, we've looked at this. The potency is not different, but I cannot say for sure how that's going to translate ultimately when we talk to or when we look at translating this into humans. I can't say definitively that it's going to be equal potent, but I imagine even if there's a drop in potency, we're still going to be able to get this to a significant.
I would say it's probably more active towards wild type tau, not less active. And one model that you could do this is Down syndrome models, where because as I'm sure you know, the triplication in chromosome 21, DYRK1 is there as well. So you get more activity of DYRK1, and you have also 3 copies of APP. And I believe that model also has tau phosphorylation. So that would be a kind of a nice model, animal model for.
Yeah, absolutely. We have proposals to do that. But there are publications showing really nicely that DYRK1 inhibition works well in those Down syndrome mouse models.
Oh, okay. Great.
Okay. Actually, another question from my colleague, Professor Wangdrag. He asking, which cell targets are you envisioning? Are you talking about hippocampal neurons or the microglia?
Yeah, that's an interesting question. So based on my talk, I'm going to conclude neurons and tau within neurons. However, I skipped over a whole aspect of DYRK1A and its role in neuroinflammation. So in addition to phosphorylating APP and tau, it phosphorylates some transcription factors that promote neuroinflammation. And there are some other folks that have some published data showing that DYRK1A inhibitors reduce neuroinflammation in some of these Alzheimer's mouse models. So very likely we're talking about both pathways being mediated by DYRK1A, but the mechanisms are clearly different. It's DYRK1A phosphorylation of different targets and different cell types having relevant roles.
Okay, thanks, Travis. So that we just finished this morning section. So please join me to thank all the speakers for their wonderful talks. So we will resume, this afternoon section at 12:25. So about 25 minutes. So see you guys, soon. Bye-bye.
Welcome back to our Spring Summit, highlighting academic innovations in the drug discovery ecosystem. Each speaker will present for 30 minutes, followed by 5 minutes questions. Participants are invited to submit questions during this 5 minutes following the presentation, or throughout the presentation as well, and using the Q&A at the bottom of your screen, and then the questions will be presented at the end of the talk. We will attempt to get to all your questions. If there is unanswered questions or no time for them, we will respond and share with attendees after the summit. If there are no questions, we will proceed to the next speaker. We have a great lineup of speakers this afternoon, and it's my great pleasure to start this afternoon session introducing Dr. Barbara Slusher, Professor of Neurology and Oncology at Johns Hopkins University School of Medicine, and also Director of the Drug Discovery at Johns Hopkins. She's actually Professor of Neurology, Pharmacology, Molecular Sciences, Psychiatry, Neuroscience, Medicine, and Oncology. Dr. Slusher received her undergrad degree from Dixon College, where she graduated valedictorian, majoring in chemistry. She received her PhD in pharmacology and molecular sciences from Johns Hopkins School of Medicine, while simultaneously earning her master's degree in administrative science from Johns Hopkins Business School. She has extensive experience in drug discovery through early clinical development, and she has been involved in the successful development, launch, and post-marketing support of several FDA-approved drugs, has co-founded 4 companies amassing over $115 million funding. Barbara will speak today on the discovery and development of tissue-target glutamine antagonist prodrugs. Barbara, it's yours.
Oh, thank you, Selena, for that nice introduction. Let me go ahead and share my screen. Can you see my screen okay?
Yes.
Okay, all right, good. Well, thank you. Thank you to the organizers for inviting me today, specifically Selena. And I'm going to tell you about an academic story of a discovery in academia at the bench that we've been able to take all the way to the bedside. And this is specifically the discovery of a targeted glutamine antagonist prodrug. Now, before I get into that, Selena asked me to say a couple of words about Johns Hopkins Drug Discovery, which is the academic drug discovery unit that I run. And that's because it's a little unique. Hopkins actually hired an entire drug discovery team from pharma and moved it into academia to help translate the discoveries there. So I'll talk a little bit about our unit. I'll also talk just a couple slides on the rising role that academia is playing in drug discovery, because I think it's pertinent to the summit today. But then I'll spend most of the time talking about this targeted delivery of glutamine antagonist project. Okay, so a little bit about Johns Hopkins Drug Discovery. In 2010, so about 10 years ago, an entire drug discovery team from pharma moved to Johns Hopkins. So how did that happen? There was a company called MGI Pharma that was in Baltimore, and we were acquired for almost $4 billion by ASI Pharmaceuticals, and they moved everybody up to Boston. At the same time, Hopkins was interested in bringing in a team of translational scientists into Hopkins to help translate the discoveries that were occurring there. And so we moved an entire team, about 20 of us, left pharma and joined JHU. And the idea was that faculty were good at basic science and target discovery. The faculty were also good at clinical trials, but it was this middle group that our drug discovery unit was bringing in, how to do druggability, high throughput screening, lead op, candidate selection. And so really when we got there, our new job was to work collaboratively with the faculty to translate discoveries into clinical therapeutics. And so we focus on small molecule capabilities, and we have an integrated team of experts all under one roof. So we have about 8 or 10 medicinal chemists that do small molecule chemical synthesis and SAR. We have a group that does assay development and high throughput screening. We have a team that focuses on drug metabolism and pharmacokinetics. And then we have a team that does in vivo pharmacology and toxicology. And we all work together, integrated under in the same place. And in the 10 years that we've been at Johns Hopkins, we've prosecuted 11 different drug discovery projects. And you can see they're quite diverse in virals, cancer, CNS disease, pulmonary disease. And of these 11 drug discovery projects, that's led to multiple collaborations with pharma. In fact, 2 of these projects have been licensed to pharma for development. We've also had multiple collaboration with venture capital groups, and that has led to a spin out of now 5 new companies. We had another one last week, 5 new companies that we've spun out that have amassed significant capital to be able to move these projects into development. And so today I'll be mentioning one of those projects, which is focused on glutamine antagonist with Drayson Pharmaceuticals. But before I do, I just want a couple of slides on the rising role that academia is playing in drug discovery. There's been really a dramatic increase in academia involved in drug discovery. I founded an organization called ADDC, or the Academic Drug Discovery Consortium. And based on the membership of that consortium, we looked at the number of academic drug discovery centers that existed over time. And so in 1990, you can see there's about a half a dozen centers. About 12 years later, that grown, but last year there were over 100 translational centers that were associated with universities here in the US. So a pretty dramatic increase in a small period of time. Not only are you seeing more academic drug discovery units, but you're also seeing more academia and industrial partnerships. From the time period of 2006 to 2015, there's actually a 500% increase in the number of partnerships, public-private partnerships. And then lastly, just last year, BioCentury Innovation published an article showing that academic innovation is also fueling the majority of the early stage startups. These are companies that are looking at Series A financing. So in 2018, 75% of the early stage new companies came out of academia versus a smaller percentage in industry. And so, I just wanted to make that pitch because I think this symposium is timely, and the focus of academia playing a role in the translation of new ideas is very pertinent and timely. So the last part, and mostly what I want to tell you about, is an example of an academic drug discovery program, and this is the targeted delivery of glutamine antagonists. Okay, and this focuses on the glutamine antagonist called DON, or 6-diazo-5-oxonoreleucine. And this compound is a broadly competitive antagonist of glutamine. So shown here, here's the structure of glutamine, and here's the structure of DON. And you can see they're very similar, with the exception of glutamine has this amino group here, where DON has this reactive diazo group. So DON is actually a natural product that was isolated back in the 1950s from bacteria in Peruvian soil. And it was shown to broadly inhibit, specifically, but broadly inhibit multiple glutamine utilizing enzymes. And so why did we care about this? Well, glutamine is known to be quite critical for tumor growth and tumor metabolism. And as shown here, the cells use glutamine in multiple pathways. So glutamine is used for the synthesis of nucleotides like purines and pyrimidines. It's used in coenzyme synthesis like for NAD. It's very involved in the TCA cycle in glutaminolysis, involved in amino acid synthesis, hexosamine synthesis. So it's very critical for metabolism. And people have shown that if you treat tumor cells with DON, it dramatically inhibits tumor growth in multiple different preclinical models. And so this is a target, a therapeutic target for treating tumor growth. All right, in addition to the tumor, though, glutamine is really critical in the brain. So glutamine is a critical source of neurotransmitter glutamine through the glutamate-glutamine cycle between astrocytes and neurons, where the astrocytes are providing glutamine, which is then converted into glutamate. And so again, glutamine is critical for the source of neurotransmitter glutamate, and multiple labs has shown if you inhibit glutaminase with DON, that you can block the generation of glutamate in the brain. And that is efficacious in preclinical models wherein too much glutamate or hyperglutamate transmission is presumed pathogenic. And so this data showing that inhibiting glutamine or antagonizing glutamine may have some therapeutic potential has been around for a while. And so, in fact, DON, back in the 1960s and 1970s, people took DON into the clinical trials because of this profound anti-tumor activity that was seen preclinically. And in fact, DON showed anti-tumor activity in many different cancer clinical studies. I'm just showing some of them here. These are phase 1, phase 2 studies in both adults and children showing activity of DON in a variety of cancers. The problem is that DON wasn't well tolerated, and so its development was halted. And when I say not well tolerated, most of the toxicities were GI toxicities because your gut is highly glutamine utilizing. So patients were getting nausea and vomiting and gastritis. And because of that, its development was halted. And so about 5 years ago, we were very interested in tumor metabolism. And so we wanted to try to get DON back into the clinic. And our strategy was perhaps we could create a prodrug of DON, right, that could increase its delivery to the target tissue, in this case tumor or in brain, but decrease the delivery of DON to the toxicity tissue, specifically gut. So get it more in tumor, less in gut, so that the therapeutic index would be larger and we could successfully develop this, this strategy. And so what we did is we came up with 2 basic projects, prodrug strategies, to try to enhance the delivery of DON to target tissues in order to widen the therapeutic index. And these strategies were different. So first we strategized to brain target DON. Okay, and here our strategy, here's DON, here's a picture of DON, and you can see the carboxylate and the free amine are 2 basically handles that we can put on pro-moieties. And so for brain targeting, our idea was to introduce some bulky hydrophobic groups here in order to enhance the C-log-P, make it more lipophilic so it would get across the blood-brain barrier. In addition, we wanted to maintain plasma stability so it didn't all break down before it got to the brain. And so that was our strategy for brain targeting. The second was if we could tumor target DON. And here the strategy was different. Here we wanted to put on pro-moieties at R1 and R2, which would be preferentially cleaved off by enzymes that are known to be enriched and highly upregulated in tumors, like cathepsins and HDACs and plasmin. And so let me show you what we've done in the past 5 years. I'll start with brain targeting. So first, we made a lot of compounds. We've made about 250 DON prodrugs that are brain targeted. I'm going to spare you the details. I won't go into all of this. This was our screening paradigm. So when we made compounds, the first thing we said, are they stable in plasma? Because if they fall apart in plasma, they're not going to have an opportunity to get to the brain. We then looked for brain stability because we wanted them to have the ability to be biotransformed to DON in the brain. We then looked at their PAMPA permeability. Were they highly permeable and thus brain penetrable? And then if all this was positive, we went into in vivo pharmacokinetic studies and then efficacy studies. And so this is how we screened the couple of hundred compounds that we made. And again, I'm going to spare you the details and just talk about the winners, the compounds that had the best effect. And so for brain targeting, the compound that showed the best effect through the screening paradigm was ASU-395. And again, shown here is the structure of DON. It has a C-log-P of -2.5. And by putting these hydrophobic groups on DON, the isopropyl ether and this palm ester, we were able to increase the C-log-P to almost 3, which is more ideal for a brain targeted compound. And what we showed with 395 is that it was stable. So we saw stability across species in plasma, pigs and dogs and monkeys and humans. We also found that it was highly permeable. And so DON itself had no permeability in PAMPA assays, where the JHU-395 was highly permeable, in fact, similar to the positive control verapamil. And then when we put 395 in brain, we saw instability. We saw it break down to be able to liberate DON. So it had really the ideal in vitro profile that we were looking for. And so this led us to take the compound in vivo. And so we studied 395 in both monkeys and in pigs, and we compared it to giving DON. And so what we saw is if we gave 395 or DON to monkeys or pigs, in comparison to the equimolar DON, the prodrug could increase the delivery of DON to the brain, or we saw an increase in the brain to plasma ratio in both monkeys and pigs. And so here on the left, I'm showing the CSF to plasma ratio in a monkey. So DON gave a low CSF to plasma ratio, and that was increased about 15-fold. When we gave the prodrug 395, and similarly in the pigs, this was we sacrificed the pigs, so we actually got brain here and looked at brain to plasma ratio, and again a 9-fold increase in the amount that we got in the brain, the target, and less in the systemic circulation. So again, more targeting this to the brain, less where we didn't want it to go. And this led to a couple of JMed Chem papers that go through the SAR of that studies. But after that, with these brain targeted prodrugs, we and others now, because we've given these out to many people, have shown that they're safe and they show efficacy in multiple models of CNS disease. And I'm just showing some of them here. These are diseases where abnormalities in glutamate are known to occur, things like Alzheimer's, encephalitis, neuroAIDS, depression, multiple sclerosis, cerebral malaria, stroke, brain cancer. So these compounds have not only been efficacious, but most importantly, they've been very well tolerated. So this has been where we have gone with brain targeting, but I'd say most of the lab's effort has been focused on tumor targeting. And again, the strategy is different. Here we want to use pro-moieties, which are going to be preferentially cleaved off by enzymes that are enriched in the tumor. And so let me show that. So this is a little schematic. This blue is DON, active DON, but if we stick pro-moieties on it, then it doesn't work. It's inert, it's inactive. And so we designed it such that these pro-moieties would be on, and the prodrug itself would circulate inactive and inert. But when it gets into tumor cells, where these enriched enzymes exist, it would remove the pro-moiety and then liberate DON to be active. And so that's the overall strategy, is to have these promoieties be cleaved off by enzymes that are enriched in the tumor and less abundant in other tissues, specifically gut. And so again, what we were looking for, our ideal prodrug, was a prodrug that would be stable in plasma, stable in liver, specifically stable in the gut. We don't want to generate DON in the gut, but would preferentially cleave to DON in the tumor. And so again, we made a lot. I think probably we're at 500 or so DON tumor targeted prodrugs that we've synthesized. This is a lot of iterative work and trial and error. And again, I'm not going to bore you with all of these compounds, but again, this is the screening paradigm we used. We tested all of these compounds for plasma and liver and gut stability. If it passed criteria, then we would look to see in a tumor cell if it could liberate DON. If it did, we then went into mouse and dog pharmacokinetics and ultimately tumor efficacy studies where we looked for efficacy without toxicity. And again, I'm going to show you the kind of successful and most successful examples that we had. So the first was we had acetylated lysine promoieties. So we strategized using these promoieties on DON. And the reason is that if acetylated lysine takes two enzymes, both of which are enriched in the tumor, to activate to DON. So here's drug, in this case DON, with acetylated lysine. Okay, in the presence of HDAC, the acetyl group will come off. And in the presence of cathepsin L, the lysine will come off, liberating DON. And so what's exciting about this strategy is it takes advantage of two different enzymes, both of which are enriched in the tumor, to have a sequential cleavage. And so I'm just showing the structures down here. Here's DON, and you can see the acetyl group comes off with the HDAC, and then cathepsin L cleaves off the lysine moiety. And this idea came from a Nature paper that had been published by Michael Heyman's group, showing this idea of selectively targeting tumors with tumor-enriched proteases. And so these are the structures that we made. Again, this is DON with isopropyl ester, and this is acetylated lysine. In addition, we tried multiple R groups on the amine of the lysine to try to enhance or optimize penetration into the tumor. We made about a dozen. The winner, the one that looked best, was a compound called GHU-607. And I'll show you a little bit of the characteristics of this. So again, 607 had these ideal in vitro characteristics. It was stable in plasma, it was stable in liver, it was stable in intestines, but it was not stable in tumor where it liberated DON. And so if we took human plasma with human tumor cells in it and incubated it with 607 and looked for either 607 or DON, what we found was that the prodrug was stable in plasma. So we didn't see any liberation of, or very, very few liberation of DON. Mostly it was intact, inert prodrug. However, if you looked within the tumor cells, what you see is hardly no intact prodrug, all liberation of DON. So again, we're seeing the prodrug stable in the tissues we don't want it to be in, but in the tumor cells, we're selectively liberating the active DON. Exactly what we're trying to get to. And then we took this, based on this data, we took it in vivo. And so this was super exciting. So we took mice, we gave them 607, and then we measured the DON, the liberated DON. And what we found that is when we gave 607 to mice, we saw 5-fold higher levels in the tumor, represented here in the red, versus plasma in the black. So again, we were getting more to where we wanted it to in the tumor and less in systemic circulation. But probably most impressively here on the right is we saw 12-fold higher levels of DON in the tumor, again in red, than in the GI tissue. So very low. So this is exactly more in the tumor where we want the DON to go, and less in the tox organ in the tumor. And so this was quite exciting to be able to tumor direct this DON to the tumor and out of the tox area. And so based on this, we decided to try to get this in the clinic. And so to do that, we, first we published a paper, but then to do this, we wanted to get in this clinic. So we founded a new company, and we called that company Drayson Pharmaceuticals. And we raised enough money to get it into development. It's just a a couple of press releases from that. So when Drayson took over this, this project, they picked a different compound to go into development. This was a compound called JHU-400. And as small companies often do, they immediately changed the name. So now it's DRP-104, it's the same compound. And this compound was selected to go into the clinic. And so again, here's the characteristics of this clinical candidate, DRP-104. Again, completely stable in plasma, mouse, dog, pig, human, completely stable in GI, mouse, dog, pig, human, liver, stable in liver of mouse, dog, pig, human. However, when we put it into human plasma with human tumor cells, what we see is it's stable in the plasma. We're not releasing DON in the plasma, but in tumor, it's releasing DON preferentially. And then we took this compound, again, in vivo, this time both in mice and in dogs, and looked for the preferential delivery to tumor. And so first, again, in the same mouse model, when we gave DRP-104, we saw that it delivered 6-fold higher levels in the tumor, again in red, shown in red here, versus plasma in black. And then again, most importantly, it showed 11-fold higher levels in the tumor versus the tox tissue, which is GI. And based on this data, we then decided, because we were going to the clinic, we wanted a higher species to test this concept in. And so we developed a collaboration with the Colorado State Vet School. They have companion dogs that we were permitted to dose one time, just one. This wasn't a chronic study. It wasn't an efficacy study, just a simple PK study. So we tested these dogs by giving them DRP-104 and then sampling both plasma and their tumors. These were mostly Labrador retrievers that had surface lymphomas that could be biopsied. And so what we found was the dog looked very similar to the mouse. So we had minimal DON in dog plasma. Most of what we saw was the intact prodrug, much less DON. But then when we sampled the tumor, what we saw was a 5-fold higher level of DON in the tumor than we saw in plasma. Again, very similar to what we saw in mice. So from a PK perspective, we were able to get, again, more where we wanted it, less in the tox species. And then when we looked at the efficacy of DRP-104, it had exactly the same efficacy as if you gave DON, but it had dramatically less toxicity. And so let me walk through these data with you. This is looking at EL4 tumor growth in mice. And what you can see is after you implant the tumor cells, if you're a vehicle animal, within 2 weeks, your tumor's huge, about 2,500 millimeters cubed. However, if you've got, if the mouse were treated either with DRP-104 or DON, you can see the tumors completely melt away. And we have seen this over and over again in multiple tumor models, but a huge decrease in the tumor for both of the drugs given at equal molar doses. The difference being, if you look at the weight, the DON animals, they don't have tumors, but they're at the same time that their tumors are gone, they've also lost a considerable amount of body weight, 20% to 30% of their body weight, and either have died or had to be sacrificed given this tremendous weight loss. In contrast, the animals that got the DRP-104 had really no significant weight loss at all. And so when you look at the overall survival, the animals that got the DRP-104 survived. We didn't see any deaths. And the vehicle animals all died because they had huge tumors. And the DON animals, they didn't have any tumors, but they were dying because of the toxicity. And so if you look here, we're showing just pictures of the GI systems in the vehicle animals. You can see in the vehicle animals, we see very nice intact mucosal levels, the mucosal layer is intact. We see very nice organized crypts, looks normal. But in the DON treated animals, you can see like a complete loss of crypts, severe ulceration of the mucosal level, a lot of neutrophils and lymphocytes next to this huge bacterial mat. And then in contrast, if you look at the animals treated with DRP-104, while they don't look normal, we do see some GI changes, but significantly better than what we saw with equimolar DON. You have mostly a maintained mucosal layer, some disorganization of the crypts, some epithelial hyperplasia, and some lymphocytic infiltrations, but in general, much better than the DON group. So again, showing that we can have similar efficacy, but significantly less toxicity. And so This led, so this is basically where the development led. So we licensed the compound to Drayson to take to the clinic. But one of the questions that we kept asking, and this is work done in collaboration with Jonathan Powell at Hopkins, who's an immunologist and co-founder of Drayson with me. One of the questions we kept asking, why do these things work so well? Every tumor model we put it in, either us or our collaborators that looked at our prodrugs, we saw really significant just abolishment of the tumor. And so we asked, why do they work so well? And so what we found out was that these glutamine antagonists can deplete tumors at the same time they can enhance T cell function within the tumor microenvironment. And this is quite interesting. So what we found was that in cancer cells, they take up glutamine and glucose to support their growth. But by doing this, they deplete the microenvironment of nutrients. They cause hypoxia, they cause acidosis, and all of that leads to an impairment of the T cells, the effector T cells in the microenvironment. But what happens when you put a glutamine antagonist on board is it limits the growth of the cancer cells, resulting in more nutrients that are abundant, less hypoxia, less acidosis, which is really creates a microenvironment that's favorable to the T cells. So the T cells are invigorated and can then act to kill the cancer cells. So it's this killing the tumor while activating the immune system within the tumor microenvironment that's giving us this profound activity. And so one of the things you might be asking is, so if these glutamine antagonists kill the tumor so well, why aren't they killing the T cells? And so that's a very important question. And so what we learned is that when you give these tumors glutamine blockade, it induces a completely different metabolic effect in cancer cells than it does in T cells. And so if you here on the left, if looking at tumor cells in the presence of a glutamine blockade, you see a complete inhibition of glycolysis and oxidative metabolism. Everything is shutting off. At the same time, if you look at T cells, activated T cells that have the same glutamine blockade, you actually see a marked upregulation of oxidative phosphorylation. And I could give another lecture of all the intricacies here, but basically these T cells in the presence of glutamine blockade actually upregulate their ability to metabolize acetate by ACSS1, and thus acetate can build acetyl-CoA, which can feed the TCA cycle, also activates pyruvate carboxylase, which can enhance the TCA cycle intermediates. And so, and the tumor cells can't do this. So it's really interesting. We've identified a previously unidentified difference in the metabolic vulnerability or plasticity between tumor cells and T cells. And this led to a science paper that was published about 1.5 years ago that talk about this divergent metabolic programs. In addition, the New England Journal of Medicine then did a commentary on how exciting this was as a potential therapeutic in cancer moving forward. And so I'll just end here with stealing from the Drayson pharmaceutical website and basically showing that this compound DRP-104 is now in phase 1/2 development at multiple sites in patients with cancer, and we're currently picking a second generation glutamine antagonist for the brain to move forward. So this is a real story of making a basic science discovery in academia and getting it all the way to the bedside. And so let me end with probably what's most important is the people. So this is the Johns Hopkins Drug Discovery Group. There's about 35 of us. Specifically point out Rana Race, who runs our DMPK group, and Takashi Sakamoto, who runs MedChem. They were very involved in this project, and also thankful for all the funding sources that that made this project possible. And at that point, I'll stop and say thank you and ask for any questions.
Thank you, Barbara. That's fascinating. And we have 3 questions. And I'm going to read them to you. What makes the DAWN tumor trophic? Furthermore, cancer cells are more alkaline than normal cells. How can you activate the cathepsins unless they are in the endosomal compartment?
So first, I think the tumor cells undergo Warburg physiology. So they, even though their presence of nutrients, they they take glucose and make lactic acid. So it's well known that you see hypoxia and acidity in the tumor microenvironment. The second question was, how are these enzymes active? If you could just say that again.
What it makes Dawn tumor trophic, I'm sorry.
So I don't think Dawn is trafficking to the tumor. I think what happens and what we've seen is when you give these prodrugs, they're distributing throughout the body. But we're preferentially being cleaved in the tumor to Dawn. And that's what's making it different. So it's not that they only get to tumor. The pro-moiety is going everywhere. The prodrugs is going everywhere. But the selectivity is that it's selectively being cleaved in the tumor by these enzymes that are enriched there. And I want to point out, this is in black and white, right? These are shades of gray. But what we're seeing is like tenfold more cleavage in tumor than we see in the gut, which is what gives us this improvement in the therapeutic index.
Great. So the next question is, DRP104 is recognized by the cells as non-self. How can it evade the prying eyes of the immune system unless you trick them? But I guess you already talked about the immune system. So
I asked that question again. How is
DRP104 recognized by cell? If it's recognized by cells as non-self, I am not sure I understand this. This statement. How can it evade the preying eyes of the immune system unless you tricked them?
Yeah, so this isn't tissue, this isn't protein, this is a small molecule therapeutic, and typically small molecule therapeutics are not recognized as non-self by the body. So this is simply it's very small molecule that is given and then converted in the tumor.
Okay, and the final question is, does glutamine blockading T cells induce T cells to release pro-inflammatory cytokines? How it's being regulated to avoid cytokine storms?
Yeah, so what we've, this is a great question and one that we're actually studying now. This is mainly a project that Jonathan Powell's lab is engaged with. What we see with the glutamine antagonism in the tumor cells is we do see some increase in cytokine, but it's not that robust. More what we're seeing is a long-lived phenotype of these T cells with a more robust memory. So we're seeing more active and long-lived memory T cells. It's probably the largest phenotype that we're seeing under glutamine blockade. So after we give the glutamine antagonist, if we take it away and the tumor starts to, we don't see resistance. Basically, we can come back and give the tumor again without it seeding. And I think that's based on the enhancement of the memory T cells that were observed.
That's great. Thank you. Thank you so much for participating and for the great talk. I will now, it's my great honor to introduce the next speaker, Dr. Victor Ruby, Regents Professor Emeritus in the Department of Chemistry and Biochemistry at the University of Arizona. Dr. Ruby received his bachelor's and master's degrees from the University of North Dakota and his PhD from Cornell University. He's also professor in neuroscience, professor of medical pharmacology, professor of Bio5, and has led an admirable career at the University of Arizona since 1968. His research interests include the chemistry of human behavior, particularly peptide hormones and neurotransmitters, drug design, discovery and development in multiple diseases, pain, addiction, feeding and sexual behaviors, pigmentation disease, cancer, diabetes, immune response, GPCRs, in general, biophysics, biochemistry of health and disease. Dr. Ruby is going to speak today on recent approaches to drug design and development for peptide hormones and neurotransmitters and future prospects. Dr. Ruby, floor is yours.
Thank you very much for the very kind introduction. My pleasure to be here today. My talk is going to be a little bit different than all of the others. I'm going to give a perspective talk. I've been in peptide science for 55 years since my postdoc with Vincent Duvigneau. I didn't know anything about peptides when I entered this group, but I have developed a great interest in them, and it's my passion now for a very long time. So what I want to do today is to talk about new perspectives in drug design. I think that there's going to be a revolution. We've had a molecular biology revolution. And the way that we've been doing drug design for the last 150 years has got to change if we're going to get some decent drugs. And the object of my talk today is to tell you about this and to try to find a way to let me get back to where I'm supposed to be. Okay, so let me get started. So first of all, drug design development has been a hallmark of Western science. I think everybody knows that. It's had some positive effects and also some negative effects. The positives are very obvious to most of you: treatment of diseases, amelioration of pain, prevention of diseases, improved health, both physical and, of course, our prolonged half-life. But there have been some negatives also. Virtually all of our drugs have toxicities. Even our most common drugs like aspirin and morphine and so forth have severe toxicities. And many of our drugs only treat symptoms, such as the drugs I just mentioned. Also, we tend to deliver most of our drugs by the gut, which is the worst way to develop, to send a drug into our physiological system. Because the gut has a biome, and now we've found out in the last couple of decades that this biome is very much affecting our drugs. In fact, over 50% of our drugs that have been studied so far are modified by our biome. So this is not the way to develop drugs, and who knows, a lot of our toxicities may be due to this. So it's the worst way to develop it, to send a drug. And people understand this now because there are now 20, 30 companies that I know about, and there may be others, in which alternate methods of drug delivery are being developed. And then we now have what is probably the greatest scientific and medical scandal of our time, the opioid epidemic. There is no reason for this. Discoveries have been made 20, 30 years ago already that should have prevented this, but have not been developed by the drug business for whatever reasons. Maybe because they're making so much money by allowing the drug epidemic to occur. So we need some new approaches to drug design. First, we have to recognize what the problems are. And I've already mentioned some of these. First of all, of course, we don't fully understand most disease states, so we still have a lot to learn about diseases. But most of our drugs are given orally, which has been a very big problem, as I just mentioned. And we have to pay more attention to nature. When animal life started about 1 billion years ago, everything changed in terms of the way in which biology does in multicellular animal life. Small molecules, for the most part, were no longer made in our body, except for amino acids, nucleic acids, sugars, and lipids. And vitamins, cofactors, and other things that we need for our metabolism are now gotten primarily from our diet, are not obtained are not synthesized in our body. There is a reason for this. I'm not going to have time to get into this today, but in any case, so animal life now makes primarily nucleic acids, amino acids, peptides, proteins, lipids, and sugars. And that's all the other making going on elsewhere in nature. So the conclusions are that alternative methods for drug delivery are needed, in fact, required. Most of our drugs will be amino acids, nucleic acids, sugars, lipids, peptides, and proteins, or combinations thereof, or analogs thereof. And we need a much better understanding of good health and disease states.
Dr. Ruby, could I ask you to please turn off your email so we don't get interrupted by the sound of emails coming in? If you can easily do that, it would be great.
Knowledge. I'm sorry, I don't see my email on my screen, so.
Oh, okay, no problem. Okay, that's fine.
Sorry, but I'm sorry about that. But you know, I can't get rid of something I don't have. Sorry, it is on my computer, but not on my.
Oh, don't worry. Don't worry.
Okay. So, but why peptides and proteins? Okay, they're made up of only 21 naturally occurring amino acids. But obviously we and nature have designed hundreds of other novel amino acids, most of which are non-toxic, but several of which are also very toxic. Animals make peptides and proteins and use them for almost all of their critical biological functions. We have developed highly optimized methods for the synthesis of peptides and proteins. In fact, the most effective and efficient syntheses of most organic of all organic compounds now are peptides and proteins, with the developments that have occurred in the last 40, 50 years. We have a very good understanding of the metabolism of peptides and proteins, and this can be used very effectively in drug design. And we can design peptides now that can cross the blood-brain barrier or not. And they can cross other cell barriers, including the gut and single cells and so forth, or not, depending upon what we want to do. We have methods for doing this now that we can use. So why peptides? Will they be the drugs of the future? Because nature has chosen the peptides and proteins to do everything, especially in animal life. All the initiators, modulators, and controlling bodies, most biological functions are controlled by peptides and proteins. And of course, do we get these from the nucleic acids and how they're controlled? And they're mostly controlled by peptides and proteins. So most of the ones that are found in our body have very low or minimal toxicities, especially when compared with current drugs. They have a broad spectrum of biologic activities, as we've already discussed, and we have the ability to infinitely modulate their properties in chemical and physical space. And this is very important because the biological activities depend primarily on their chemical and conformational and dynamic properties. Okay, so what can we do? What can we make? What kind of peptides can we make? Almost any kind that you want now. There's been tremendous developments in the last 40 years, especially in the development of all kinds of cyclic modifications of peptides. And I'm not going to go through this list, but I'm going to tell you that we could use almost all the power that we have from organic chemistry for synthesis, and we can apply these to developing cyclic analogs. Why cyclic analogs? Because cyclic analogs tend to be more stable. In fact, nature has used this with the disulfide bond. So nature has a way of stabilizing its molecules, and we have many other ways that we've discovered that are compatible with nature. And this lists some of them: backbone modifications, lactones, lactams, esters, ethers, cyclic carba analogs, backbone to backbone rings, end to end, side chain to side chain, and so forth. We can also modify molecules in chi-space. Most of you probably have not thought about chi-space, but all the amino acids except glycine have side chains. These side chains can exist in different conformations. Maybe you don't realize this, but these different conformations, as I'm going to show you in one example that we've done, and we've done many, many others, lead to changes in biological activity. You don't have to change anything about the peptide backbone or protein structure. So all you have to do is change the way you arrange their side chain groups with each other. So this is a very powerful tool that hardly anybody's using, but it's there. And then we have, of course, all kinds of other things like N-terminal modifications, beta-terminal mimetics, and so on and so forth. Plus, we can make peptide conjugates with almost all these cellular simple molecules: sugars, lipids, phosphate group, etc., etc. We can make places where we can have metals bind, and so on and so forth. All of this can be designed into peptides to give us the kinds of structural properties that we need for some biological function. And very interesting, in the last, especially 20 years, methods have been developed in our group and a few others now for multivalent ligands. That is, ligands that can have 2 or 3 biological functions, very specific biological functions that are necessary for some biological application. Okay. Another important aspect of design now is peptide membrane interactions. People have complained for decades about peptides because they don't cross membrane barriers. You know, they stay in the gut, or they don't cross the blood-brain barrier. This is nonsense now. It used to be true, but this is no longer true. We have found methods that in which we can make any peptide we want to cross membranes of various kinds, including the blood-brain barrier, the gut barrier, and various different kinds of cells, depending upon if we want to be on the outside of the cell or go into the cell. All of this can now be designed into the properties of your peptide. So the things that people complained about or think they still should be complaining about, these problems have been solved. Not all of the problems, but they've been solved in principle. And if you can solve some of the problems, which only some have been, then why not the rest by using these or similar chemical methods? Remember, we've had the molecular biology revolution, right? It's about molecules. Okay, so what are these new paradigms for drug discovery? Okay, so in many diseases such as chronic pain, cancer, diabetes, addiction, and so forth, there are changes in the ways in which genes are expressed. And these are often related to our treatment attempts. So therapies become ineffective. So what we have to do in drug design now is not only consider the effect, but the system level adaptations that are occurring. And this increases complexity. And so, but we have to consider these, and this is where peptides and proteins are particularly useful, because as opposed to small molecules, they generally do not cause these kinds of system level adaptations. And advantages then therefore can be taken of these changes using peptides and proteins. Secondly, as I said, multivalency is becoming very necessary and important, so that you want a drug that interacts with more than one target. This is very difficult to do with small molecules without making them larger and more complex and more difficult to understand. Targets can be nearby in biological space, or they can be separated very much in the body, and one can design to make them go either to a specific place or to several places. As I've said before, many diseases are multivalent, so you have to create multivalent ligands for these diseases. And what's interesting about peptides and proteins, generally when you make one, even if it's multivalent, it has a single metabolism. So therefore, you can predict how it's going to behave when it's in a biological system. So what I want to do now, and what I have in time, is to just give an example from one of the areas of research that we've been involved in. And this is one of the most important areas that has been developed in the last 20 to 30 years. And these are the melanocortin receptors. Now, a couple of the ligands for them, ACTH and alpha MSH, have been known for decades. In fact, ACTH goes back 100 years. But the receptors for them have only become popular in the last 20 years or so, and have only been cloned in the last 20, 25 years. Now, why is this system important? It is one of the primordial systems in animal life. It's found in all animals, insects, fish, lizards, humans. You name the animal, you find this system in it. It's not found in other living systems, just in animals. There are 5 receptors in this system, and they affect virtually all of our metabolic processes. So they're called the MC1R, 2, 3, 4, and 5. And you can see their functions here are fantastically variable. So the melanocortin-1 receptor, these are not a full list of their effects, and new effects are being discovered every week almost, it seems. You just read the literature. So it regulates pigmentation, hair color, pain. The MC2R receptor, this is fear, flight, glycolysis, stress. Okay? MC3 receptor, obesity, regulation of energy homeostasis, inflammation, sexuality, MCF4 receptor, feeding behavior, obesity and anorexia, sexual behavior, neuronal function, all kinds of neuronal functions having to do with neuronal diseases. And the MC5R receptor is involved in thermal regulation. That it's modulating the temperature in your body as you sit here and listen to this. And the sebaceous gland secretion, which is very important for many biological functions. So you can see this has a very broad spectrum. And the modes of regulation, some of them are given here. But the point is that this system has been around now for about 1 billion years, and it's found only in animals. And what's very interesting, if you look at the receptors and the ligands for them, which I'm going to discuss in just a minute, they have virtually been unchanged during this 1 billion years of evolution, which is very unusual. So therefore, their importance must be very high because nature has maintained their structures and their functions over all of these billion years. Okay, so now here's where you get the ligand from a single protein. Oop, what happened? I went backwards. How do I go backwards now? Oh, you want to go backwards? Okay. I'm sorry. Okay, I see. I'm right. Okay, so let me go backwards and not forwards. Okay, so here's this one gene. It's called pro-opiomelanocortin. And depending upon where it's processed in the body, it uses different molecules. Gamma MSH, alpha MSH, ACTH, beta MSH, and beta endorphin. As I say, these structures are very similar in all of animal life over the last 1 billion years, in flies and in humans. They're virtually identical. So here's their structures once they're processed. And you can see their amino acid structures of various sizes. ACTH is very selective for the MC2R receptor. The other 3 ligands interact with all of the 4 other receptors, but do not interact with the MC2 receptor. And unfortunately, all of these compounds are non-selective. So they have selectivities, but they're non-selective overall. That is, the selectivity differences are less than 10 in most cases for the different receptors. So if you want to design molecules, one of the first things you have to do is design ligands that are highly selective if you just want to interact with one or more, with one of these receptors. Okay, so what are some of their functions and biological applications? Well, I'm not going to go through all the functions, but if you take this, you look at the functions, and this is not a complete list, you'll see that it involves almost all of the basic biological functions that are necessary for your health and disease, for your health. You know, all of these things, if they're not working properly, can lead to disease states. Okay, so here's some of the medical applications. And again, this is just this is a short list. Enhanced pigmentation without sun. So you can protect yourself from the sun without UV radiation. And you should use these compounds instead of tanning salons, because tanning salons use UV radiation. You can pigment yourself without any UV at all. It doesn't make any difference how light or dark you are. You'll get darker if you use these molecules or derivatives that are stable enough to be used. Cancer prevention, memory retention, pain modulation, anti-inflammatory, repair of nerve damage, obesity, anorexia, erectile function, treatment of addictions, natriuresis, antiparesis, heart function, stress-related disorders, skin disorders, central nervous system diseases, and metabolic disorders of various kinds. So it's had a tremendous potential for drug design and development. We happen to have three of our peptides on the market, and they're about the only compounds for these receptors and so forth that are on the market yet. So there's a tremendous opportunity here, not only for ourselves, of course, but for the whole scientific community. Okay, now I want to tell you some of the things we can do with the melanocortin receptor. Okay, the first one of these, the first example I'm going to give is a very simple example of structure-activity relationships. And the modifications I'm going to make now have nothing to do with changing the structure in terms of the atoms that are involved in the structure. Is all we are changing things here are in chi space, not even the backbone conformation. It stays the same of this particular isomer. I'm just changing chi space for one of the amino acids, which is very important for its biological activity. This is known tryptophan. And we've done this by making all 4 isomers possible for beta-methyltryptophan. That's the only change we made. Add a methyl group for a hydrogen in the beta position and made all asymmetric possibilities. 2s, 3s, 2s, 3r, 2r, 3s, and 2r, 3r. And we put these then into the peptide, and we looked at the biological effect of these different amino acids. That's the only change that's been made, and the only changes we see are in chi space. Now, the chi space that's affected is not only the amino acid of interest here, namely chi1, and it can exist either as trans or gauche plus or gauche minus. And it does differently depending upon what is going on in the other at the other residues. So when it's gauche minus with 2s, 3s, it's trans at all the others, and so on and so forth, as you can see here. Now, what's so amazing about this, at least to me, even yet after we've done this several years ago now, is how much of a change in biological activity occurs. Based on NMR, there is no other change in conformation except the side chain conformations in relationship to each other. And yet, looking at a biological effect, okay, it ranges for an effect that can go on with 2R, 3R for hours, days, in fact. 2R, 3S, even after 3 hours, you start seeing a decrease. And after about a day, you return to normal coloration. And with the 2S, 3R and 2S, 3S, much less. So, with peptides and proteins, you can change a lot of things about their biological function by changing nothing except the relationships of the side chain groups that are important to the biological activity. This is a very powerful tool in drug design. We can, of course, show by conformational analysis using NMR, in some cases X-ray crystallography, that if you want to get different biological activities, let's say which Hsp9199, an antagonist activity versus a superagonist activity, what are the changes in conformation that occur? Just small changes in conformation, which you might want to design in chi space. Because if you notice, the backbone conformation is basically unchanged. It's all this changing again is the relationship of the side chain groups, and you go from a superagonist to an antagonist. So very small structural changes can lead to very large biological effects. And this is a very powerful tool in drug design, as you might imagine. Okay, now let's tell you what some of the things that you can do with these compounds at the melanocortin receptors. So this is using MT2, which is one of our derivatives for it, that's an antagonist at the MC4 receptor. And you can see that when an animal takes this derivative, that it eats less. See, this is just the cumulative amount of food intake in grams versus time in hours. These are free mice, and the number of mice in each case are 10. Okay, and this is the statistical variation that occurs when this happens. Okay, so what is the net effect of this? You lose weight. You eat less, you're going to lose weight. So these mice, normal mice, they lose weight. So if you're obese, you might want to take one of these derivatives. And just taking it, you'll eat less because you won't be as hungry, and you will lose weight because you eat less. So that's the idea anyway. Unfortunately, none of our drugs are on the market do this, but you can actually get one of our compounds on the internet that will do this. It's interesting. Nowadays, a lot of things are on the internet, in case you didn't know. Drugs that are being developed, you can get them on the internet. I'm not getting them, but you can. Okay, about a decade, more than a decade ago now, a decade and a half ago, we took one of our compounds also, which has an effect on erectile function. This is an agonist for the MC3 receptor. Okay, and the procedure we used was with Dr. Hunter Vessels, who's a urologist who was here at the University of Arizona at the time, but is now at the University of Washington, and Dr. Norm Levine, who's a dermatologist at the University of Arizona, now he's in private practice in town. I use him for my dermatological doctor. Anyway, so it was a double-blind experiment. The subjects served as their own controls. And what we did was measure the extent to which they would have erectile function. Remember, they have erectile dysfunction. Okay, 8 of the 10 people in this clinical trial had erections. This was done quantitatively by using measurements of erectile function that can be done quantitatively. So both doses were effective. Of course, the higher dose was more effective, and it generally took a lesser time for onset in these cases. So, the melanotropins can cause erections when given peripherally, even though it's well known that this effect is a central effect. That is, it involves your brain and your spinal column. So this is not related to the drugs that are given for erectile function, which have to do with enzymes. This is where sex really occurs, in your brain. And that's why this works so well. It isn't a drug yet, but it will be, I suspect. Okay, now I'll give you just a few other examples in the few minutes I have left of what other things we can do and have done with peptides. We have taken linear peptide hormones, made cyclic derivatives that are now highly selective for one of the receptors. For example, DPDPE, which is selective for the delta opioid receptor, and it's not active at the mu or kappa receptors. Convert natural hormone from one class of compounds into another class. And we made somatostatin into a mu opioid receptor antagonist, CTAP and CTOP, which are used worldwide to this day. We discovered these things over 25 years ago now, but everybody uses it. Why? Because they're antagonists and they cross the blood-brain barrier. So you can study what the mu opioid receptor is doing in the brain using these compounds. Okay, we can make unstable, short-lived hormones into very prolonged hormones. In the case of NDP alpha MSH, this has prolonged activity for days, whereas the natural hormone only is active for a couple of hours, depending upon the animal, for a couple of minutes. Its stability in the bloodstream, its half-life is less than 2 minutes. We made the first glucagon antagonist, glucagon inverse agonist, and the latter I think should become a drug at some point, because for reasons that I won't get into here. And we made the first multivalent peptide hormone neurotransmitters mu opioid agonist and a neurokinin 1 receptor antagonist, which can be used to treat addiction and tolerance. So we've done a lot of different things with these molecules. And of course, this has required an enormous amount of effort. I want to talk about my collaborators. I have to give several things here. So let me just give a general overview of the people that have worked with me over the last 50 years I've been here at the U of A. I have over 400 former undergraduate students, graduate students, postdoctoral fellows, visiting science, and professors from all 5 continents who have been in my laboratory. I now have former students who are professors on all 5 continents. I have over 50 collaborators worldwide that we have published with over the last 50 years. With 6 of them, I have over 100 publications. The first 3, Professor Hadley, Burks, and Yamamura, have passed away, but they were all world-class scientists. The other 3 here, Todd Vanderall, Frank Perek, and Min-Yin Kai, are still alive. They're still here at the University of Arizona, and we're still collaborating with them very extensively. And I've had over 100 publications with each of these people. It's absolutely, in some cases, 2 or 3 of them were on the same publication, but it's really an amazing accomplishment that we have had by collaboration, one of the most powerful tools in science. And it's not used enough, but it should be. And then for support, of course, we've had very good support from the National Institutes of Health, the National Science Foundation, U of A Tech Launch, several companies and several private foundations. So I want to thank you very much for listening, and I'll be happy to answer any questions.
Thank you, Dr. Ruby, for this fascinating prospect on peptide drug development. We have a question here for you. It says, peptides regulate most physiological processes, acting at some sites as endocrine or paracrine signals, and at others as neurotransmitters or growth factors. The problem is these peptides have been in the body to maintain homeostasis, so that they are exposed to the immune system, and the body produces anti-peptide antibodies that will make these peptides less efficacious.
Well, of course. I didn't talk about this, but this is something we've been studying also for decades. Of course, it's well known that peptides, especially peptides from other living systems, can cause immune responses. But we don't, and we sometimes get immune responses to some of our own peptides, especially if they get changed, or proteins, especially if they get changed biologically. So the immune system has a way of cleaning up. This is what the immune system does, getting rid of peptides and proteins that it doesn't want to have around. Okay, so what do you have to do? You have to design peptides that are not affected by the immune system. And we've learned a lot about how to do this. And we've developed many peptides. All the ones that I mentioned today are not attacked by the immune system, at least, for days or weeks or months, which is the most we've studied them so far. Now, whether they would, later on become attacked by the immune system if you had to use these for years is something we still haven't studied, nor have other people. We're not the only people in the world that have studied this, but this, of course, is very well known. And a lot of peptides that were made early on caused immune responses, and that led to another reason why lots of people sort of gave up on peptides. But that's no longer, we know a lot of things about how to avoid that now. So, and you just have to read the literature, learn.
All
So
right.
yes, it's a problem. It's a real problem, and you have to solve it when you design your peptides. Yes, you do.
Perfect. Thank you again, Dr. Ruby. We're going to move to the next speaker.
Okay, thank you very much.
Thank you. You can mute and
My pleasure.
Stop sharing. I'm really thrilled to introduce now Dr. Eric Ryman as our next speaker. Dr. Ryman is the executive director of the Banner Alzheimer's Institute. He's the CEO of the Banner Research, professor of psychiatry at the University of Arizona, professor of neuroscience at Arizona State University, clinical director of neurogenomics at the Translational Genomics Research Institute, TGen, and director of the Arizona Alzheimer's Consortium. He received his undergraduate medical education at Duke University and his psychiatry residence training at Duke and Washington University in St. Louis. He has played leadership roles in brain imaging, brain mapping, genomics research, as well as early detection and tracking of Alzheimer's disease biomarker development, and has accelerated evaluation of Alzheimer's prevention therapies. Dr. Ryman and his Banner Alzheimer's colleagues established the Alzheimer's Prevention Initiative to launch a new era in Alzheimer's prevention research. Eric, I've actually been in Alzheimer's field for many years. I've heard of Eric many times. I've seen his talks. I've always wanted to come to Arizona to be close to Eric, so here's my opportunity. He will speak today biomarkers, genetics, and the predisposition to protection from and prevention of Alzheimer's disease. Eric.
Thank you very much, Selena, and it's great to have you here in Arizona. I'm trying to share my screen as we speak. And I hope you can see it now.
We can.
Okay, great. So again, I want to thank you and the other organizers for the invitation to participate in this really terrific summit. and I want to congratulate the center on its progress to date. Today, I'd like to talk about a little bit brain imaging, cerebral spinal fluid, and blood-based biomarkers, genetic factors involved in the predisposition to and protection from Alzheimer's disease, and the implications they may have for the discovery, evaluation, and accelerated approval of disease-modifying and prevention therapies. These are my financial disclosures. Among other things, I am an advisor to several diagnostics and therapeutics companies. I am a PI of several NIH and other research grants, including NIH industry and philanthropy supported prevention trials in our Alzheimer's Prevention Initiative. Some of the discussion I will have will be relevant to an old patent we have, and some will be relevant to a company that we've just started to try to advance the role of blood-based biomarkers in Alzheimer's research, treatment development, and clinical care. As you heard from Dr. Dunckley, Alzheimer's disease is the most common form of dementia in older adults. It's thought to affect nearly 10% of people over the age of 65, between 30 and 45% of those over 85, and with a growing number of people living to older ages, the number of individuals who will be affected by this one age-related disorder will triple by 2050, unless we can address the problem now at a financially overwhelming toll to countries around the world. It takes a devastating toll on the affected person, an intolerable toll on family caregivers, and we all need to do what we can to address this problem as soon as possible. There's been great progress in the scientific study of Alzheimer's disease. I will illustrate some of that progress today, but the glaring exception to that progress has been the lack of success in the development of effective disease-modifying treatments. I will suggest why I remain excited about opportunities for the discovery of new treatments and their evaluation and approval sooner than you might think. So as Travis noted, the neuropathological features of Alzheimer's disease include amyloid plaques, the major constituent of which is a form of the amyloid peptide known as A-beta-42, tau tangles, a major component of which is a phosphorylated aggregated form of the microtubule-associated protein tau, and a characteristic loss of synapses and neurons in certain regions of the brain. There are other processes that occur as well, as illustrated in this figure with inflammatory changes, some of which appear to be protective and some of which appear to be harmful. For the last 30 years, the scientific study and development of Alzheimer's disease and the development of drugs has focused to a large extent on targeting the amyloid beta peptide, the major component of plaques. And when we developed this figure for a 2016 perspective piece, it was accompanying an early phase trial of aducanumab, an amyloid plaque reducing antibody therapy that was the first to show a dramatic reduction in amyloid plaques in symptomatic patients. I suggested that that if what was ongoing pivotal trials demonstrated a clinical benefit, that would be a game changer for the field, in suggesting that amyloid in some of its species had at least some role in the development of Alzheimer's disease. As many of you know, the pivotal trials for aducanumab were prematurely discontinued due to a pre-specified futility analysis and a premature declaration of futility. 6 months later, after having more data come in, the sponsor Biogen acknowledged the findings were incorrect, that indeed the treatment may be working, and aducanumab is now under consideration for approval by FDA, possibly with a decision on June 7th. based on one prematurely discontinued trial that showed a benefit, another that failed to show a benefit, and the possibility that the dose and duration of treatment may account for reconciling those findings. So FDA will need to be able to make a Solomonic decision that will have a number of consequences either way. In the meantime, there have been a couple of other antibody treatments that are plaque reducers that have also been suggested to show some of a benefit, which is nice to hear. But even before then, there were many of us in the field who have thought that even if an anti-amyloid treatment failed to work in clinically affected individuals, it would beg the question of what would happen if these treatments were started earlier, when people were cognitively unimpaired and before the disease has ravaged the brain. And that has been our interest in the development of strategies to accelerate the evaluation and indeed approval of prevention therapies. I'm a brain imaging researcher by background, and I became interested in this problem when I thought about Alzheimer's prevention. At the time we thought about this, nearly 30 years ago, we wondered, what if we had a prevention therapy that was available today, even a lifestyle intervention? It would take too many healthy volunteers and too many years to wait for enough people in the placebo group who were healthy, perhaps starting in late middle age, to go on to develop symptoms to see if the treatment worked. What can we do to accelerate that possibilities? So I became personally interested in the problem of Alzheimer's disease from the perspective of a biomarker researcher. And biomarkers have now had a transformational impact on Alzheimer's disease research and a growing impact on drug development. So we now have a number of brain imaging biomarkers, those for amyloid PET measurements of plaque deposition, tau PET measurements of tau tangle deposition, long-standing imaging techniques to provide indicators of neurodegeneration, for instance, FDG PET measurements of the decline in cerebral glucose metabolism in characteristic brain regions as shown on these statistical brain maps, and MRI measurements of brain atrophy is reflected by hippocampal and whole brain atrophy or cortical thinning. Brain images have had a profound impact in the field, but they are expensive and they are limited in their scalability. Cerebral spinal fluid measurements are also a little bit limited by the relatively invasive nature, although safer than people appreciate. And until the last few years, some problems with a lack of reliability when conducted in different labs or even at different times. There have been really rather significant progress in developing more robust cerebral spinal fluid biomarkers, including those that are used in the research and indeed clinical setting for indicators of amyloid plaque deposition, tau tangle deposition, and neurodegeneration, and a growing number of other changes, including inflammatory changes, other indicators of neuronal injury or neurodegeneration like NFL, or presynaptic or postsynaptic changes. We'd still like to see other cerebral spinal fluid biomarkers for other pathologies that can co-occur with Alzheimer's disease, particularly at older ages, but still somewhat limited in their scalability and widespread use. If you had asked thought leaders in the field 3 to 4 years ago, the idea of having robust blood-based biomarkers for Alzheimer's disease might have been considered something of a pipe dream, but there has been extraordinary progress in the development of blood-based biomarkers for Alzheimer's disease and related diseases in the last 2 years. These include plasma measurements of A-beta 42 to 40 ratios. When A-beta 42 is sequestered in plaque, CSF and plasma A-beta 42 goes down, and so does the ratio. And accuracy for in comparison to amyloid PET of about 87% in these blood-based biomarkers. Perhaps even more promising are recent developments, some of which we're involved in ourselves, of plasma p-tau. This is p-tau 181, which has been most commonly used, p-tau 217, which looks better, and I'm going to show you some data about, and also p-tau231. These are measurements of amyloid-related tau pathophysiology that have extraordinary promise. And commonly used for many years are plasma measurements of NFL, neurofilament light, an indicator of neuronal injury or neurodegeneration that can occur in Alzheimer's disease, but almost all other neurological disorders. Some of the advantages of p-tau and NFL are that NFL has been shown in studies with effective treatments for other disorders like multiple sclerosis and spinal muscular atrophy, a relationship between the treatment's biomarker effects and clinical outcome. And that's a big deal, to have biomarkers that are shown to be theragnostic. Similarly, in cerebrospinal fluid, the antibody treatments, starting with aducanumab, that have been shown to reduce plaques also reduce CSF p-tau. I can't wait to see the results for blood-based biomarkers in these trials. You don't have just a limited number of people who have CSF biomarkers, but you can look at every participant at every time point to see better correlation between a treatment's clinical benefit and their biomarker effects, so that we can go beyond guessing whether the treatment's biomarker effects are reasonably likely to be associated with the clinical benefit. We were very excited to participate in a study of 1,400 individuals in 3 cohorts, looking at phospho-tau217 and comparing it to p-tau181 in blood. These 3 cohorts allowed us to compare these blood-based biomarkers to amyloid and tau PET measurements in the Swedish Biofinder cohort, and appeared to be highly accurate. It allowed us in our autosomal dominant Alzheimer's disease kindred in Colombia to detect these changes more than 2 decades before the onset of symptoms. And it allowed us in our Arizona, our Banner Sun Health Research Institute brain and body donation program to show a very close relationship between the assay results and blood samples obtained in the last 2 years of life compared to their autopsy diagnosis. As shown it here, if you compare individuals with who have amyloid plaques and tangles that are spatially extensive beyond the temporal lobes, we call that high likelihood neuropathology, versus those who don't have it, the area under the curve is 0.98, incredibly accurate. If you include individuals who have plaques plus tangles that are limited to the temporal lobe, the area under the curve was 0.89. It distinguished AD from other conditions, including primary tauopathies. It was correlated with tangle counts in and only in those individuals with Alzheimer's disease. In addition to biomarkers, so with regard to blood-based biomarkers, the reason they're going to have such a profound benefit is they're relatively affordable, they're highly scalable, they're rapidly repeatable. You can go back to prior studies and prior trials and analyze samples with that. You can include more individuals from underrepresented groups who have been reluctant to have LPs or participate even in brain imaging studies. And I do think it's going to have a profound impact not only on drug discovery and development, but on clinical care sooner than one might think. Still a bit of work to do before it's ready in that regard, but an exciting time for the field. We now know of a number of genetic risk factors for Alzheimer's disease, many rare mutations of the presenilin-1, presenilin-2, and amyloid precursor protein that account for early onset autosomal dominant Alzheimer's disease. These mutations are virtually Alzheimer's causing, with typically a mild cognitive impairment at the average age of about 44 and dementia at the average age of 49. Down syndrome, which includes an extra copy of the chromosome that includes the amyloid precursor protein, has a similar pattern. The major genetic risk factor for Alzheimer's disease is not an Alzheimer's causing gene, but a susceptibility gene called ApoE. There are 3 common flavors of ApoE, 2, 3, and 4, giving rise to 6 common genotypes. And this is the best studied genetic factor we have, risk factor we have for Alzheimer's disease, and a big part of the studies that we do that I will describe. We know of a growing number of other susceptibility genes that have a more modest effect, unlikely in themselves to influence a person's care, but even a modest effect has been helpful in terms of thinking about targets at which to aim new treatments. And there's continued use, there's use of polygenic risk scores, both in people with and even better without the ApoE4 gene, to see if that could help to influence a person's risk. Again, I became interested in the problem of Alzheimer's disease following the discovery by Duke investigators led by my late friend Alan Roses, in which they showed in a retrospective case control study that each additional copy of ApoE4 in a person's genotype was associated with a higher risk of Alzheimer's dementia and a younger age at dementia onset. This finding has been replicated over and over. I would just caution you to note that this is a retrospective case control study, and there are more people than you might expect with 2 copies of the ApoE4 gene who live to older ages despite this genetic risk factor. And as we've been studying them, they provide clues about those factors and the related biological processes that might protect them from Alzheimer's disease and provide targets at which to aim new treatments. But when this discovery was made, I was simply interested in the idea of using this brain imaging technique, starting with what we had way back when, FDG PET and MRI, and now a growing number of tools, to study cognitively unimpaired late middle-aged people with 1 or no copies of the ApoE4 gene, to see if we could, what was an out-of-the-box idea at the time, detect and track Alzheimer's disease well before the onset of symptoms, provide information to inform the design and size of prevention trials, use biomarkers as endophenotypes, biological measurements more closely related to the predisposition to Alzheimer's disease than the clinical syndrome itself, to look at those risk factors, potentially to help identify potentially repurposable drugs, now that we can scale that with blood-based biomarkers, and help to advance the field. So for quite a number of years, we've been using biomarkers in cognitively unimpaired people with 2, 1 or no copies of the ApoE4 gene in the world's largest autosomal dominant Alzheimer's disease kindred in Colombia, and we also in related studies of people who are not chosen based on their genetics alone, but based on biomarker evidence of Alzheimer's. To give you a sense of this, amyloid plaques are found in about 25% of cognitively unimpaired people over the age of 65, and about 40% of those over the age of 85, and are thought to occur many years before the onset of symptoms. So over the years, we've done quite a number of studies with these techniques to detect and track these changes earlier than you might think, including developmental non-progressive changes unrelated to pathology in infants, infant ApoE4 carriers. And it's illustrated in this, these brain maps in looking at people in their 50s and early 60s who are cognitively unimpaired. You see the magnitude and spatial extent of amyloid plaque deposition in people with no copies, 1 copy, 2 copies of the ApoE4 gene. By comparison, you can see that in clinically affected patients with Alzheimer's dementia, by which time amyloid plaques had already plateaued virtually. We've used these studies, among other things, to suggest that we could do prevention trials in 2 years with biomarker endpoints with a relatively small number of individuals. We proposed that a long time ago, and we're very interested in that possibility. But there is a challenge with proposing that. The challenge is that FDA is quite aware that there are plenty of biomarkers in other disease areas in which the treatment's biomarker effects are not associated with a clinical benefit. And before you could not only inform the evaluation of treatments, but potentially get accelerated approval based on a biomarker endpoint alone, a surrogate endpoint, you will need to show in trials themselves that a treatment's biomarker effects are associated with a clinical benefit. And that might sound like a catch-22. If we had clinically effective treatments, why would we need the biomarker in the first place? And how do we get there? But we think there's a path to be able to get there sooner than one might think. And I'll tell you a little bit about that later. So we're always planning to do our first prevention trial, which turned out to be our second and third prevention trials at APOE for homozygotes and heterozygotes, close to the expected age at clinical onset. By studying those individuals, we could study people who turned out to be amyloid positive and amyloid negative so that we could look at primary and secondary prevention trials. We had endpoints that were adequately powered with biomarkers and cognitive changes in that group. But we decided for our first prevention trial to open the window open to doing prevention trials for the field, to secure a grant, to have a competition among companies, to use philanthropy as well, and propose a prevention trial in the world's largest autosomal dominant kindred. You might ask, where are you going to find enough autosomal dominant mutation carriers who are at virtually certain risk for developing Alzheimer's disease at a younger age if there are only 500 to 1,000 kindreds known around the world? And we have partnered with and supported a colleague, Francisco Lopera, and his team in the Medellin, Colombia area. So think of your largest family reunion, and now think of this kindred that we now have traced back to a common ancestor from the late 1600s with 6,000 living members, 1,200 people who carry a mutation that causes them with virtual certainty to develop Alzheimer's disease at a young age, 95% between the ages of 42 and 46 who develop mild cognitive impairment, 95% between the ages of 47 and 51 who develop dementia. So when one meets these families, you cannot help but understand the urgency when it comes to finding a way to treat and prevent this disorder, given the toll it takes on the affected person, the caregivers, the young adults who have to care for their parents, and the concern they have about imposing the same burden on their own children. So by knowing that they develop mild cognitive impairment at 44, median age of 44, and dementia at 49, you could track these biomarker changes and see when they distinguish carriers from non-carriers and how many years that occurs before the onset of mild cognitive impairment and dementia. And we've done that with a range of these different biomarkers of amyloid, tau, neurodegenerative pathology. And we've done this now also in much larger numbers with blood-based biomarkers. And you can see these changes more than 20 years before the onset of MCI and 25 years before the onset of dementia, which I see as windows of opportunity to intervene with prevention therapies. So we mentioned progress in the field, but we've had this challenge with regard to clinical trials. Since 2002, 99.8% of treatments have failed to demonstrate efficacy, not a winning proposition in drug development for that time. And there may be several reasons for this. One may be that one's aiming for the wrong target. Another may be that they're aiming at the wrong time before a patient has had the equivalent of metastatic cancer and the disease has ravaged the brain and that one might need to start earlier. We all recognize the need for a wider portfolio of promising treatments to target different elements of the disease. But we think there are other challenges as well. In drug discovery, you have the challenge of animal models that don't replicate all the features of this distinctly human disease. In human studies, while we can look at changes in the brain, for instance, we spend a lot of time looking at gene expression profiles as well as genetics in the brain. You don't know what's a cause or a consequence or compensation or coincidence. But I think with the emergence of new multi-omics data sets and big data analytic techniques that can begin to develop these multi-scale causal networks and putative drivers of these networks, and efforts to foster a push-pull relationship between folks working in human cohorts and experimental studies where you can look at causal connections in drug discovery, we're going to start seeing more success than we have if you limited your work to either human or animal studies alone. Further challenges, well, I'm a psychiatrist. We have drugs that work in psychiatry. And the discovery of those drugs was not based on understanding mechanisms. It was based on some serendipitous observation and the development of drugs that mimicked in one way or other those originally identified drugs, and developed biological tests to provide information before you got to a time-consuming, expensive pivotal trial. So I think if it turns out aducanumab and these other antibody therapies show a benefit, we will gain a lot more information about which of those biological changes turn out to be theragnostic, and we'll be able to employ them earlier stages. Some of those biomarkers are generalizable to the animal models and other conditions as well. So I think we will begin to have a more informed approach to drug development than we've had in the past. And one of the things I'm excited about is using very large data sets, psychiatry on steroids for drug discovery, I would call it, of using either large EHR data sets with and without large blood-based biomarker endophenotypes to find those treatments that have been shown associated with a slower decline over time to complement more traditional mechanistic-based findings. We have had this long-standing interest in trying to find and support the approval, affordability, and availability of prevention therapies, and my colleagues Pierre Theriault, Jessica Langbaum, now Bob Alexander, and I oversee the Alzheimer's Prevention Initiative, which was designed to do the studies in cognitively unimpaired people at genetic or biomarker risk, introducing new experimental paradigms and endpoints, forging relationships, public-private partnerships to overcome hurdles that one would have within one group, requiring data and sample sharing after the studies are over, and even baseline data sharing while the trial is underway to have the best possible impact, and helping others in the field to figure out better ways to test prevention therapies. Right now, these prevention trials, starting with our API, autosomal dominant Alzheimer's disease Columbia trial, which started in, we got funding for in 2012. That study will read out next year after quite a long time with a cognitive endpoint in that group, people close to their expected age at onset within 15 years. We painfully had to discontinue our API generation studies of ApoE for homozygotes and heterozygotes after enrolling by far the largest number of ApoE4 homozygotes known anywhere in the world due to an unexpected problem of a mild initial cognitive worsening that turned out to be reversible following discontinuation of treatment with all BACE inhibitors. And we'll have a little bit of additional information we'll provide about an active amyloid immunization therapy this summer in relationship to that trial. We have a funded trial that we're working on to start later this year with an industry sponsor of looking at one of the plaque-reducing treatments that might work in clinically affected patients, but studying them in cognitively unimpaired amyloid-positive individuals, and proposing the idea that we would begin with a 2-year trial. At least that would be the interim analysis. If by then the readout in the pivotal trial show a relationship between the biomarker endpoints of interest and a clinical benefit, we would seek to evaluate and support the accelerated approval of a prevention therapy, say by late 2025, with the same drug in the based on those biomarker endpoints, hopefully with a convergence of different biomarker endpoints and maybe some of the clinical endpoints at that point. If not, we would follow them longer with our cognitive endpoints in case the treatments were just too little, too late by the time they had symptoms. And we're looking at some other trials as well, one of which I'll tell you in a moment. We've developed mechanisms to find a lot of people, to engage research participants, and for instance, in our 350,000 person Alzheimer's prevention registry where people can learn about information, get information. Our roughly $100,000 per person GMATCH program where we do ApoE genotypes. Don't disclose that information, but connect them to trials. And we're working on blood-based biomarker development programs. And a lot of this is just highly collaborative. So I mentioned that we've had an interest in ApoE from the standpoint of enriching our studies, but we also have an interest in it from the standpoint for another reason. It turns out in Medellin, Colombia, we had, even though people were virtually certain they're overproducing amyloid from birth with this mutation, and they develop Alzheimer's symptoms at a young age, we have We had a person who did not develop dementia, developed mild cognitive impairment in their 70s, more than 30 years beyond the onset of symptoms. So my colleagues, Jaquel Quiroz, Joseph Arboleta, and Francisco Lopera and I, we did whole genome sequencing and a genomizer analysis to see if we could find a protective genetic factor. We found that she had 2 copies of a rare form of ApoE, the genetic factor that we associate with older ages, known as the Christchurch mutation, which could happen to occur on an ApoE 2, 3, or 4 gene. And like people with the protective ApoE 2 gene, some people with the protective ApoE 2 gene, the person had type 3 hyperlipoproteinemia. So she's been overproducing amyloid since birth. She has an ApoE variant that's protective. ApoE does a range of things, but the area that's received the greatest focus is its effects on contributing to amyloid plaque deposition. You might ask, what did her amyloid plaques look like? Well, it turned out she had the highest amyloid plaque burden in the kindred and still didn't have dementia. She had relatively minimal tau PET and neurodegenerative changes with imaging. And it suggests that ApoE may have a stronger effect, a greater effect than just another anti-amyloid treatment. So we're interested in that. Because this seems to be an autosomal recessive mutation, there's been a big question about whether ApoE and its variants represent a progressive gain of function. ApoE4 worse than 3, worse than 2, worse than nothing. Turn off ApoE, or ApoE4 in particular, or do they differ to the extent they're a progressive loss of function? And you should turn ApoE and ApoE4 in particular on. We're in the ApoE turn it off school, gene silencing school, but there's a big debate about it, and this provided support for that. And I'm going to suggest how we will put that to the test sooner in the next few years. ApoE, in related experimental studies, we found like the protective ApoE2 gene, it was associated with less amyloid aggregation. She probably would have had even more amyloid deposition if she was able to live into her 70s without that mutation. We found in terms of its binding to the heparan sulfate polyglycan receptor, previously reported difference between ApoE4 more than 3, more than 2, and ApoE3 Christchurch much less than the others. So we developed an antibody targeting the this binding domain, ApoE Christchurch binding domain, and replicated the same reduction in HSPG binding. So we're very interested now in the idea that ApoE gene silencing treatment, starting with ASOs and RNAis that should be available, we hope, if the toxicology works out within the next 3 years. We'd like to put that to the test in ApoE4 homozygotes, starting with those individuals, small number of individuals who already have elevations in p-tau and NFL, and see if we could drive it down in a short period of time, as early as a few months, and help to understand which direction to go. And we're also interested in HSPG and also its binding with ApoE as a potential target. This was just the experimental findings showing the reduction in heparin binding with the Christchurch isoform, and our ability to replicate that same finding by having an antibody target that on wild type ApoE. And HSPG has been implicated in a range of activities, but among other things, it seems to be a protein on the cell surface, on every cell, but including neurons, that is needed for uptake from one cell to the next by tau. And so in addition to the idea of an amyloid effect, we're interested in amyloid, ApoE's effects on the morphology rather than the magnitude of amyloid aggregation, its effects on inflammation, and its direct effects on tau cellular transmission. And we think that experimental studies like the ones we have in humans are the way to get there. So if this was so protective, what about people with the more common 2 copies of the ApoE2? It's not very common, 1% of the population of ApoE2. And could we show that ApoE has a more profound effect on a person's risk than people thought, motivating drug developments to target ApoE? So rather than looking at a bunch of people who you didn't know for sure, who were clinically diagnosed, but you weren't positive that they had Alzheimer's disease, and older controls, 25% of them who have clinical Alzheimer's, We looked at more than 5,000 individuals in which we knew the Alzheimer's dementia cases had Alzheimer's and the unimpaired controls didn't. And the impact of ApoE and its variants are much bigger than one would expect. So if you could turn somebody with 2 copies of ApoE4 into somebody with 2 copies of ApoE2, the odds ratio here of 0.004 is that you could reduce the risk, a person's risk, by as much as 99.6%. It'd be a good deal. So there are a lot of challenges with regard to ApoE drug discovery and development, but we think there is going to be some emerging opportunities, and it would be among the opportunities we have to diversify our drug discovery. I've mentioned how we want to put ApoE and other Alzheimer's disease modifying treatments to the test in people who are already elevated with p-tau and NFL and see if we can drive them down, particularly if those biomarker endpoints are associated with a clinical effect in the ongoing trials. I've talked about how we might get accelerated approval and use blood-based biomarkers as a screening technique once a treatment is approved for prevention, for screening people every 2 years rather than an expensive imaging technique or an LP. I've talked about our approaches to drug discovery. In the meantime, we have a woefully inadequate standard of clinical care. 60% of patients with dementia never have an evaluation. What if we could figure out how to provide a blood test in the clinical setting, refer for busy physicians, these individuals, to a virtual navigator to let them know about life after a diagnosis, with or without treatments, what's the prognostic implications, diagnostic implications, possibly the therapeutic implications, and begin to address that problem within the next 3 years. What I love about our research is that it's so multidisciplinary. It's a team sport, and we all have an opportunity to work together. And we're extremely grateful to our volunteers. We're extremely grateful to all of our funders, our colleagues and collaborators, and I thank you for your time. Thank you very much.
Thank you, Eric, for an amazing talk. I think Alzheimer's is relevant to everyone, so it's great that new things are coming up, and we have a light in the end of the tunnel for our future. We have a couple of questions for you, and we're going to go quickly here because we're already over time. One question is, have you looked at extracellular vesicles? I'm assuming the question is about exosomes as a source of Alzheimer's biomarkers.
There's a great deal of interest in exosomes. I have a personal interest in exosomes because my colleague Kendall Jensen's, her lab manager, is my daughter, and we're waiting to see how that works. We've seen some preliminary findings that look good, but the question ultimately at the end of the day is, how do we put it to the test in large numbers? How do we use older existing samples that is low-hanging fruit? How do we figure out how to scale it? And there are great opportunities, but not quite as far along as those other blood-based biomarkers just yet, or CSF biomarkers.
Yeah, and the other question is also related to exosomes, but now it's cargos to deliver, for example, RNAi against ApoE or other targets that might be relevant for Alzheimer's.
Well, I think there are some real opportunities in that space, and I think we're going to be learning a lot more about how they behave. There's also the issue of, while we're talking about cargos, about figuring out how to transfer both small molecules and large molecules into the brain. And with regard to tau therapeutics, our dependence on the idea that you're going to catch them at that moment, but there's this possibility, but not certainty, of transcellular spread and not get into the neurons, a bit of a challenge, but an exciting time.
Yeah. Thank you so much again for your time and a wonderful talk. I am going to introduce now, my pleasure to introduce our last speaker of the symposium, Dr. Cindy Miranti. She's a professor of cellular molecular medicine, leader of the prostate cancer research group, co-lead of the cancer biology program, and chair of the cancer biology graduate interdisciplinary program at the University of Arizona College of Medicine and the Arizona Cancer Center. Dr. Miranti earned her bachelor's degree in microbiology from Southern Illinois University, a master's in microbiology from Colorado State. She then obtained her PhD in biochemistry from Harvard Medical School and completed her postdoctoral training at Ariad Pharmaceuticals and Harvard Medical School. Her research interests are focused on cell adhesion through integrins, specifically how integrins control the interaction of tumor cells with the surrounding tissues, and how they influence prostate cancer development, drug resistance, and bone metastasis. Dr. Cindy will speak today about targeting G quadruplex in the androgen receptor promoter in drug-resistant prostate cancer. Dr. Miranti.
Thank you very much for the invitation to speak at this great summit today. This is a project that I started once I moved to University of Arizona about 4 years ago, trying to target the quadruplex in the androgen receptor. So hopefully everyone can see my slides. Let me verify that first.
We can.
Okay.
Yep. But it's actually, right now, we don't have the slides. We just have your Zoom screen.
Oh, okay. Let me reverse that. Okay, is that better?
Not yet. We still see your Zoom screen.
Oh, okay. Hold on. Let me share it. Sorry about this. Okay. Is that better?
Yes, now we see it. If you can put it, yeah, perfect.
All right. So, are you seeing the correct format?
Yes, but we can you put it in the, it's in the presenter mode.
Yeah.
Can you put in the, yeah.
Okay. How's that?
Yes, we got it.
All right. Thanks. All right. So, let me back. I think I'm a couple slides off here. Okay. So today I'm going to talk about this collaboration, actually with Dr. Hurley here at University of Arizona, and our attempt to target the androgen receptor. The androgen receptor is the major driver of prostate cancer. So first, I'll just provide a little bit of background on current prostate cancer therapies. So the majority of prostate cancer that's diagnosed these days is actually quite curable. So it's only a small amount, a few patients in which we actually treat aggressively for therapy. So the primary tumors, the localized tumors, which have not metastasized, are typically treated with surgery or radiation therapy. And for the most part, this is pretty much curable. The problem comes when we have patients that are presented with metastatic disease. So either they undergo the surgery and the radiation and then years later develop metastatic disease, or they actually have metastatic disease at the time of onset. And it's at this point where we actually start or begin to, targeted therapies or thinking about, alternative therapies. So, and of course, the first line of therapy is the anti-androgen therapy, or ADT, and that's aimed specifically at, inhibiting androgen binding to the androgen receptor. Now, unfortunately, inevitably, all these patients develop resistance to ADT, and this comes out as what we call castrate-resistant prostate cancer, or CRPC. In this setting, then, we have a couple of second-line therapies, so either stronger or better agonists of the androgen receptors, such as enzalutamide, or inhibitors of actually androgen synthesis, which is abiraterone, standard chemotherapy, and there's actually a vaccine out there, Provenge, which has some efficacy, and then some, again, more radiation therapy for the bone involvement. But overall, these are not curative. And so ultimately, these patients will die. And so they always die with this castrate-resistant disease. So the problem, the primary mechanism of resistance is the reactivation of the androgen receptor. There's a bunch of different mechanisms, splice variants, mutations, other signaling pathways that activate it, co-regulators, but the commonality of all of these is that this receptor is now able to function without androgen or very low levels of androgen, or uses other steroids. And so the problem is that the androgen receptor becomes reactivated. And so there's a need to find other ways to target the androgen receptor to overcome this drug resistance. And so this is just a diagram of the androgen receptor gene, and a majority of the targeting that has been done in this receptor is around this ligand binding domain, which is where the androgen binds and activates the receptor. A few companies have started to look at other regions of the androgen receptor protein than this nuclear domain, which is the transactivating domain of this transcription factor, as well as the DNA binding domain. But unfortunately, these have low affinity and aren't really proving to be very effective. And so the best target, or the best way to really deal with this might be finding ways to actually get rid of the protein altogether, not try to, let the protein even be made or degrade it in some way. And in fact, there are current therapies in the market or in clinical trials to target, siRNA or to destabilize the protein. The strategy that we've decided to go after is to try to understand, to suppress its actual transcription or expression. And this is really brought about by this sort of finding that the androgen receptor promoter contains about 100 base pairs upstream, a structure, what we call a quadruplex structure. So this sequence is demonstrated here. There's a sequence of 5 sets of guanines in triplicates, which is required to form a standard classical quadruplex. And when these quadruplex structures form within the few hundred base pairs of a transcription initiation site, this creates a structure that's resistant to polymerase attachment. So it prevents polymerase from attaching and transcribing through the transcription initiation site. And so if we could find ways to stabilize these structures using drugs, then one would presumably presume that you would then prevent or reduce the amount of transcription of this gene and lower its expression levels in the cells. And so what is the feasibility of targeting G quadruplexes? So there are over 300,000 or more predicted G4 structures in the human genome. They're enriched promoters, telomeres, and first exons, suggesting that they do have a major transcriptional regulatory effect or major regulatory mechanisms in the cells. And numerous classes of agents have identified that have G4 stabilizing activity as measured in vitro, but many are toxic and they are poor drug design. So really a lot needs to be brought forward to this field to really come up with more specificity and more thoughtful, careful thinking about how we design drugs to target these structures. Another aspect of this is that DNA damage is actually a site on which quadruplex structures can also form, or when quadruplex structures form, they contort the DNA in such a way that you may have an increase in single strand breaks. So there's a lot of association of quadruplex structures potentially with DNA damage. And the most characterized of the quadruplexes out there are those that are found in front of numerous oncogenes for obvious reasons. Obviously, targeting oncogenes is a huge area of interest to inhibit a lot of different cancers. And this is really exciting, particularly for these non-targetable oncogenes such as MYC and RAS, where it's been very difficult to develop good therapeutic targets. And this might be a way to inhibit these types of oncogenes. And at this point, we're arguing that the androgen receptor falls into this kind of category at this point. Okay, so has it been done? So there is actually one quadruplex binding drug in phase 1 trials currently, and that's CX-5461. And as I mentioned, sometimes these quadruplex structures are associated with DNA damage, and this is a case where a drug that is recruited to DNA damage or associated with DNA damage in the cells. And so the trial that's being undergone is based on patients that have cancers with known mutations in DNA repair genes. And so it's just a phase 1 trial, dose escalation. And in this trial, they had 8 of their 32 patients had some sort of response that had mutations in their DNA repair genes. Patients that had prior PARP treatment did not respond, suggesting that this would be sort of a first line or separate from a PARP inhibitor. These are based on quinolone derivatives. And the major side effect, and really the only major side effect, was this photosensitivity, which suggests, so the patients subjected to sunlight, get severe sunburns and their, sensitivity in their eyes as well, which would suggest that at least it's targeting in some aspects, some selectivity with respect to the DNA damage response. So this seems promising that this might have some potential in the clinic. So the most well-characterized G4 is this MYC AR G4 structure, and it alongside of the known structure of AR here on the slide. And what we can see about both of these is that there are 5 stretches of Gs where you only really need 4 to form a quadruplex structure. So the question becomes, which set of 4 of these Gs is actually being used to form the quadruplex structure? And for the MYC G4, it's known that there are actually 2 possible structures. One which is initiated with the second set of Gs, 1, 2, 3, 4, and creates a very tight and very stable structure, which there's only one nucleotide between each of the G plates, if you will. And alternatively, if you started and didn't use this middle G, you could have a second structure where you use the first sets of Gs and the last two sets of Gs, and this would generate a structure where you have a 6 base pair loop between the middle of these G plates. The androgen receptor similarly has these 5 Gs, 5 sets of Gs, and again the question is which of the 4 is important. But no matter which one you use, whether you use the first 4 Gs or the last 4 Gs, you always end up with a base pair structure, a huge loop of about 11 to 13 base pairs. And this is about the longest base pair structure that's been reported for a G4. So there's something a little bit different about the androgen structure, a G4 structure, for which this loop actually might be important and that we could potentially target. And so this loop structure actually presents another regulatory mechanism for our G4 regulation. And that nucleolin, which is a nuclear scaffolding protein, has a strong affinity for G4s with long loops. And in fact, this nucleolin binding onto these structures is required to effectively stabilize these long loop G4s. So this implies then that maybe there's something about, nucleolin that might be important in stabilizing the AR. And so this led to our predictive hypothesis that this A4 loop structure in the AR gene might be important for effective drug targeting, and that drugs that optimally stabilize this quadruplex either need to use nucleolin or will behave like nucleolin in some way to stabilize the structure. And that potentially we can use this loop structure to increase the specificity of nucleolin or of quadruplex stabilizing drugs. And so the first thing we did was to test the importance of nucleolin in AR expression. So in the first experiment, we actually just did a chip experiment to determine whether nucleolin actually binds to this structure in the AR gene using 4 different cell lines in which the androgen receptor is well expressed, as well as 1 cell line which the androgen receptor is not expressed. What we could immediately see is that nucleolin was able to bind to this element, but not to exon 1 region in these 3 cell lines that have the androgen receptor, but not in these cell lines that did not have the androgen receptor. We were able to show that if we knock down nucleolin in the cells, we can increase the expression of the androgen receptor message as well as its downstream targets, KLK2 and PSA. And then we could show using a luciferase reporter assay that the ability that knockdown of nucleolin increased AR expression, and this was dependent on that G4 structure. So if the G4 region was deleted from that promoter reporter construct, then it could no longer increase AR expression, suggesting that nucleolin was very critical for regulating the expression of the androgen receptor. So based on, based on that data, then we started to partner with Dr. Hurley to screen one of his new GSA libraries. And I initially screened 5 compounds and initially found this one compound, 0932. It was very effective at inhibiting AR expression and these 2 cell lines, as well as 4 of the known AR positives prostate cancer cell lines out there in the lower micromolar range. And at the same time, we also identified 1502 as a non-targeting, so it's unable to actually inhibit the expression of AR. This not only inhibited the protein expression, it also inhibited the message expression in these cell lines, and it also inhibited the target, the PSA target of the androgen receptor in these cell lines. So we also then showed that when we treat these cells with the drugs, so 1502 is our net negative drug that doesn't increase, suppress AR expression, and 0932 is the drug we chose to move forward with, showing that it has what it does then is recruit nucleolin to that promoter. So the drugs are very effective at recruiting nucleolin to that promoter. And if we knock down nucleolin in these cells, then the drug is unable to suppress AR transcription, suggesting that nucleolin is required for this drug to effectively suppress AR. We went on to show that this drug preferentially kills the AR positive tumor cells. So this is the kill curves at the different doses. And so PC3 and RWP are non-AR expressing cells. So they're actually more resistant to the drug, whereas the 4 cell lines here that express the androgen receptor are more effectively targeted. So some selectivity with respect to AR expression in these cells. And then we were then curious to know, what form of this AR quadruplex is stabilized by the drug. And so we tested AR2 versus AR3 as full length. And what we could see is that AR2, shown in this dark gray here, was more preferentially stabilized based on circular dichroism studies and melting temperature by the drug, whereas the 1502, which did not have a suppressive effect on the androgen receptor, was able to stabilize the AR3 structure. So this suggests that it is the AR2 structure that's being targeted specifically by this drug. And obviously based on the nucleolin studies, that nucleolin is very important for this process. So the next was to investigate the potential role of the loop structure. So we generated a bunch of oligos in which we sequentially deleted the loop structure in the molecule, and then again measured its stability using circular dichroism. So our initial full-length 11 nucleotide based loop has a melting temperature of 68 degrees. But as you see, as we increasingly decrease the number of nucleotides in that loop, we increase the melting temperature and increase stabilization of this quadruplex, which is more or less expected. So that long loop structure creates a not so stable structure, hence why you need nucleolin maybe to stabilize it. And then we tested the drug, and so the drug would cause a temperature increase of 8.6 degrees, but as we sequentially deleted these nucleotides, we started to see a reduced ability of this drug to stabilize this quadruplex structure. So suggesting then that this loop structure was really part of the ability of this drug to stabilize the quadruplex. So specificity. All right, so we continue to screen the library and look for additional compounds, and we identified two additional ones, 0903 and 1503, which had very similar characteristics to 0932. And so then we want to move forward with those three compounds. We also then tested a little bit about specificity. And so we first tested the MYC promoter to see whether MYC was being suppressed also by these drugs. And indeed, we do see that all three of our lead compounds are suppressing MYC activity. This is surprising and not surprising. It's not surprising in the fact that this library was originally designed to inhibit MYC-AR, or the MYC-G4 quadruplex, and that these two structures have a lot of similarity in terms of the loop structure or potential dependence on a loop structure. And so this could be a good thing and a bad thing, depending on how you look at it. But in prostate cancer, over 80% of the tumors are actually have MYC overexpression. So actually simultaneously targeting MYC and AR may not actually be a bad thing when it comes to the clinic. But of course, we don't know what other targets may or may not be targeted by these drugs. So there's still some work to do to figure out, the absolute specificity of our compounds. So these particular compounds are based on a quinoline analog synthesis, and that our groups, our most effective groups are those that have very long arms off of this R1 group here. This 1503 has some very strange properties. We've gone on to test it further, and so we probably aren't going to continue with it. But these two are initially two lead compounds. So just some of the pharmacokinetic properties of this drug. Tissue absorption is really good. It's quite soluble. Haven't had any solubility issues. And it's quite stable, at room temperature, in PBS. It's bound to rat plasma protein, but its half-life in liver microsomes is only about 10 minutes. So the compound as it currently existed would not be good for, oral dosing. So we can either correct that, or perhaps, as Ruby indicated, that maybe we don't want to give it orally. We did a half-life study in blood, and it has about 3.6-hour half-life blood. So it has a very short half-life. So potentially could be improved. Some improvements could be made on increasing the half-life, or we have to give it more frequently. The Cmax reached was about 2.8 micromolar, and this drug is effective, in the 1 micromolar range for some of our tumor cell lines. So we have the potential to reach some maximal activity in the bloodstream. And we also know that it doesn't cooperate with PARP inhibitors in the DNA damage mutations. So in that regard, it is significantly different from the previous CXR5431. So in summary, we have demonstrated that suppression of AR expression via the quadruplex requires nucleolin, and drugs that stabilize and inhibit this ARG4 require this nucleolin recruitment. And the AR2 version of the ARG4 quadruplex is stabilized most by our drugs. However, the concern still remains around the specificity of this targeting. So more chemistry and virtual modeling will be required to develop, a more true drug molecule. Future experiments will address this 11 base pair loop in this structure, the ability of these drugs to repress genes, how that gene repression is happening, and determine how oxidation of the G4 residues within this AR-based structure might be associated with CRPC. So prostate cancer is characterized by a high hypoxic and high oxidative environment. And if you oxidate, and in those conditions that would cause oxidation of these guanines, and oxidation of those guanines is going to interfere with the G4 structure folding. So we would like to know to the extent to which these drugs might overcome any kind of oxidative stress that might be going on in the cells and stabilize these structures even more. And I would like to acknowledge the people in the group. So this collaboration could not have ever, started without the help of Elsa Reyes. It was really her initiative and looking at role of nucleolin in prostate cancer that really initiated these studies. And then Lawrence Hurley for his valuable insight for all the drug development ideas. This has not been my area of expertise, and so it's been a lot of fun to learn about pharmacology. And then Vijay, who also has been very important in these studies. And then in my laboratory, Sorik and Nizabana are both people working in my lab who've been working on these actual assays. And thanks to the cores and the funding that's been supporting this, particularly to Tech Launch. Thank you.
Thank you, Cindy. We have a couple of questions for you. The first question is, do any of the compounds also bind to telomere DNA and telomeric RNA? Do you know?
We have not tested that. It's a possibility.
And then the second question is the effect of this drug hormone dependent?
No, it is not. It is not hormone dependent.
Okay, that's great. Okay, so I guess we're done with the summit, so with the speakers, and I'd like to thank Cindy one more time for a fascinating talk on G quadruplex. It's just, I got to learn a little bit when I was consulting for Regulonix, which I think is amazing field to be in. And also to all the other speakers that gave fantastic talks, and I'd like to thank them again for their time and for participating, and hope to see you in the future soon. I'll pass then to Dr. Wei, Wang, and Schnellman for closing of the summit.
Can we hear you? Oh, you can't. Okay.
Do you know what she said? It's for me now. All right. Well, we're finishing up a great day, and a little bit of laughter always is good. So I want to thank all of our speakers and our attendees for attending our ACDD program. We will have another one on October 5th in the fall. We hope that we can have it in person in Phoenix on that day. So please put it on your calendar. We also, that day is picked because Arizona Bio actually starts that afternoon. So there's a couple of different reasons then why you might want to attend. Again, thanks for our speakers, our attendees, and Selena and Wei for shepherding through the day. So thank you.
Thank you, the Dean. So thank you all. Particularly, I wanted to thank, Arnie, Liz, and Tony put this together. So thank you guys. See you on October. Bye-bye.